So, good afternoon and welcome to the Lou Guillette Jr. Memorial Symposium. Welcome to those in the room and those on the webcast. So I'm Jerry Heindel and I'm the director of HEADS, which stands for Healthy Environment and Endocrine Disruptor Strategies. And we're the proud sponsor of this symposium. And if you're in this room, you should know about HEADS and look up on the website, because our job is to keep people working on endocrine disruptors up to speed with what's going on in the field. So we're updating our website all the time. We put out a monthly newsletter and lots of other interesting, fun things to help you keep up with what's going on in the field. So this two-day symposium is going to focus on Lou Guillette Jr.'s impact on science, communication, and mentoring. As we all know, he was very unique, outstanding both in his science, his communication skills, and his mentoring skills. So over the next 2 days, you're going to hear a lot of stories from those who worked with and for Lou. This is going to be a wonderful trip down memory lane. So I want to start by thanking Nancy Denslow and Sarah Kaplan, who did all the work setting this meeting up, the venue, the hotel, the lunches, the reception. They made everything so easy, even though it wasn't at all. But we couldn't have done it without their help. And I want to thank all of you, where we didn't really want to have a registration fee, but we needed to raise some money. And I want to thank you all very much, because I believe we raised enough money to pretty much cover all the expenses of the meeting. So thank you all very much. And part of that was due to the University of Florida Physiology Department, which paid for the lunch tomorrow. Okay. With that, we'll get started, and we're going to start this first session with members of Lou's family. And we start with Teresa Guillette. Teresa is a senior environmental scientist at Arcadia, and she has a PhD from the Medical University of South Carolina. And of course, her dissertation focused on environmental contaminants and associated effect on crocodiles. So she's going to present kind of an overview of the life and legacy of Lou.
Well, thank you so much for coming today. I can't tell you how much this means. I met Lou in 2011 as a lady grad student and joined his lab in 2012. Fell in love with his son during that time and ended up joining his family during my dissertation. And this is a really hard talk to me, probably because another person was supposed to give this one, and it had about 3 days to actually put it together. But it's just very personal. And I think that's what's going to make it hard for all of you as well, because you're all a family. Colleagues, mentees, everyone. You have that ability to actually bring you into this sphere and make you feel like you were a part of his family, because you were. He considered all of his mentees and colleagues close family. So with that, I'll get started. So Lou got his education from New Mexico Highlands University. I didn't realize he was such an avid rock climber until looking at some of the old family photos and seeing, and I was like, oh wow, check him out. But it was just really impressive, because as Matt and some other people will talk about, he was really a Renaissance man and did so many things so early in life. He had positions at Wichita State University, which is a pretty funny story. He used to tell this one about bringing Matt into one of his seminars one time as a kid and talking about reproductive biology and going on, and Matt pointed out, hey Dad, dogs don't have eggs. So I think it was really nice that he was the kind of person to really bring in so many of that from his family and include them in the adventure. He started in University of Florida, Gainesville, where he did most of his just translational and transcendent work. And then finally became an endowed chair in marine genomics at the Hollings Marine Laboratory, which is where I met him and started in his lab. He had numerous awards, numerous accolades, and over 29,000 citations as of right now, according to Google Scholar. Actually, I did a kind of fun thing. So as part of my job, I do AI training and try and basically make sure that the models are understanding and delivering what they should be doing. And so I asked, okay, who was Lou Guillette and what was his contribution to science? And I actually liked what it said, what it spit out. It said his work was instrumental in advancing the field of environmental health and influenced regulatory discussions. But I thought that was so right, but also so limited. Because he was more than just his contributions to science. And I can see that here in all of you and seeing where everybody has landed, where everybody is still helping out each other, collaborating, working together, and that was his true legacy. So he used to really say this kind of thing with all of his students. He told us that as a scientist, you're an adventurer, a detective, and I think you all know the rest. Detective and storyteller, but I kind of wanted to break that down into little moments that I saw from where that came from. So first off was adventurer. The biggest smile that you would ever see on his face was when he was on the back of an alligator or taking an amazing photograph or looking at wildlife or hiking. He loved nature. His home was in the natural environment and he just had the most amazing time out there. This picture was taken in, I believe, 2011, we believe? 13, thank you. And so looking through some of those photographs, you can really see just this big grin on his face from every time. And it's so lovely to like go back to that and realize that he had such a passion for his work. And he really didn't think it was work because of that passion. So again, Lou started his career after a biology degree from New Mexico Highlands University. He worked as a wildlife biologist observing trend behavior in New Mexico, and then during graduate school, he approached Dick Jones at the University of Colorado to join his lab. Dick was very impressed after meeting Lou, mainly saying that he had built an incredible ability to form preliminary research questions with an unusual amount of excitement and a passion and enthusiasm to the detective work of creating research in biology. So he completed his master's in 1979, a PhD in 1981, with over 10 publications, which, 5 years for a PhD? Wow, like talk about impressive. I think many students now still see that and they're like, whoa, that's fast. But he just had such a passion that he was able to really take that and spark it into a lifelong research career. He started with the evolution of the reproductive system, particularly in sex determination, viviparity, and placentation, and set the groundwork which was to become a fantastic research career. So this is him in the lab, as well as him with some of his colleagues and the self-mentioned students. So that's Theo Coldborn and Howard Berg. So after a brief stint as an assistant professor at Wichita University, he was recruited to UF in 1985, where he set up his own laboratory in comparative endocrinology. As a self-described reproductive biologist, when he got a knock on the door to study alligator reproduction, he became really quite interested in the reproduction of the American alligator, and it was this basic research that really led to this important discussion and transcendent endocrine.
But really, his fundamental research was very much inspired, or as he'd like to say, thought of from Rachel Carson in The Silent Spring. So he had such a respect for her and her work that he actually named the dog after Rachel. And that golden retriever was the loveliest one that he ever had, I think, other than Charles Darwin. I will say, I also love that a lot about Lou, is that at his core, he was such a feminist. He had so much respect for women and so much respect for women in science, and I think creating that collaborative type of environment in a lab was just so inspirational and, to be honest, a little rare, especially for his time. So I'd like to thank him on that. Now let's get to the detective work. You'll see this picture come up probably time and time again from different slides. But he did so much research on alligators within the University of Florida research program. And he used to go out, kind of eyeshine surveys, do everything with a basic biology look. So he started kind of really from the basics and trying to understand what is normal, and then going out and seeing, okay, now that I've defined this normal, now that I know this, how then do I discuss and see what's abnormal in a system? And I think that's really powerful. His philosophy was that we could use sentinel species to watch and warn ourselves about what's going on in the environment. And so these are two of his more famous photographs. He loved taking pictures of osprey, and I learned a lot about photography from him, especially birds. He was actually really good at that, as well as the famous crutch one. And another interesting part about the detective aspect of Lou was that he didn't just do the normal thing. He really turned toxicology on its head. So toxicology is this idea that dose makes the poison. And that there's a lethal versus toxic. But I think he was very inspirational in his idea of it's not only the dose makes the poison, it's the timing of the dose. It's where it's happening in that organizational system. And that acute high doses are not always predictive of chronic. And that high is not always predictive of low. So there can be different mechanisms meaning different outcomes. So there's definitely a difference of dose ranges. And in that, he really used a lot of our basic concepts of endocrinology to kind of turn that idea of dose makes the poison on its head. So we know now, and we have known not only from his research, but building on it, that there are different things that we can say from both toxicology and endocrinology, and bringing the two together is that intersection can be very powerful. I always love this slide of his, and it always reminded me that we can't really discuss what's happening in the alterations of a system without first knowing the normal. So understanding that basic biology and basic reproduction. So his main hypothesis starting this work was that the increased mortality of alligator embryos is due to altered ovarian development, or oocyte health, not necessarily the toxicity of hormones present. And you'll see throughout this presentation so many of his pictures because, as I mentioned before, he was an amazing photographer. So this is basically looking at alligator egg through a lot of the work that he did, starting from both the egg to the animal to the ecosystem. And so one of the main things that he found was that a certain lake within Florida had an unusually large amount of DDT, so Lake Apopka. Now we know from looking at different things such as reference sites that there are other contaminants present, and mixtures might matter, but these were the main driver of the work going on. And so his highly cited publication from EHP in 1994 actually looked at this and looked at the oocyte and multi-oocytic follicles in Lake Apopka versus the reference site in Lake Woodruff. And he was always very careful to tell his students that it's not necessarily a control and a dose, because what you're doing is studying wildlife. You can't control the dose there. So you have to look at reference populations of maybe high and low contamination to understand what's going on, because we're not just going to go dose babies. Well, human babies did dose an alligator once. But as I mentioned before, he was so collaborative, and so he worked with several different people to really understand what was happening in both mammalian systems as well as what he was seeing in the alligator, and tie that together into something really, really special. And you can see the different ones as far as looking in both Tyson's work and a couple of others, how there is a lot of overlap between them if you actually looked at it. Some of his main findings within the polluted lakes from Florida, looking at the basic biology of the alligator, was that the differences in the lakes were low density of juveniles, low egg viability, elevated pesticides, altered sex steroid hormones, altered development, and the reduced phallus size in juvenile males. And I will say that there's been so much work spurred on from this. Really excited to see a lot of the both epigenetic as well as more transcriptional work coming out. I think Matthew Hale's going to be here tomorrow and did an amazing study on that. And he also had a lot of inspiration from others as well. So the wing spread statement and getting together with that group of physicians, reproductive biologists, wildlife biologists, all together to bring out this certain hypothesis, which was a large number of man-made chemicals that have been released into the environment have the potential to disrupt the endocrine system of animals, including humans. And I think now this is so accepted, but back then we really had a very limited view of what the environment was actually doing to us. And it's really crazy to see that now and how chemical regulation has come to the forefront. I mean, we're hearing about emerging contaminants now in popular media like John Oliver, things like that on PFAS. So how the public has embraced this concept that chemicals can affect your health is nothing short of astounding, and this work really helped lay the foundation for that. He was also really great at kind of bringing the science of the alligator and what was happening there and translating that into humans and what that actually means. And helped contribute to the understanding of the developmental origins of adult disease, as well as contaminant exposure and women's health. And it's really the idea that your environment during development is so important for you later in life. So functional changes can really disrupt that developmental programming and alter those things later. As well as, he liked to say, a bad start lasts a lifetime and maybe several lifetimes. Now we're coming on to the artistry part of it, which you don't really see a lot through his photos. It was really fun to watch him go out and take some. I think many of you have been inspired by that, like myself, and are also photographers and love to do that. But it was just so fun to watch him go out and find that perfect shot and get it. I love seeing that smile on his face when he knew he got the great shot. One funny time is when we were doing some field work in Africa, him and Hannes were going to meet us at this one field site. And he was so excited to show us he got a leopard in a tree. But then, as he looked to his left, a couple of tourists right next to him got it on an iPad. And so here he is with his $10,000 camera, getting like a perfect shot, and they actually had a little bit better photos because they were closer with the iPad. So he was a little mad at that, but at the same time, just a tiny bit competitive on photography. And you can see a bit of that in some of his work and his talks and how he brought that to life. He did used to always use a lot of great visuals because I think he realized that pictures worth 1,000 words. And so these are some of his from various different fieldwork. I am so happy to upload and share so many of his photographs. Please let me know if anybody wants any. I'll be putting them on a Google Drive, just taking a while because there's probably about 4 terabytes of data. But here are a couple of great ones. There's one of the leopards that he was so happy to get, the Nile crocodile, and then there's Matt and Russ looking at a herd of elephants in Kruger National Park. As I mentioned, he used a lot of his photography to make very impactful statements. And just watching him talk, watching him give a presentation was a treat. I think so many of his mentees looked up to him that way because as you get into the field, you try to learn your own voice and you try to learn your own speaking style and things like that. And he was always he was happy to let you be yourself, which is something rare in a mentor. Something incredibly rare that they don't want to make you an image of them, but they want to make you be the best person of yourself. I have so much respect for that. Because he could talk to Congress. He can tell them all about their phallus size and make a joke about it. But he could also talk to groups of students while on the back of an alligator having a grand old time. So I loved that his charisma and his personality and his communication style was so open, so welcoming in a way that he could make you feel like you were the only person in the room. And there are a couple more ones of his beautiful photographs, as well as something that he always used to like to tell his students, which is that association isn't causation. And I think we all appreciate that and know that there's a lot more complexity to these things than we might have realized. But I did just absolutely agree in the fact that, in science there are no certainties, but we can only work towards something. This was a couple more of his pictures and fieldwork in South Africa, as well as some other ones. And then I actually want to end with his thank you slide, because he was always very effusive and thankful for his mentees, colleagues, and people that he surrounded himself by. In fact, one of his words on legacy was that it wasn't what you did, it was the people that you left behind. And I think that's how I try to live my life. It's not necessarily what I do, what my career is, what I'm doing that way, but the people that I can inspire, mentor, and live on. With that, thank you.
Thank you, Jerry, and thank you to everybody for making the time out of their busy lives and schedules to be here. I've been looking forward to this ever since it started being talked about, and I'm really happy to be here today with you. I know that we have all gathered here because of my father's scientific contributions, laurels, what he did in the field, but I think more of us are here to celebrate the man, who he was to us, what he taught us by example, what he showed us how to look at the world differently. Very beautiful. So I was able for decades to watch while Dad mentored and worked with his colleagues and really see the special relationships, things they discovered, and the love that they shared. It's really been one of the greatest joys of my life to see my dad around his academic family and see how much he treated them like children. He would protect you when you needed it. He would help you find your own path to your goal, to chase your dreams, to change your circumstances. basically just to grow. And I saw that by being around him and then by being around the community, people could grow in ways that they never expected when they came into the lab as a grad student or for collaboration or anything like that. Okay, so with the slides. So here's that, and this, Teresa obviously talked about this already. The combination of Dad's scientific and artistic skills is what made him such a captivating speaker and presenter. He taught that a good story oftentimes was better than anything you could rehearse and put on paper. So Teresa touched on the artist a little bit. This is actually a picture of Dad, very close to an alligator. I know he has the telephoto lens, but he was really close. We all had like stuff while he was taking this picture, just in case. But this is just how he saw the world. It's reflecting in an alligator's eyes, but he could see it through gator's eyes, other animals he worked on, other people he met. I mean, his communication set him apart. You could have the most complex idea, and he would be able to break it down to whether it was an elementary school student or a professor that's an expert in a similar field. And I loved getting to see him work with so many people from different fields and entering the medical world and being recognized there, working alongside vets. I mean, teamwork is making the dream work, and quite honestly, that's what we're looking at today. Everybody in this room and everybody that couldn't make it doing the hard work. All right, so little fun one here. This is the three of us, Russ, Dad, and myself in Belize. Picture taken by Dr. Thomas Rainwater. And the funny thing is that Thomas was expecting during this trip. So we were waiting on the raindrop, AKA Talon Rainwater. So honestly, Dad's trip's there, and when Thomas was great enough to let me join, we really fell in love with Belize. Not only to follow up Thomas's work, I work with people in Belize currently that are continuing Thomas's work and Dad's work, and they're just huge heroes to the young people that I mentor, and I really love seeing that. It makes me really happy. So I think Teresa touched a little bit on this, but you know, Dad was one of the first people to really talk about how the environment played a role. It wasn't just genes and germs, and it wasn't just really even he was just very untraditional in the way he thought about things. And like Teresa said, he wasn't just worried about the people being exposed. He knew that it would last for generations and that the work could not be done in one person's lifetime, which is why it's so wonderful we are gathered here. Pictured here is the Wingspread Statement reunion, same thing that Teresa touched on. And let's see, we also talked about you can't understand normal without understanding abnormal. So Lou believed that understanding normal in nature was very different than understanding normal in a controlled laboratory setting. So we really had to go out and find not a control, but a reference site where fecundity and animal health and the offspring's health would be towards the peak of what we find in other areas that are undisturbed. And then we would relate this to different contaminated sites with obviously complex signatures, which is great because I got to see my dad learn a lot of chemistry and just really integrate complex knowledge through the years and share that knowledge with everybody that he touched and knew and mentored. So let's see. All right, so here are a couple of sentinel species. Dad actually worked on many different species because they all had advantages and disadvantages, of course. So birds, gators, marine mammals, sharks, because they're global, versus gators and crocodiles, they're very dedicated to one site, let's say. And here is one of his favorite iconic slides where two miners took a canary down in a coal mine to test for methane, and it dies. And they're like, this means something. I wish I could remember, but I think their mine's already being affected by the methane. Anyways, it was just great. It was a model for how we use our various sentinel species. And of course, he taught us not to overstate our science that association is not causation. He was just very creative in how many angles he could look at things from and really tease apart the complexity of the field that he fell in love with. And I really do think he fell in love with it because there were so many moving parts and so much to learn. And he was his happiest when he was learning and teaching. We covered the alligator eye shine, but one thing is I just loved going out on these nights with Dad. Seeing all those eyes staring back at you, I mean, this is how it started. Before you can describe normal, you need to develop a way to do population counts and figure out the distribution of that population, especially when doing reproductive work. So this is one of his iconic photographs, and you can actually look at size classes, numbers, and him and Woody and several of the other FWC people learned how to properly take counts and census of populations and monitor them, and that's done all across the US and the world today on different species. Okay, Teresa touched on this, so I'm not going to really do that. She explained it better than I could. Same for that. Okay. So, the fun part, of course, for me, it was always being with Dad in the field whenever possible. He was just a big kid and a great energy to be around. And when I was out there, I didn't feel like I needed to grow up, since Dad really didn't in some ways. But this is an aerial photo of a nest. We would usually find them from a helicopter. Sometimes we just bird dog them on foot, which was no fun. Today we have drones, so we are working on that technology, of course. But here you see a guard pool, mama gator defending, and the mama gators, let me tell you, my dad and I were always a lot more concerned about those than the bull gators, the slow big males. They were testosterone poisoned and not dumb. The mom had a plan on how to get you. So here's Dad collecting eggs, and what you don't see is somebody guarding him in case Mom pops out. Okay. Okay, so final slide of his to share. This is sometimes what he ended talks with, but it represents the fact that what Dad was really working towards as a reproductive biologist and ecotoxicologist was to have healthy moms and babies, whether it was alligators or humans or really any species. He thought we need to preserve that. And today I see so many people working towards that goal, which is honestly more worthwhile than ever. Okay, that is my last slide, but as we all know, like everybody that met Dad, he was a special person. To know Lou was to love Lou. I really believe that. And seeing him teach and break down complex things was impressive. But seeing the love that he shared and the people come back today and the spirit, his spirit that they carry with them, is really what gives me hope for the future. And that we have so many capable individuals that have mentees of their own, and for that I really do think that Dad has achieved legacy, because I see everyone supporting and nurturing the next generation, because that's what it's going to take. And that's why I'm so happy to have so many new young people I can meet doing work that honestly we didn't have the technology or the instrumentation to do when Dad was alive. So, like many words, Teresa touched on this, but legacy for Dad was the people he left behind and the lives that they had enjoyed, shared with others, the science they did, the way they went about the science, the way they looked at the world. In the past decade, it's become really difficult. I don't think that Dad could have predicted where we are now when he passed, and I don't think any of us could. But I do know that if he was still here, he'd be doing everything he could for the academic family, the young scientists, but he has so many people carrying that torch and doing everything they can. We truly have a strong academic family that has helped each other over the years, both with successes, failures, hard times, personal things. So unfortunately, a lot of federal employees couldn't be here today, and that is very sad and frustrating. I know they wanted to be here and share their own truth and stories, but it is tough times right now. But I know that if Dad were here, much like all of you, you know that you can't give up the fight. You know that it means more now than ever, and that if we are quit or cowed, then our future might not look as bright. So, to the future, and let's see, my mother and Tammy, I will introduce them, Dr. Buzzy Gillette and Tammy Mandel. They're going to come up and share some stories and words about Lou.
Hi, thank you all for coming, and I feel really privileged to talk about Lou, my husband. We first met right after I was divorced. I had 3 young kids, and they all had multiple pets. I believe me, multiple. And I got a job as a camp nurse at a YMCA camp. Of course, girls are not supposed to be at boys' camp. But those kids joined in anyway, and no one complained. And Lou was the outward bound instructor. And he saw me with all those animals, and he thought, someone with so many animals must be okay. So we became friends. And all that year, we developed really an in-depth friendship. I think that's the best way to describe it. So the next summer again, I was a camp nurse, and Lou was the outward bound instructor, and the 3 kids were there in camp. And they all had fun playing and interacting with Lou. And our relationship slowly developed into love. And at the end of that summer, we decided, let's go to Colorado together. Sounded like fun, so we went up to Estes Park for a few days, and then stopped by to see my sister who lived in the mountains outside of Boulder. And I really liked it. And she said, there's some houses for sale up here. Well, by the end of that weekend, we had bought a house. Lou was in school in North Carolina. I moved out there with the children, and he'd drive up on weekends with his guitar, and he'd sing songs to them every evening. And we just had a good time together. So we decided when we got marriage was coming up, 1976, and he decided, we all decided, he should sing during the wedding ceremony. And the song he picked out was John Denver's Follow Me. And I don't know if you remember that song, Follow Me Where I Go, What I Do, and ends with, I will follow you. Excuse me. And this became the basis of our marriage. Throughout our 40 years of marriage, we would support each other and give encouragement and new ideas in both directions for our research and for our work, our hobbies, or whatever. It soon became known that we had likes and dislikes. And how does this fit into Follow Me? Well, Lou realized I really disliked animals, picking up animals that lived in water, or lizards, or snakes, or things like that. I just didn't want to touch them. Well, I must admit this. One time when we were out, Lou's students put a baby alligator in a bucket of water and said, Buzzy, you gotta pick this up. And I said, no way. And they said, yes, you gotta pick it up. And one of those students happens to be here today. I won't say who. I closed my eyes and reached in and picked up this alligator, and they all laughed, but at least I didn't. Well, Lou recognized I didn't like to do this, so I was the official note taker for a lot of his expeditions. I'd sit there and take notes on how long the alligator was, how long his phallus was, and so forth and so on. And that was really important. And I recognized that Lou did not like to interview or talk to people. But, excuse me. I did my PhD research in Botswana, interviewing elderly for what was going on, the situation of the elderly in Botswana. And Lou just could not do it. He did not like it. But he offered to drive me any place I wanted to go. So he drove me to towns and villages all over Botswana without complaint. I'd hop out of the car when we got there, go talk to the village chief, maybe have a dinner, get a nice view of the whole village, and all kinds of information. Lou would just patiently sit in the car till I came back. Then we go on someplace else. That was his job, to drive. So we built on each other's strengths and weaknesses as we followed each other through our journeys. Then, throughout our marriage, we always had a blend of interests, particularly the role of contamination on animals and people. And in this way, it was really good because we could throw ideas off of each other and help each other with our research. Sometimes things, hey, that won't work. Other times, yep, maybe that will work if you do it this way. So the idea of follow me where I go, what I do, and I will follow you, worked very well with us. And with that, I want to thank you for listening, putting up with this life. Who still gets emotional about Lou. And I'll turn this over now to his daughter, Tammy, who will talk about Lou.
I've got a tough act to follow. And I think I'm going to have to keep my emotions in check too. So if you see me getting teary-eyed or trembly-lipped, make a face at me or something. So as to talk about the early Lou, how do you encompass Lou, right? Somebody as big as Lou in a short presentation. And so I wanted to give it a little bit of structure. So I thought I would build it around things that we're all so familiar with, which are some of his expressions. And because truthfully, these were all blossoming in Lou. Oh my gosh, oh, here we go, long before he started using them in science. And so my mom had referenced how they met at Camp Lincoln. This was in New Hampshire. And Camp Lincoln at the end of each summer would send out a little, summer yearbook about, they had collages and pictures of everything that had happened and quotes from the campers. So I just snapped this picture because you can see at the top there's my mom with my younger brother John, my sister Kaya, and myself. And then over to the right there, you see Lou. Yes, that is Lou, with a camper at Camp Lincoln. And also, within this Lincoln log, is what they called them, were captions from the students. And, Lou had that Tom Sawyer quality about him, where he could get people to do the dirty work and still make it seem like fun, and they'd still walk away from it loving in the end, right? Like, how many times did you listen to him present? And almost every time, when people started asking questions, one of the questions would be, how did you get the alligator eggs? And he would always respond, that's what students are for, right? And not that he didn't love to do it himself, of course, but I know this is all very small text, but in this, what you saw were campers that came, they had the messiest cabin, they were rowdy, and by the end of camp, they had the best cabin, it was clean, and right, Lou had a way of just inspiring people to do things, get things done, and still love camp loving him. We all absolutely loved him at Camp Lincoln. Oops, sorry, wrong way. One of the first adventures we did outside of campus, he took us hiking up Mount Washington, which is a mountain, it's in the White Mountain Range in New Hampshire. And it's known as being the top of the peak. It's the only place on Earth where they've recorded the fastest wind speeds on the planet. And the weather changes there almost instantaneously as well. So we started out in the morning, nice warm summer morning, trekked up that mountain. I have two distinct memories. One is, if ever of you have been a kid walking behind an adult on a steep incline, all you really see are the back of their legs and their feet, right? The whole way. And we're climbing up, and I'm watching his feet, and I'm watching his legs, and I had that sense of security. I watched where he carefully put each foot, and we climbed the mountain, and I just thought, I feel so safe with this person. And when we got to the top, the weather had drastically changed. It was cold, it was windy, and we were waiting to actually ride the cog railway back down. And so it was coming down on us. You can see in our expressions, we are not having a good time, right? So, any guesses here in the audience? What did Lou say? Yes, it builds character. And that is something that we heard and continue to hear, right? Always, right? This true belief in, let's take on our challenges and grow from them and not complain. Let's see what we can do about it. So of course, we had many more adventures with Lou. This is just a few that I just threw in there. I don't want this to be you guys feeling trapped looking at your neighbor's family vacation photos. But, we adventured to California. We went to Israel. During the summers in Colorado, we'd go sledding down the glaciers. Always with that classic laugh, that Lou laugh, right? That just, made everything magical. And then as my mom mentioned, we did move to Colorado, and there was that gap year. And, but Lou was never someone to stand still. So he did work professionally as a carpenter at that time, became very involved with the community. He and my mom became EMTs for the Sugarloaf Volunteer Fire Department. That's him driving the fire truck. He built a privacy wall for their outdoor bedroom. He built an amazing shed for my horse. And always living life with a sense of purpose. And where's Dimitri in the room? Because Dimitri, at one of Lou's memorials, did the best imitation of Lou walking down the hallway. When Lou had someone he wanted to talk to, or he wanted to get something from another lab, he didn't do this. He did this. And honestly, that's something that I've incorporated in my life as well. I call it walking with a purpose, and people who know me don't always appreciate it because I tend to walk too fast. Okay. Oops. There we go. So this slide, in reality, could be a collage of everybody here in the audience. Lou was all about collaborations. Rather than trying to get all of your pictures in here, I just put in a sequence of him and my mom, because that so represents his philosophy on the value of working together and how much more you can do when you work in collaboration. And as a teenager, I once asked him, right, a young teenager, how do you know if you're in love? And how do you know if it's a good relationship? And he said, well, Tammy, think of it this way. 1 + 1 should equal 3. Each of you on your own are your best persons. But when you come together, you create something so much more. And when you find that, that's love. And I think that was his philosophy as well when he worked with, right, all of his collaborators. He loved, right, sharing ideas and building ideas you all know this, right? Preaching to the choir here. Okay, you might have to make a face at me on this slide. Lou also always said, let the science do the talking. Because he knew that if he wanted to communicate a message, the best way to do that was to back it with the evidence and the facts. And with your actions. And so here I have let the love do the talking, because, let me take a quick drink here. I promise it's just soda. Lou was very expressive, very verbal in telling us how much he loved us. But the truth is even if he had never said those words, we knew. Because everything that he did for us, with us, expressed his love. And, forever I will feel like the luckiest child on the planet to have had him as my father. And I have no doubt he was the same, and I know I saw it with his grad students and his colleagues and friends. And as Lisa alluded, we are all family. And so I am hopeful that you still feel that love, from him and from us, who so appreciate everything that continues. So another thing with Lou, right? What is the question? And I know I heard him say this so many times, right? In his presentations, no doubt with, every one of his students and colleagues, because, in that case, he really meant it as, if we're going to design a good experiment, we need to know what is the question, right? So that we can actually create something that's going to give us the answers that we're looking for. And, but at the same time, when I hear that, what is the question, it also just reminds me of how he loved to cultivate curiosity. And so here he is. I know I've jumped forward in time a little, but with 3 of his grandchildren. And we were at my brother's wedding, and of course he caught a frog. And I don't know if any of you ever heard Lou imitate a frog ribbiting. It was magical. But again, I just wanted to throw this picture in because it's just so representative of how he caught this frog. They're all very, very timid, but eventually he had them asking the questions and touching the frog. How many of you here have been to a Darwin party? Oh yeah. How many of you here went to a Darwin party and then went again the following year and brought a friend and deliberately didn't tell them there'd be an exam about Darwin? What? No one's daring to raise their hand? I know I did. One of the things about Lou is that he could bring people together under controversial situations and have the conversations. And this is actually the last Darwin party that we had, and 32 years, right? I think we went through 4 different addresses for Darwin parties. And I believe starting in Kansas, we had actually moved from Boulder, Colorado to Wichita, Kansas, and the culture shift was pretty amazing, going from a very liberal, anything goes environment to more of a, yeah. Enough said. And I know, right, so Matt and Teresa, and I know all of you, right, the legacy of a mentor, right, that was something that was so important to Lou. And I had the privilege of watching him with his mentors. When I was a teenager after school, instead of taking the bus home up the mountain, I would walk over to his lab, actually Dick Jones's lab, and he would put me to work. I measured lizard eggs and ovaries and did all that fun stuff. But I got to see him interacting with Dick Jones and Hobart Smith, and I saw really a lot of what I think inspired him to become who he was as well, because it was a circle of not only high respect, but of friendship as well. And I just saw that grow with him, as he continued on his own journey. And I do have to point out, right, so this, sorry, I'm just gonna back up real quick, if I can, maybe not. In that photo, that was actually one of the last photos of me with him. That was in the spring of 2015. And we went to South Africa, of course, and absolutely spectacular. So I don't need to belabor this slide, but what I'm going to say is that while I had the privilege of him being my father, I also had the privilege of watching him grow up. And, going from not even being in graduate school, right, being an undergrad, and going through, graduate school and his career, and my goodness, I am so proud to be his daughter. And, and he certainly taught me so much, as Matt's alluded, and I think we all know, right, just how to approach life and science. couple days after he passed away, and you might have to make faces at me again, I went to his office. This was at the Harbor Master's house on James Island in South Carolina. And I sat down at his desk. I so desperately just wanted to feel his presence. I desperately wanted to just store away in my heart every memory I had of him. And I sat down, and this is what I saw. This was the window that was looking out over the harbor. And I just thought, I can see the world through Lou's eyes. And I just wanted to keep looking. I couldn't break myself away. And eventually I thought, I'm just going to take a picture, and then I can always come back to this. And so I want to share this window with you. You know, if ever you need to maybe revisit the world through Lou's eyes or recapture that, here it is. But I will say on a happy note that after I took the picture, you know, I stepped away from the desk and I looked around, and his office was full of mementos from graduate students, colleagues, friends, and I could tell that probably 90% of them had some hidden joke in them, right? That truly, at the time that gift was exchanged, there was laughter. And it just reminded me again of the legacy that he had and that he built, and how much fun it was doing it too. So, almost done, I promise. Oh no. Did I miss a slide? I might have missed a slide. Let me see. No, I didn't. Okay. So, I think we've all heard this expression too, the take home. So, Lou had a way of seeing the vastness of the world and then breaking it down into its smaller parts and communicating them in a very effective manner, no matter the level, right, of understanding of who he was talking to. And we've been to many presentations where he would be weaving together complicated experiments and data sets. And every once in a while he would stop and say, the take home is, right? Just to make sure that people were following him and were truly appreciating the message that he was conveying. And I remember seeing this picture when I was 12 years old. My mom took this picture. It was actually from their honeymoon in the mountains, right near where we lived and outside of Boulder. And those were in the days of the Instamatics, and it took 2 weeks to get your roll of film back. But I remember seeing this for the first time, and even as a 12-year-old, I was just struck by how it captured the essence of Lou. Just looking out at the world, appreciating its beauty, and still being able to come down to that very fine level and be questioning what is it that makes this work. And so one of my favorites. Okay, sorry, is it working? So, last slide, I promise. Another thing that Lou said very frequently was andale, andale. Right? And he said it all the time when I was a kid, if I wasn't moving fast enough or he was in a rush, andale, andale. And originally when I was making this slide, I was thinking it's, kind of our way of saying, come on, keep up the work, right? And so I wanted to put this up there, and I'd written andale, spelled very phonetically, on the slide, and it occurred to me I did not spell that correctly. So I Googled it to get the correct spelling, and I had always assumed, because of the way he had used it with me, that it meant hurry, hurry. But when I looked at the literal translation in Spanish, it was, come on. And that is Lou, right? Always inviting you to come on and to follow me, and I will follow you. And so, in conclusion, he also loved to speak Spanish whenever he could. Actually, it was his first language. Gracias. Thank you for listening. Thank you for affording me this opportunity, and really look forward to what's to come.
Wow, what a terrific start, and thank you so much to all the members of the family for coming and sharing with us today. Now, I don't know how anybody could follow that, but guess what? Marty Cohn, you're going to follow that. Marty Cohn from the University of Florida is going to present a talk titled Down the Rabbit Hole with Lou, Investigating the Developmental Biology of endocrine disruption. So thank you so much.
So, thank you. I have to say that in preparing this talk, I discovered I also get emotional talking about Lou. So forgive me for the first slide. There are quite a few things I want to say that are personal remarks, and I felt like the safest bet was for me to write this down. So I first have to say that I'm really honored and delighted to be able to speak in this symposium about my friend and colleague, Lou Guillette. He was not only a friend and a colleague, but he was a mentor and a role model. When I was a new junior faculty here at UF, Lou is the reason, really the primary reason why I came to UF, so I can blame him. And also the reason why I started working on endocrine disruptors. So he was the chair of the search committee for an evolutionary developmental geneticist. Oh, is that where the feedback is coming from? Thank you. And when I saw the ad and I looked up UF, I could find one other developmental biologist branded as such on the whole campus. But I saw that there was this amazing reproductive endocrinologist who had been studying genital malformations in alligators, and I was studying the genetics of external genital development in mice, and I thought that we would have some common interests. So I was invited for an interview, and I flew from London to Gainesville. I'd been in the UK for 10.5 years, and unlike every other place I had visited, when the secretary would email me and tell me the agenda, tell me to take a taxi from the airport, and the next day someone would pick me up. Lou said he would pick me up at the airport, and I still remember him telling me, I was describing myself to him, and I remember him telling me, "You can't miss me, I look like Santa Claus," which was cute. So I was wiped out from this transatlantic flight, just dying to get some sleep, and Lou was there, picked me up, asked me if I was hungry. Always the wrong question, that's always a yes. And so we went to a diner, I think it was the clock, to get a bite, and we sat there for about 2 hours, just talking and bonding, and I felt like I had met this long lost friend. And that's just sort of how he was, right, the whole time I knew him. But from that very first meeting, I really felt that discussion. I remember describing it to my wife as it was just electric. I was just so inspired and excited. And so that had a lot to do with, even before I got onto campus, feeling like, oh, this place feels at home. That was Lou. So after my seminar, which focused on genetic experiments that we had done and to understand how the genital tubercle, which is the precursor of the penis and the clitoris that I'll talk about in some of the slides, how that's patterned in the mouse embryo. Lou came up to me after the seminar. He didn't ask this in the public Q&A, which I'm grateful for, but he asked me right after the seminar, I was still standing on the stage, whether we were looking at sex steroids and endocrine disrupting chemicals. And I remember saying something to the effect of, by the time hormone receptors are expressed, all the interesting patterning events are over. And Lou smiled and laughed. And over the years, as we really got to know each other, I learned that Lou has two laughs. And there's a very subtle distinction between them. So one is when he finds something just genuinely funny. The other laugh was when he heard someone say something really stupid. And my answer got the second laugh. I didn't know it until a few years later. But I sat on enough committee meetings and defenses and qualifying exams that ridiculous answers really tickled him. Mine was ridiculous. So in hindsight, my answer to that question was so stupid that it should have been disqualifying in this job search, but I think Lou gave me the benefit of the doubt and surmised that I was just naive, genetics guy, and he really introduced me to the world of environmental endocrine disruption, and he changed my worldview and my science. And so I think of Lou, it's one of my favorite books, I think of Lou as my white rabbit. I was the naive Alice, and he led me down the rabbit hole into this whole new world. He introduced me to giants in the field like Nancy Denslow and Tayaguchi and Earl Grey. And this speaking slot actually was supposed to be part me and part Earl. And I got the happiest declined email from Earl when he said, "I'm retiring from the federal government in 30 days. And he's out. But I think he's on Zoom, so hello, Earl. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from endocrine disrupting chemicals and how they pattern in the mouse embryo. And what I want to do now is just make a transition from en these personal reflections to talk a little bit about Lou's impact on me, and then present you with a little bit of science that I hope reflects some of his influence. So he, like I said, he recruited me to UF. He was not assigned to me as my faculty mentor, but he was my de facto unofficial faculty mentor. Introduced me to thinking about systemically circulating hormones and exogenous factors that could mimic hormones, and it changed my way of thinking from locally acting gene networks that might signal over a couple of cell diameters to how all of these patterning events are playing out in the context of different hormonal contexts. My first NIH grant was an R21 with Lou. We were comparing genital development between mice and alligators. I think during the time that he was here, and I was here, I think we served on the PhD committees of all of each other's students. When he became an HHMI professor, I was so proud of him and so pleased, and he encouraged me to apply for an HHMI Early Career Scientist Award, and which I got, and he could not have been more thrilled for me. He always treated me like his colleague and his peer. I never really saw it that way. So Lou was about 15 years older than me, and so accomplished that I just didn't feel that we were, I felt like he was my advisor, frankly. And we were very close, and we collaborated, and I think it was a give and take relationship, but I just had such respect and admiration for him, and I modeled my mentoring style after his. And you've already heard, this is really, I think what will come out over the course of the meeting is the same themes over and over again about his style. And the first one that I noticed with him was that people come first. And it was all about the people. And I thought that was a very important mindset to have in advising students and postdocs. Also, that credit is earned and is infinitely divisible. That was that's a Louism. Credit's infinitely divisible. And the other thing I learned from Lou that he never articulated, but this is something I just got by sort of osmosis, and that was just be yourself. Right? There's no need to, I think especially when you're junior, you feel like you need to kind of rein in some of your wackiness and be a little bit more straight. Lou didn't do that. And I thought, he kind of gave me permission to just be me and share your enthusiasm. He was about lifting up others, taking risks, putting the students' interests ahead of his own interests, and cut people slack when you can. Those are some lessons that I learned from Lou, really by, like I said, not from things that he said, but it was just by example. So I feel forever indebted to him for not just our friendship, but for showing me how it should be done. Okay, so we initially connected, I mean, it sounds weird, but we initially connected over genitalia, and I was interested and still am interested in understanding the causes of variation, morphological variation in two contexts. One, phylogenetic context, how do you get diversification of anatomical form? And the other is pathologic variation, and an example of that. And this is a series of photographs of newborn boys. It's taken from a paper that Larry Baskin and Theo Colburn published in 2001 in EHP. And all of these kids have a defect called hypospadias, which is an abnormal closure of the urethral tube. Some of the kids have a slightly offset urethral meatus. The urethral meatus can open in the shaft, further down. It can, in these two kids, they really have ambiguous genitalia. You have a hard time telling the parents whether they had a boy or a girl based on their genitalia. But they're all newborn boys, and they've had a complete failure of urethral tube closure. And my thinking at the time, and it's still my thinking, is that if we want to understand how this sort of variation arises, we need to understand what's going on in the embryo in this structure, the genital tubercle. And so I'll introduce you to the mouse genital tubercle. In the mouse embryo, at about 10 and a half days, the E represents embryonic days, 10 and a half days of development, at the tail end of the embryo, this is where the viscera would be, there's the leg buds coming off here. At the tail end of the embryo, at the base of the tail, are these paired swellings called the genital swellings. and they grow together to form this single bud of cells called the genital tubercle, within which the urethra will develop. And then through a series of, it's really an iterative series of budding events, another set of buds develop at the base of the genital tubercle. These are called the preputial buds, or the preputial swellings, that will grow out laterally and ventrally, and they will envelop the glands, as you can see here, to form the prepuce, or the foreskin, and females the clitoral hood. And then there's a tertiary set of swellings down here, the labioscrotal swellings, that will remain unfused in girls to form the labia, or they'll fuse to form the scrotum in males. And I think this was from my job talk, actually. When we cut some sections through, when we first started working on the genital tubercle, we cut some sections through the embryonic genital tubercle, and I was really taken by the pattern, that there seemed to be this arrangement of cellular condensations and early developing structures that seemed to be polarized around this central structure, which is the developing urethral epithelium. And at birth, and to be perfectly honest, before I came here, we didn't even let the embryos go to birth. We were kind of interested in what was happening at all these early stages. At birth, you can start to see some of the differences. The urethral tube is completely enclosed in the male, and in the female, the urethral plate. The urethra itself terminates close to the vagina, in the vestibule of the vulva, but they also have a urethral plate that extends into the clitoris, and it remains open on the ventral side. And of course, after puberty, you can see the sex differences are very pronounced. And I had started making a living by doing a screen for genes that were expressed in the tissues that are affected in hypospadias. And that's the urethral epithelium, and in these buds that envelop and internalize the urethra and the glands. And one of the genes we found was a receptor for a fibroblast growth factor called FGF receptor 2, and the purple shows you where the mRNA is localized. It's only found in the urethral epithelium and out here in these nascent buds that will grow around and internalize the urethra. And we had shown that when deleted, when knocked out, this is a mutant, you can see straight into the urethra without doing any sort of dissection. The urethral tube is open. And before birth, if you compare a normal littermate to a mutant, it's as if you took a pair of dissecting scissors and snipped it right up the middle and flattened it out like a fillet. The entire urethral tube is open. So that's a severe hypospadias, and we're very excited about that. We had also found that a member of the hedgehog gene family called sonic hedgehog, when deleted, that gene's also expressed in the urethral epithelium, and when deleted, those mice had a complete absence of their external genitalia. They also have persistence of the cloaca. They don't separate the urogenital from the anorectal sinus. I'm hitting all the wrong buttons here. There we go. The genitalia in the sonic hedgehog mutant are initiated. Those paired buds that I showed you in the normal embryo can be found in the mutant, but by this stage, that should be a well-developed phallus. And they're arrested at that very early stage. And so over the course of many years dissecting the functions of these genes, we became interested in understanding later events, the ones that I said were not very interesting, that were different between males and females. We found that another member of the hedgehog family called Indian hedgehog also expressed in the urethral epithelium and around the glands. It was sexually dimorphic in its expression. And when we did a conditional deletion, leaving the gonads completely intact, but deleting this gene only in the genital tubercle, the mice had these very unusual penises in which some of the male-specific features, like this urogenital mating protuberance and the ridge around it, and some of the sort of ornamentation that you see in male phalluses but not females, that was gone in the mutant. So they really had lost. There was a disruption of some of the masculinization. Okay, so there was an opportunity by coming here to start thinking about how the factors that we had been studying, transcription factors and growth factors that act over short distances, interact with these systemically circulating factors, and how that information is integrated to generate the external genitalia. when do these interactions occur? Where do they occur? Which cell types do they occur in? How do the hormonal cues regulate these locally acting genes that I've just told you about? And those were questions that were really unknown. And I'm going to show you a relatively recent result that I think would make Lou laugh. I'm not sure which one of his laughs he would give me on this, but Lou was right. So we had done some single-cell analyses, single-cell RNA sequencing of the genital tubercles at different stages of development. And you don't really see any sexual differentiation occurring until about 15 and a half days of development. Males and females are indistinguishable up to that point. But at least as early as embryonic day 13.5, these are clusters of cells that are defined by their transcriptional profiles. There are 23 populations of cells in the genital tubercle at that early sexually indeterminate stage. And when we colored the male cells, this is integrated, the male and the female cells are all pooled together, but when we asked where are the male cells and where are the female cells, we made female cells this sort of brownish pink and the male cells blue, you can see that they're not evenly distributed. And when you quantify this, you can see that there are very different proportions of certain cell types between males and females that cannot be, at this stage, due to hormonal differences. They're more likely due to differences in the sex chromosomes. So I think Lou would love that, because I am publicly eating those words that all the interesting stuff is early and the sex differences stuff is late. So what I really wanted to do, and I think this was only made possible by coming here and being able to interact with people like Lou and like Nancy, was ask whether we could take some of the tools of developmental genetics and genomics and try and address some of the questions that had historically been within the realm of environmental toxicology, to see if we could identify some of the genetic targets and some of the mechanisms that are affected by endocrine disrupting chemicals in a good genetic model, like the mouse. For all of its limitations, it is a good genetic model. And so one of my postdocs started by asking, when are the genitourinary organs sensitive to endocrine disruption? And this is the work of Patrick Zhang, who has his own lab at SIU now. And what Patrick did was identified the window of sensitivity, the temporal window during which the external genitalia are responsive to exposure to anti-androgenic chemicals. And he started doing this with flutamide. And what he found was that there's a 3-day window in the mouse embryo during which an exposure to an androgen receptor antagonist, and that could be a pharmacologic antagonist or an environmental antagonist, can induce a severe hypospadias. This is the effect of a 3-day exposure, and now you're looking at the penis of an adult mouse, and the urethra is just completely open here. It's also tethered by this soft tissue, which gives a curvature known as a chordee. That's another congenital anomaly of the penis that occurs in humans. And here you can see that the urethral tube extends down into that tether. So that exposure, just for that 3-day period, causes a very severe abnormality of the external genitalia. And we did some genetic experiments to ask what would happen if we delete androgen receptor at specific stages. And we found that there is that the pharmacologic experiments and the exposure experiments were validated by the genetic experiments. If we deleted the androgen receptor just in the genital tubercle, not touching the gonads, by embryonic day 13.5, the mice had sex reversal of their external genitalia, but not the internal genitalia. If we waited a little bit later, they had genitalia that looked a little bit like that Indian hedgehog knockout, where they have micropenis and are missing some of the male-specific ornamentation. And so what we concluded from this is that you could induce this full range of congenital penile anomalies. And if you know any accountants, you should tell them that the abbreviation CPA does mean congenital penile anomaly. You could induce this full range simply by varying the time of exposure. And we then started moving into some environmental antagonists. And this was also a result of Lou introducing me to Earl Gray, who had published some absolutely beautiful papers on the effects of encloseline on rat external genitalia. And so we really built on that to try and use the genetics of the mouse to identify what are the targets, how are these gene regulatory networks being affected by the exposure. And so Kelsey Lewis, who also has her own lab now at University of Buffalo, she repeated Earle's experiments in mice and got the same sort of a phenotype, and that was good because we could then go from there to ask how this is happening. And in collaboration with Nancy Denslow and her team, we started looking at how the maternal and the fetal hormones are affected by the exposure, and we found that the vinclozolin treatment of the mother really causes testosterone level in the male pup, in the embryo, to just crash, lower than even the wild-type female has. We also started screening for other genetic changes that occur, transcriptional changes that occur in response to the exposure, and we were really intrigued by this suite of genes that are part of the hedgehog signaling pathway that are important for regulating apoptosis. And something that Lou and I had done together was we compared patterns of apoptosis in the alligator and in the mouse external genitalia. And at the time, we didn't really appreciate this because we were just looking at males, but when we looked at females, we noticed that there's a sexually dimorphic pattern of apoptosis, of cell death. The male has this death that occurs between the urethral epithelium and the underside of the penis, and you don't see that in the female. And we thought that might be important for separating the urethra from the skin and allowing it to really internalize. And when we looked at the effect of vinclozolin on that, we found that it causes the female pattern of apoptosis, as it really represses apoptosis, and we think as a result, the urethra stays tethered to the underside of the penis. And association is not causality, right? So is it apoptosis that's responsible for, is this the real mechanism by which vinclozolin causes penile anomalies? And we could test that genetically. We did this in collaboration with Saunders Ching and Ophir Klein at UCSF, and we used a mouse that they were using. This is the BCL2 pathway, which many of you are probably familiar with, that leads to apoptosis by suppressing this Bax-Back pathway. And there's a mouse that's available in which Bax-Back can be conditionally deleted in whatever cell type you like. And so we just removed that apoptotic mechanism and asked what is the consequence genetically in unexposed mice. And what Kelsey and Saunders found is that just removing that apoptotic machinery generated a phenocopy of the vinclozolin exposed mice. They have an open urethral tube. This is a normal pattern of apoptosis, and this is a mutant, and they have an open urethral tube or a hypospadias in the same place that we were seeing it in the vinclozolin exposed mice. So we think that the way that vinclozolin is working to cause this penile anomaly in mice is both by repressing fetal levels of testosterone, antagonizing the cell death pathway, causing persistence of these cells and persistence of the urethra on the underside of the penis and failure of closure. And so just to conclude, this was also something that I think has been alluded to in earlier talks, and that is, what is the mechanism and can you keep digging? And when you find one mechanism, can you then drill a little further and a little further and a little further? And that was sort of Lou's way, right? The answer was never the end. The answer was the start to the next set of questions. And so what we discovered by doing genetic profiling, or transcriptional profiling of these mice that had been exposed to anti-androgenic contaminants, was a lot of genes that we had already shown, we and others, including Gen Yamada in Japan, had already shown when you delete those genes, when there are mutations in those genes, the mice get hypospadias. Here, these mice were just exposed to an anti-androgen at that particularly sensitive window, and we found that a lot of those genes have significant drops in their level of expression. This is FGF receptor 2. Remember, when you knock out that gene, you get a severe hypospadias. Here, in an exposed animal, the level is dropping by about 4-fold. Indian hedgehog, important for masculinization of the penis, the level is dropping by an even greater amount. And so from this is just to remind you what the mutants look like. From this, we think we might have an explanation for why so few children who, first of all, hypospadias is incredibly common. It's about 1 in 125 newborn males have a hypospadias. There have been a huge number of attempts to identify a genetic cause of that, screening in children, and they've been more or less fruitless. There's some copy number variants that have been identified, but that number of children and the increase, more than doubling over the past 50 years, that can't be genetic. A mutation can't move through a population throughout the industrialized world that fast. It has to be something other than genetics. And so environmental contaminants are a very likely candidate. And now we think we might have some potential explanation for why kids with hypospadias look to be genetically normal, and really are genetically normal. And that is that a genetically normal embryo is motoring along through development happily. It hits that sensitive period, and either by an acute exposure, which I think is extremely unlikely, more likely is it hits that sensitive window, and the maternal body burden of accumulated anti-androgenic contaminants causes this transient drop in the expression of these genes that are critically important for urethral tube closure. So it's effectively causing a knockdown in these genes, and then the levels recover, and the embryo fetus continues on through development, and you do a genetic screen of a kid who has hypospadias, and it's genetically normal, and there's no retrospective indicator that an exposure event happened. But we think that there could be. And so we're interested in finding, for example, epigenetic signatures of these exposure events that might allow you to explain how these kids with hypospadias ended up with that penile malformation. So I'll just conclude. I think I've acknowledged everyone as I've gone, but I want to just once again thank the organizers for inviting me to talk. Thank the family for their amazing discussions of Lou and introductions to Lou, and thank the big man himself for all of the inspiration and guidance that he gave to me. Thank you.
Well, I'm ashamed of myself for thinking that Marty Cohen wouldn't be up to the task. So okay, I'm now going to introduce Elizabeth Brammer Robbins. She's a PhD candidate in the College of Veterinary Medicine here at the University of Florida, and she's going to chair the next session.
Who will be presenting Tuesdays with Lou, Gators, Sons and Daughters, and the Pursuit of How Alligators Compare to Humans.
Thank you. I'd like to thank the organizers for inviting me, Matt and Nancy and Jerry, and really it's an honor to be here. So thank everybody for a master class this morning in composure. I'm impressed because I'm not sure I could be that composed, and we'll see how it goes. Where is my talk? So one of the things you'll see in my talk is, I called it Tuesdays with Lou. Lou and I did not talk every Tuesday. This is built off of a book that was a bestseller in the 90s called Tuesdays with Maury. And as I look back, that's kind of what we had. Lou and I talked on the phone quite a bit. Sorry. While we look for my talk. So we talked on the phone often. And the blue one here. This is the first slide. Okay. So I thought I would give the talk in 3 parts. And the first part is talk about Lou, which he wouldn't like. He'd be like, stop talking about me, talk about the data. I'm going to talk a little bit about data. I'm not going to talk a lot about data. And then I'm going to talk, third part, about our contribution to his legacy. So you've heard what a great photographer Lou is. I'm not. So this is, Lou and I, when we were in the field, we didn't take a whole lot of selfies. And so these were 2 pictures I could find on a day that we were both in the field together. And the picture of me, which is bright and clear and nice, Lou took with his camera, and the one of him that's kind of blurry, I took with my iPhone. So you can see he's a much better photographer than me. But we had some unique traits that I really valued. So one, he was a kindred spirit. I grew up in my career in medical schools. I was not Lou's student. I was not his postdoc. We were just colleagues. But I was working in a medical school. I worked on whales. He was working in medical school when I met him. He worked on alligators. It's not very common to work on wildlife in a medical school, and that has a lot of challenges. So it was very nice to find somebody else doing it that we could talk about some of those challenges. I mean, to give you a feel for it, when I was in the Yale School of Medicine and I got my first marine mammal grant, they said, we think that grant you have on fish is just great. So I would actually have to teach them that marine mammals are not fish. So that was a cool part of our many, many conversations on the phone. We also had a similar sense of humor. So I was in Maine, working in Maine when I met Lou and we started working together. And on a day like this, which was very common in Maine to have a snowstorm like this, Lou always made a point of calling me and saying, you know, it's 70 and sunny here in Charleston. And so I would growl at him and say, okay, all right, I got it, yep. So then I would call him on days like this. All right, this was, and I would say, hey Lou, it's like 70 and real cool up here in Maine in the summer. And so he would growl at me, and so we had a lot of fun with that. The other thing that was unique, that I found very unique, is we both worked with our sons. You can see Matt and Johnny there. Not often you encounter that. And that led to a lot of very useful conversations about what do you do when you work with your son. Johnny actually almost did his PhD with Lou, but there was a paperwork snafu, and so by the time he got accepted, Johnny had already agreed to go to Purdue, where he was doing his PhD in neuroscience. Then something I didn't get to share with Lou, but that is really, well, no, before that one, there's another one. We were also big into teaching. I don't know if this video is going to play or not, but this is actually my daughter holding a gator heart because I wanted to look at some effects in lung cells, and I'm going to show you the effects of gator in lung cells. But while we were taking the lungs, Lou couldn't resist the opportunity to teach, so the heart got put into a petri dish, and Lou took the moment to educate the students present, and let's see if it'll play. Well, it's not going to play. Okay. Oop, got ahead of myself. Anyway, in it, he talks about the beating heart, how the sinoatrial node, and how it works, and it's really a poignant moment because we were not working on the heart, we were there to work on the lung. This is the thing I didn't get to share with Lou, is we have this really unique feature in common, in that for both of us, our graduate student became our daughter-in-law. So for Lou, that was Teresa, and for me, that's Rachel. And you can see it's our sons that worked with us the most in the field that actually married our graduate students, Matt and Teresa, of course, Johnny and Rachel. And so that's a cool thing. So that's Lou for me. Now let me move on to science where I can maintain my composure a little bit better and talk about the pursuit of how alligators compare to humans. That was how we got to gators, sons and daughters. So in my lab, our central question is we're trying to understand how a normal cell becomes a tumor cell by alterations in the karyotype after environmental exposures. And one thing that we study a lot, and I'm not going to get into the background on it much, is hexavalent chromium, which is a global environmental pollutant and a human lung carcinogen. And you treat normal lung cells with particulate chromium because that's the really potent form. And after acute exposure, you get DNA double-strand breaks. These breaks occur in late S and G2, a phase of the cell cycle. And so what we discovered through a lot of work was homologous recombination repair fixes these. So you get proficient repair, and that protects the karyotype in the genome after acute exposure. This is in human lung cells. But when you go to a prolonged exposure, you actually inhibit the double-strand break repair by targeting two proteins, RAD51 and RAD51D, and preventing the repair from happening. And since this repair can't happen, the cell has to switch to other pathways, and you end up with this highly rearranged karyotype and a tumorigenic cell. So you've gone from a normal karyotype of 46, this one is actually, I think it's 72. All the red arrows point to structural aberrations, and it's a highly damaged karyotype, and these cells will grow and auger, and they grow new mice. So that's what happens in human lung cells. And we've translated this from human lung fibroblast cells to epithelial cells to rat cells to guinea pig cells. And then we went into, well, there. We went into animal models and we showed this mechanism occurs in rat lung tissue. We have data coming out showing it happens in guinea pig lung tissue. And then we worked in tumors from workers, and we showed this mechanism occurs in chromium-induced tumors from humans who worked in the chromium industry and died of lung cancer, and a little bit of data in adjacent normal lung tissue. So the point being, this is a very robust mechanism that translates across models and levels of organization. So I've been thinking about this, and so this is why we isolated lung cells. As I was like, Lou, I want to look at DNA repair in alligator lung. So he said, all right, come on down to NASA and we'll get a gator and we'll figure it out. So we asked this question, does this actually happen in alligator lung cells? And so I take it, this is work done by Aggie Williams, who's actually going to defend this data in about 2 weeks and graduate with her PhD. And we went back to this mechanism and first asked, does chromium cause double-strand breaks in alligator cells? So we treated the cells with chromium, and we met with for either 24 hours continuously or 120 hours continuously, used a common assay to measure the damage. So that's an assay where you put the cells in an agarose gel, get rid of everything but the DNA, and the DNA being negative runs to the positive end. And in that picture, you can see the longer the tail or the brighter the tail, the more breaks you have because smaller broken pieces move faster than big pieces. So on the y-axis, we quantify the tail length, we divide it by the control, and you can see at all of our concentrations, we're above 100%, so there's double-strand breaks occurring. And we've measured this with other measures as well. Yes, chromium induces breaks in alligator lung cells. In fact, when you look at it in human lung cells, it's about the same amount of breaks. So the number of breaks is consistent from the exposure across species. So that was a yes. So then we asked the question, does it inhibit DNA double-strand break repair? And here, you focused on RAD51. Again, we have other measures, but I wanted to keep it short today. So in that picture, you see green dots, and each one of those is a RAD51 foci using an immunofluorescent antibody for RAD51. So each dot is considered one DNA double-strand break that's being repaired. So on the y-axis, you have the percent of cells with RAD51 foci. We use something, we take the level at which 95%, where the control, 95% of the control has that level. So in humans, it's 5, and I think in alligators it was 11, or it could be the other way around. Anyway, we quantified the RAD51 foci, 24-hour and 120-hour. These are continuous exposures. There's breaks at all these doses, and indeed, alligators have repair going up at these doses. But remember, well, that's just showing that they're going up. Remember what I said, in human cells, this was inhibited. So in human cells, we see the exact opposite outcome. So in human cells, chromium is inhibiting repair, but in alligator cells, it's not. So that was a really cool outcome, and the whole reason we got the alligator lung to start with. So that doesn't happen. What about, we get proficient repair, so what about the protecting the genome? So what happens there? So we've looked at that, and this is what happens in alligator lung cells. So here we did a chromosome damage assay where we measured the amount of breaks in the chromosomes or fusions and things like that. Shown in the picture, you see 2 broken chromosomes. On the y-axis, we have the total damage, and here we subtracted out the background from the control, which was low, but we did it to make it easier to compare. And you can see in alligator lung cells with a 24-hour exposure, there's some damage, but with chronic exposure, with repair being up, there's actually less damage. And when you compare that to the human cells, you can see indeed you get a lot more damage because repair is inhibited. So I'm sure Lou would have loved this. And we're really excited about it. So that doesn't happen, and we suspect that then that doesn't happen either, that we haven't measured that. So does this happen in alligator lung cells? Yeah, no, it does not. And so Aggie's now trying to work on, well, she's going to defend, so this is going to get passed to a different student. But how are they resisting DNA repair? We've also got studies looking at chemical levels in the alligator populations in Brevard County, and then going back to our work at NASA with Lou and Russ, and we're looking at DNA damage levels in wild alligator populations. We're actually about to submit that for publication probably this week when I have time at SOT. So the gator work goes on. So how do we see ourselves contributing? First, there's the exciting gator research that we do. We actually I have this talk today, but then tonight I'm actually leaving in a few minutes to go to Orlando because I got to give a gator talk to a bunch of undergraduate students there. And a bunch of posters at SOT, and then Thomas is holding a conference 2 weeks. So there's a lot of exciting gator research. We have enduring friendships. I mean, for me, Russ and I became very good friends. Matt and Teresa and I have become friends. There's lots of enduring friendships that emerge from our time working with Lou. We have some new established partnerships. This is us with my wife Janie and I with Frank Robb, where we now have an ongoing study of DNA damage levels and pollutant levels in gators in Brevard County, Florida. In fact, Frank goes out every weekend and sends us blood samples every week, and we get down to see him to collect gators ourselves probably 3 times a year. And then family. As you can see, the whole family is involved in alligator research. And I think that comes from Lou as well. So that's my wife, Jamie, and my son, Johnny James. James is the only one who's not with an alligator. He does all of our GIS mapping and stuff like that. And if you ask James, he'll say, I've never invited him on a gator trip. I wonder how many of you in this room believe, with all those pictures, that I've never invited James on an alligator trip. I would tell you that James is actually a professor at LSU. He hates hot weather, and he hates bugs, but he's got alligator cells growing, and he does all the mapping, and he's heavily involved, but he does not go out with the gators. And then Emma and Addie went this year with us, with Frank there, our 13-year-old twins, and Emma actually went to a conference with us in Spain this fall and presented on our Brevard County gator work. So it's still it's a family business for us, and so we continue that in the spirit of Lou. So kind of the last slide before the acknowledgments. This was the book Tuesdays with Maury. I think if I was giving this talk at Lou's retirement dinner, I certainly would have called him the old man and me the young man, but it seemed he's not around to tease like that. And then Life's Greatest Lesson. And this is what came out of the book. Devote yourself to loving others, devote yourself to your community around you, and devote yourself to creating something that gives you purpose and meaning, is what Mitch, the author, interpreted from his Professor Maury as the meaning of life. And it sounded to me a lot like Lou. So first acknowledgement, I thank Lou. And then I thank everybody who's helping with this. People in yellow have worked on the data and the alligator catching that we've shown here. I, of course, thank our generous funding and NIHS, who I think is somewhere in the room. I thought I saw him. Requires me to say that this is my views and not the views of NIHS. That's it for me. So.
Go Wolfpack. Alligator Tears, One Health in Deteriorating Environments, and building on Lou's EDC Foundation.
Thanks, and I have a habit of kind of dropping my hands, so just somebody, if you can't hear me, just let me know. I'm going to get these slides up, and I think I got most of my emotions out yesterday. I was blaming it on the plane on allergies, but I think I'm going to be good today. I wasn't quite sure, and this was a hard talk to put together, not out of grief or any loss, but all of the joy that I went back and found trying to think how to put this talk together. So, and was up on the, no. Thank you. And Jerry, I know I'm always late with my slides, so I'm creating my own problems, but I'm going to hopefully go fast. So I am, because of all that, I am going to try to use the script here so I won't move around and jump around as much, probably not have to say, but as I was working on putting this talk together and really thinking about it, you know, I really wondered what I could say or, you know, in all honesty, really had a right to say or comment about Lou's foundation on EDCs or even the foundation and the influence that it had on my science. And so I came up with this really crappy title. I think I was like working on a really depressive grant that I put in. Actually today, it's when I told Matthew about that I've been building up to. So that got submitted today, and it was really focused on some really awful environments in Africa. So, you know, I didn't want to talk about this because I know I'm definitely speaking to the choir here. I know many of you, I've had the privilege of working with some of you, been on the stage often with John, I've given talks with Ty, and so, you know, this is our people, you know, it's the choir. And, you know, many of you have been working on EDCs like Lou did with a mission for many years before I got involved in this. And you've been battling a true fight for years. So we're going to dump that title. We're going to go a little bit of a little different direction and build on some things that Matthew said about how times have changed, science has changed. We're frankly all under attack. And so I'm going to have a little bit of a policy beginning here that's going to shift into to the important parts. So with that, yeah, we all know we have a system that's meant to act as a steward of the planet, much like Lou's work was always done. So I don't need to tell you that, you know, the system is currently not protecting the planet, nor its inhabitants. And we all know that we as humans are not living in balance with the planet. Oops, there we go. I may click twice here if it's a little slow, I apologize. And yeah, we know our governments as our representatives. They're failing us. They're failing us through abandoning their responsibility of stewards to the planet and life on the planet. Oh, okay, thank you. So I was just using the computer. And frankly, you know, our societal balance is not in step with science. And that balance between public safety and environmental safety and amassing absurd amounts of wealth is out of balance. And, you know, frankly, scientifically wrong. And because it's wrong, we're now living in a state where even our purified water is tainted with toxic chemicals. And if we ever have, we're certainly now not regulating chemicals in a thoughtful, logical way. But instead, we've created a decades-long global experiment where we pollute, we wait for people and ecosystems to become ill, and then we litigate the human and planetary cost. And we've also known for a very long time, and a big turning point while I was a postdoc was Lou and Andy's and Drew's paper. It was just stunning. At the time I was looking at estrogens in the brain. And I was a molecular geneticist, and that's all we did is try to one-up each other with the most elegantly simple experiment possible. And for me, this resonated on this. And I also got to tell people about this cool experiment that, you know, these guys with really big gonads doing science measuring alligator penis. And it's like, you know, this is perfect. This is perfect for reaching the community. This is perfect science. And so we had that information. Lou gave us upon stacks and stacks of pre-existing evidence. The understanding that EDCs and chemicals in the environment are bad. But what was really elegant here, and what really talked to me, and thankfully nobody's mentioned it yet, the real elegance in this experiment, that sometimes doesn't get mentioned. Lou had the insight to know that the alligator was the right system. The right system to remove the majority of maternal influences away from the critical developmental windows. And that was the genius about it, because we could get at those points during development in ways through the alligators that we couldn't with mammals. And that's kind of where I came in. So I decided I wanted to change my title, because Lou's impact on me was not so much that science, which I greatly appreciated, but it was much more. It was much more than a foundation to study. What it did was brought joy to my science again. I too was in medical school, so I got tired of hearing about hearts and calcium and contraction and all that. Came to NC State for family. And so what Lou actually gave to me was a gift that I feel that was faded. And it was a gift of love, and it was a gift of family. So we are coming back to individual themes that are solid. And and I do want to, a lot of these pictures are Teresa's, so she did get some of Lou's photography skills. And she also told me too that, well, yeah, Lou always bought all his camera stuff on his grants. You need to buy some. So, you know, I suck at photography, but I've got some nice equipment now because of Teresa. So, you know, part of the big question is, you know, how does this guy in his shiny medical school lab looking at his, you know, lovely little colorful water bottles and, you know, in this nice, clean, tidy environment, go from this kind of, I don't know, boring scientist to some guy who is just dragging around happy mice and rats, and then all of a sudden you're sitting on 12.5-foot fluffy. Well, it was a process, and I want to try to take you there because it was really faded from the universe. And at this time I was really, you know, looking at BPA, and to reiterate, my connection to Lou really is indirectly. And it is through the nexus of my center of the universe. You know her, she's in the back corner there, Heather Pattisall, who made it in spite of government orders. And wasn't I supposed to say that? You're on vacation? Okay, great. We have strict conflict of interest, so I don't know what the hell she's doing on any given day. So anyway, she had a big part of this, because she was one of Lou's undergraduates, and she can tell those stories better than I. But shortly after Lou passed, Heather was planning the EDC, Endocrine Disruptor Gordon Conference. And in the way that she does, she took it on as a slightly enthusiastic mission and raised excessive amounts of money to make sure all of Lou's orphan trainees got to this endocrine disruptor meeting. And arguably the best, I'm biased, but best endocrine disruptor EDC meeting we've had. And as you do it at EDC meetings, you talk with students, you're kind of like, yeah, well, this, you know, where's the real bright ones? And you're talking, and so I'm up there late at night after some wine, talking with, you know, the student that just won the poster contest and stuff. We start talking, we talk about ridiculous things, and kind of, if I knew Lou's laugh of, there was a few back and forth stupid, hey, yeah, that's a stupid question, dude, that's a stupid question, student, type laughs going on. But as it turned out, I ended up hiring Teresa Cantu as a postdoc. And you know, all of a sudden, Teresa Guillette shows up at the door. And there's paperwork coming from HR, and I was like, what the hell, is this a mistake? I don't know what's going on. But so, you know, it's about 3 days in the lab, and we're walking down the hall, and who's now Dr. Guillette, who was there all ready to go with transgenic mouse work, which, you know, after a few months we decided to change projects. And she mentions that her husband we'll need something to do. He's worked in labs before and stuff. And I said, yeah, let's have lunch. And then she has that look on her face as we go to lunch, as she sees my little, like, two neurons light up and go, oh, that's connected to Lou. and that was the next thing that was fated, because I didn't get somebody in the lab. I got my brother from another mother. Matt and I really, it was a gift. It was a gift from Lou, from the universe. And means the world to me. And that was the big part. Without him, I had Heather saying, gosh, you and Teresa wrote this grant together. You realize you've never seen an alligator in the wild, right? That's all right. I got good teachers. We got this. And so, and we did. And subsequent to that, now we've really realized, and we're trying to be poster childs, which, like Florida, we're in the middle of one of the global biodiversity hotspots, and we've worked on dang near everything that we can, and we've got some more stuff coming up the pipe with new species, and just the joy of doing that was the gift that I received. And yeah, we still used mice and collaborated with a lot of folks. Jackie Begma, who's not here, is a big part of our life. And that joy and that love was Lou's legacy, and Lou's legacy to my lab. And I just wanted to leave this last slide with you. Not say a whole lot about it, but just the amazing amount of family crossover. We had the first lab baby during this time, and it's pure joy. And as we've adopted from Frank, who we just saw working with John, one of our buddies, Lou, we love you, man. And with that, that's all I got to say.
My name is Tyson Iguchi. Currently, I'm at the Yokohama City University. Before this university, I was in the National Institute for Basic Biology, Japan, which is located in middle of Japan. Actually, I went to the Howard Byrne as a postdoc, and he introduced Lou Guillette to me. And that was 1994. I didn't know he can't drink alcohol. So, me either. So, we are holding the Coca-Cola and then step away from the drinkers. So we chatted. He was a very attracted people. And then they are always friendly. He is a very nice storyteller. So everybody wanted to know what he talks. Every time he has a new ideas or findings. This year, the John McLachlan, Tulane University, organized the meeting in Washington, D.C., I think, Estrogen in the Environment 3. And at that time, Howard introduced Lou to me because I worked on the mouse. When you inject the female mouse, newborn mouse, to estrogen, the estrogen induced the one phenomenon in the ovary. The mouse developed the polyovular follicle. Usually, one follicle contains one oocyte. But if you inject the mouse, but early age, the newborn mouse, one follicle contains more than two oocytes. So we worked on that, the mechanism also. And Lou, I didn't know he was working similar thing in the alligator. The Howard asked me to talk to Lou, but the same thing happening in the mouse and the contaminated lake alligator egg. So that is the first time I met with Lou. And instantly we liked each other. And so the 1996, I invited him to come over to Australia. We had the Asian Oceanian Comparative Endocrinology Congress. So the organizer asked me to have session of the endocrine disruptor things. So I invited him come over to Sydney. I also invited the John Sumpter from UK and Tyrone Hayes in UC Berkeley, etc. And we had a small, I think this is the first endocrine disruptor session in the comparative endocrinology. And the next year, 1997, I need to organize the International Conference of Comparative Endocrinology and the session of the endocrine disruptor things. So I invited him, Lou Guillette, and John Sumpter, and the Tyrone Hayes to Yokohama, Japan. This is the start of the collaboration with Lou. So he asked me to come over to Gainesville to work on the alligator. At that time, Lou didn't have molecular biology techniques in his laboratory. At that time, my graduate student is finishing a PhD, and the Lou wanted him to come over to Gainesville, University of Florida, as a postdoc. And he started teaching the molecular biology techniques to the graduate students. So that is a much more close relationship. We can send the molecular tools to Lou, and the Kono Satomi taught molecular biology to the Lou's graduate students. We thought, what should do with alligator? We don't have alligator in Japan, and it's very hard to bring the alligator eggs to Japan. It's impossible. This is a very good collaboration. I can come over to the University of Florida every summer and helping collecting alligator eggs and also catching young alligators to measure the blood hormones, etc. So that's continued for more than 10 years. Every year I went to University of Florida. Also, Lou came to Japan. We invited him several times for the endocrine disruptor meeting in Japan. Finally, we should do something interesting. And the first thing is he wanted to have the estrogen receptor in alligators. He tried to clone, I think, for a long time, but he didn't succeed. So I asked him to send a tissue sample to me, and he sent me the piece of the alligator. So we cloned the estrogen receptor in a week, the full length sequence, and then I emailed the sequence, amino acid sequence. So he astonished, and then he can use those techniques every time in his laboratory. And then also we cloned the progesterone receptor, androgen receptors, and several things. I think at that time I was working on the Daphnia, water-free. The female produce only female. Parthenogenesis, no male needed. And we are working on the environmental sex determination. Textbooks said that the Daphnia is the environmental sex determination. If the environment condition is getting worse, the female lay eggs, which will develop to male, and male and female copulate, and resting eggs can be made. The nesting egg developed two females from the one egg. Actually, two oocytes are in the nesting egg. So the alligator is also environmental sex determination. If you keep the egg at the higher temperature, like 33 centigrade, that temperature induces all male. And if you keep the alligator egg at the 30 centigrade, that produce only female. So temperature decide the male or female. So we wanted to know the mechanism, how the alligator sense the temperature. And then we worked together and they found the one candidate that the temperature sensor, which sense 33, 34 centigrade, that is a receptor, the temperature sensor, transient receptor potential. That is the gene name, TRP, V4. So we found this, the protein sense the 33, 34. So we can block this sensor using some chemical. So I can't do these things in Japan. So I asked us to set his laboratory for a little bigger incubator. Every summer we can collect the alligator egg and clean Lake Woodruff, and then bring back to the laboratory. And then we painted the temperature sensor blocker chemical on the alligator egg and kept in the 33 incubator. The egg should be developed to male. But if you paint the chemical, block the temperature sensor, even incubated at 33 centigrade, the egg developed to female. We wrote a paper 2015. That was a little late. He passed away in the summer of the 2015. But we published the paper several months later. So that is my very sad part. We could maybe enjoy 2015. We had a meeting for Lou Guillette at the Charleston. So I visited at the ceremony and I reported him that was published. So it was a very sad part for me. And of course we enjoyed together and chatted a lot. We traveled to South Africa and also the Mexico. And then we talked a lot of sciences. And also we cloned the genes from the crocodilians and some turtles, etc. And also whale shark. And also we visited the Cape Canaveral. NASA allowed us to catch 10 male alligator. So we caught 3 to 3.5 meters only male alligator. And then that was fun. He gave a lot of lectures in Japan. Then lots of students are inspired by him to work on the chemical things. Have lots of friends every time he comes. So he enjoyed in Japan. He started collecting masks every time I took him to the some antique shops, several places. He collected quite many masks in Japan. I think he exhibited his masks in somewhere in Gainesville. And the other thing is he came to my home and he talked to my daughter. And my daughter inspired, she wanted to go to University of Florida. Actually, she went to University of Florida, but not science part, but the music part, and she enjoyed there. So it was expensive for me to send this.
Hi, everyone. I'm Matthew. For those that don't know me, I was a graduate student who worked with Lou starting in 2013 when he was at MUSC. Nowadays, I'm a postdoc. I work in a genomics lab here in the DC area that does infectious disease work. I think I can say I was Lou's last doctoral student to graduate. Of course, with most of the credit for that going to Ben for dragging me along to University of Georgia to get me to finish my PhD. Um, I don't know, I was really lucky to have met Lou as an undergrad when I was working with Drew Crane at Maryville College. And in fact, it was Drew then who kind of convinced me to go to grad school in the first place. But when Jerry reached out about the opportunity to join this panel discussion, I was disappointed. I'm probably on a plane right now while y'all are listening to this, that I wouldn't be able to join in person, but I just had to put something together considering Lou, either directly or indirectly, has been doing a lot shaping what the last 15 years of my life have been like. And I thought I would just share one really quick Lou story. And it's not anything about how impactful his science was or how great of a leader he was. Everyone in the audience has some story behind that, and many people can tell it better than I can. Instead, I just have this little vignette that I think about all the time of what it was like to work with Lou towards the end, in the middle of his crazy, nonstop flurry of research activities, his international travel. I'm sure he was still testifying at the time, like everything, all of the stuff he was doing all the time, and how amongst all of this, he was still able to make us, his graduate students, feel really important and feel really connected to him personally, instead of just these little like caffeine and stipend fueled data generating machines that sometimes graduate school can make you feel like. So this was in 2014 in May, which was like, in the spring, the calm before the storm, because it's right before egg season and it's right before croc season, when they're going to South Africa. And I remember one day we got called into the old DNR lab that was at Fort Johnson, for anyone who is familiar with our campus in Charleston. And I remember we get called into the lab for something special, something like it had like a secret feeling to it because we were all hush hush about it. And I don't remember who was there, probably Ben and Satomi at least, because it felt like they slept at the lab at that time. Some of the other graduate students, Brenna, Thomas, Jackie was probably there, Teresa. And we all get ushered over to these, the big standing incubators that we used to use during egg incubations. Imagine 6 feet tall, huge swinging aluminum steel door. And I don't know who does this, but they pull the door open and somebody takes out very gingerly this tub, these big plastic bins that we used to use to transport hatchlings around. And you set it down on the bench top and they carefully open the lid and take it off. And inside is like the most perfect lily white hatchling alligator. I assume most people in the room can probably imagine like a pixel perfect neonate alligator down to like the very last scute, exactly where everything goes. So most of us probably know that the stock color for alligator hatchlings is not white, but this thing was perfect white with like a one little brown patch on the top of its head, almost like a little alligator mohawk, and eyes that were so blue they were almost black. So, none of the typical cool gold and black irises.
In the
And
middle,
I remember so crazy about these things is that they were thin. Like I don't think anyone has any misgivings about how hard it is to be a hatchling alligator in the real world, but these guys were, these little alligators were proof of it. And it turned out that this was all happening because Thomas Rainwater, probably sitting in the audience somewhere now, had somehow come across a number of these perfect leucistic hatchlings somewhere in South Carolina. And because leucistic gators are pretty hard to come by, I mean, like genetic lottery, this whole air of secrecy and mystery and trying to stay very quiet about this was because we were worried that if word got out about the location of where these alligators were or where they came from, folks, poachers, would tear the place apart to try to find them and get as many as they could and sell them, because an animal like that is probably worth a lot of money. So Thomas, and I'm assuming his folks at I think, what were you at U.S. Fish and Wildlife at the time, scooped all of these hatchlings up and brought them back to us in Charleston to rehab them, and to do so very carefully because these things were really precious, with the hope that eventually we'd be able to turn them over to someone else. I think they ended up at a couple of aquariums. And we, the grad students, got to spend at least a month, I can't remember, just like carefully watching, checking, feeding, like poring over every detail of these alligators, like how many crickets they ate per hour. Like, the first one took 3 poops today and the other one hasn't pooped in 2 days. So, we're on poop watch. And it was all like under this guise of being sworn to secrecy, as if like the folks in the sea turtle lab next door, if they heard about them, they were going to break in and steal the hatchlings like they were in the Italian Job or something. But where Lou fits into all this story, other than the fact that Lou is the reason that this was all happening, is that I remember one day I got an email from Lou tipping me off that when I came into the lab the next day, I needed to bring all my camera gear. And this is relevant because in grad school and to this day, I got really big into wildlife photography because of Lou and because of Drew Crane. And because, Lou probably knew that I was chomping at the bit to photograph these animals. They're just so incredible. You don't see them in the wild. You barely see them if you're lucky enough to find them in an aquarium. And because he knew that I knew that he also wanted to have a full-on alligator photo shoot with these like perfect little hatchlings. And that's how we ended up the next day out in the dirt and the sand somewhere on Fort Johnson, with Lou laying on his belly in his khaki shorts and polo taking pictures of this hatchling alligator. And in fact, let's see if I can get this to share. Like, this is just one of the photos that we were able to get that day. And I still think back to this photo. It's been 11 years now since I took this, and the photo itself is a really cool thing to have and think back on. But the point of the story isn't really the photo. It's that most of the memories that I have of Lou was just how, amidst the craziness that was his life, 2013 to 2015, he spent so much time and energy amidst all of that, making sure that we, his graduate students, always felt connected to him and important to him. And just in the process of like trying to put this story back together, I pulled up old emails I had from him. And it wasn't just this. Like, this was just obviously the neatest one because it's got a white alligator in it. And I hoping Thomas Rainwater actually tells the accurate and correct version of this story too. But yeah, so in the middle, that's just the story that I felt compelled to share, and I can't wait to see you all tomorrow. And that's it. Bye, everyone.
So the Lou Guillette Jr. Outstanding Young Investigator Award is given to a young investigator working in some aspect of endocrine disruptor field. Someone who embodies Lou's love of science, is a good communicator and a terrific mentor. It provides a measure of immortality to Lou and his vision of science. So this is actually the only award in the endocrine disruptor field. And it's funded through an anonymous donor, and it's overseen by HEEDS. So it consists of a $5,000 monetary award. So awardees are chosen then based on their science, communication skills, and love of mentoring, and that's shown by some of the mentors writing letters about them. So there are 5 award winners so far. Kelly Ferguson from NIEHS, Rita Strakowski from Michigan State University, Genoa Warner from the New Jersey Institute of Technology, Ann Starling from University of North Carolina, and Chris Casotis from Wayne State University. So 2 of the winners are here today and are going to present some of their research. The first one is Genoa Warner, the 2022 award winner from the New Jersey Institute of Technology.
I'm really honored to have won this award. I was the 2022 winner. I didn't know Lou personally. My first Gordon conference, my first real exposure to the endocrine disruption field was that 2016 Gordon conference. But I've heard so much about not just Lou's science, but Lou's mentoring, and I think that's that's really important part of this award. And really, I brought some of my students here with me. I just want to say, I guess I take that part very seriously. Let's see. So I'm an assistant professor at the New Jersey Institute of Technology, which you might not have heard of. It's in Newark, New Jersey. It's a public polytechnic school. And I know we have some students in the audience here, right? I put some slides in here to talk about my career path because I'm a chemist, really. This is going to be a little bit different from all the developmental biology we've heard so far. So I'm going to tell you a little bit about how I, as a chemist, got into endocrine disruption. So I was a chemistry major as an undergrad, and I did a chemistry PhD, and my interest was always in environment. And I ended up working on endocrine disrupting chemicals. I don't know, it wasn't planned. My first project as an undergrad, I was working in a green chemistry lab, and we were working on making a chemical to replace BPA. Although when I first joined the lab, I was just like, I'm gonna do science. And then I realized after that's what we were doing. And I was so into that I wanted to do an environment-focused PhD. And so in my PhD, I was working on environmental remediation. And that was also focused on endocrine disrupting chemicals. And that was when I really got hooked. And I told my advisor, I want to do more EDC stuff. So he brought me to the Gordon conference. And that was where I met my postdoc mentor. And I got into the biology there. So that was how I totally switched, bench work at least, fields to do a reproductive biology postdoc. And so that was where I started studying the ovary and phthalates, which are endocrine disrupting chemicals that I'm assuming we've all heard of. Oh, there we go. So there's my postdoc where I learned how to handle mice, which was a big jump from doing a lot of synthetic chemistry before. So there we go. So now I have my own lab, and my own lab puts all these things together. So we study endocrine disrupting chemicals, we do toxicology, but we're also trying to understand things from a chemistry perspective and using lots of chemical tools. In this last little bit is that I really try in my work to do research that's going to make a difference, to try to do some little tagline policy-driven science. So my lab has 4 different areas of research. The first is trying to understand mechanisms of endocrine disruption. So we're using chemical biology tools to do this. Historically, I've been working on phthalates, so most of this is in the phthalate area. We have moved on to studying phthalate alternatives because there are so many replacements these days. And that's what I'm going to talk about in the science coming up. Our other areas of study are in nanoplastics and the placenta, and looking at PFAS content in artificial turf. This is, very much in the policy-driven science area. I actually usually don't advertise that I do this. I don't want to piss anyone off before I've published this, but I figured this is a fairly safe audience. And one of my students has a poster on her work in this. There we go. So I'm going to talk about phthalate alternatives, because alternatives are everywhere, and consumers love alternatives. We want things that are free of stuff, free of phthalates, free of parabens, free of things. And they say, free of everything, but they don't tell us what they put in there instead. And so I started looking into that. This was a screenshot that I took when I was shopping for my chemistry lab, setting it up, trying to buy tubing. We use a lot of plastic tubing. This kind of plastic tubing is up to half its weight in plasticizers. So lots of plasticizers in there. And the description for our tubing said, free from orthophthalate DEHP plasticizers, which actually that phrase chemically doesn't really make a lot of sense. But what's right here is DEHP. This is our classic phthalate plasticizer. Phthalates are by definition ortho. Ortho means that these two side chains here are right next to each other. This chemical right here, these are para-substituted. The side chains are now opposite each other. So these are what we call isomers. They have the same types of atoms and the same numbers of each, and we've just slightly rearranged the structures. And I'm pretty sure this is what they're referring to when they're saying free of orthophthalate DEHP plasticizers. They mean it's not DEHP and it's not ortho-substituted. The most common phthalate replacement is this chemical, which is para-substituted. So all we've done is move this side chain over here. I first started getting interested in this chemical when I heard about it from an epidemiology study at the University of Illinois, where I did my postdoc. They have a pregnancy cohort there, and they've been measuring phthalates in urine, and they at one point decided that they wanted to start measuring some alternatives. So they picked a handful of alternatives, and one of them was DEHTP, that chemical I just showed you, and they found more DEHTP metabolites in their women than any other chemical that they measured, like way more than any of the phthalates. So one of those grad students came running over to my lab, which studied phthalates, and she came to me because I'm a chemist, and she's like, do you know what this chemical is? Are you guys studying this? And thus all this line of research was born. It turns out this is a replacement made by Eastman Chemical Company, and they call it Eastman 168. And they advertise it for all sorts of uses in plasticizing, basically anywhere you've used DEHP. And there has been some biomonitoring of DEHTP and its metabolites in humans. So, for example, from 2009 to 2016, there was an annual increase of 50% DEHTP metabolites per year in people. So it's been on the rise significantly. And now there's more DEHTP in most of us than there is DEHP or any other phthalate. And this is consistent in NHANES as well. So the goal of this line of research is, to look into the toxicity of DEHTP, and in our work this is focusing in the ovary. But not only do we want to know if it's toxic, but we want to know if it acts like this. If making this structural change makes a difference in its toxicity. All right, I don't think I need to tell this audience too much about the ovary. When I was talking to my own department, I have to tell them a lot about biology, and they still roll their eyes. I'm like, we're chemists here. All right, so, ovary, source of female germ cells. We have focused on two different main functions of the ovary, the development of follicles and the production of hormones. and I'm going to talk a little bit about development of follicles and how we have used that as an endpoint to measure phthalate toxicity. So there we go, folliculogenesis, development of follicles. Our primordial follicle pool here is set at birth, and they develop through this process here where they become eventually antral follicles, which ovulate or die. And so we're interested in the progression of their development and if there is any damage to our follicle pool. Yeesh. This microphone is heavy. I'm not used to having a big microphone in my hand. Okay, so we did a pilot experiment. This was like really early on in getting my lab started. We did a pilot experiment. This was in vivo, so we took young adult mice and dosed them with DEHTP for 10 days. We collected ovaries and blood, and we wanted to see effects on folliculogenesis. And we picked doses that were environmentally relevant, similar to phthalate exposure. And then we had one really big high dose because we've done a lot of phthalate studies in the past with these doses so that we can directly compare them. Okay, so here's a little bit of results. What we are looking at here, over here, these are the different types of follicles. So as they progress, until we get to the end, which are the abnormal ones. And this is the percent of follicles in the ovary. So our yellow bars are our controls. So these are the number of follicles of each type that are expected in unexposed ovaries. And then when we expose them to DEHTP, we have decreased number of primordial follicles, increased number of primary follicles. This isn't only in the highest dose. And then we have this also potential borderline decrease in preantral follicles. So this is somewhat suggesting we have an acceleration in our follicle development, that more of these are becoming these faster than they should be. And then we have our slight decrease here, and also all this stuff going on with the abnormal follicles. And so we're wondering, one of the things that we've been looking into is this acceleration continuing all the way to the antral follicle stage, or is it only going so far and then there's damage and they die? And one of the other endpoints that we've been trying to associate this with is with cell cycle, because there's a lot of cell cycle that's necessary for follicle development. And so we use this as an accessible marker to be able to try to figure out what's going on. And phthalates tend to screw with the cell cycle. They can inhibit cell cycle progression. Sometimes we see no cell cycle, which means less cell death, but that is not always what we see. And, oh no, there we go. I think I simplified this slide from before. I had another line there that was basically explaining that when we do the same analysis with DEHP versus a phthalate mixture versus DBP and all these different phthalates, we get different effects. All of them screw with the cell cycle, but some of them are moving some of these factors up and some of them are moving other factors down. I deleted all the extra ones because I thought it was too complicated. So what the arrows that are left here are what DEHTP specifically did, which it turned out was fairly similar to what we saw from DEHP, different from what we've seen from other phthalates. And then lastly, we also saw some inhibition of pro-apoptotic factors. So this was a pilot study, our very first one, so we're still trying to figure out what's going on here. But if we looking back at our overall goal, which was to figure out if this is an ovarian toxicant, it certainly didn't come back with nothing, right? We're not giving DEHTP a pass here. It seems to disrupt some aspects of ovarian function. And the effects were not exactly the same as phthalates. I showed you the cell cycle stuff, which was fairly similar to DEHP. We saw very few effects on steroidogenesis from this phthalate, which is another area that phthalates target. So some effects were the same, some effects were not. This was just one phthalate. though, or one phthalate replacement. It's not technically a phthalate, it's a terephthalate. And so what we're looking for to do in the future is to not just study one chemical, but to study a mixture. And we've done a lot of studies using a mixture of these 6 phthalates here. The mixture's based on that study at the University of Illinois, where they measured phthalates in urine. These are the proportions that they were measured in urine. If we include DEHTP in that analysis, it'll make up 45% of our mixture. So that's where we're going next, trying to get from single back into mixtures. So that's our basic phthalate replacement toxicity assessment that we're working on. But what are we doing with this information? This is sort of the chemical perspective of my work, is that we're not just trying to figure out if something is toxic. What we really want to do is to figure out if we can create something safer, or even identify something safer, a safer replacement. And so we get into this problem. We get into our problems where we have regrettable substitutes that just do the same thing as the chemical before it, like BPA and all its replacements, because we don't check for toxicity when we're developing our compounds. And I'm specifically referring to endocrine disruption here, right? We do not check for endocrine disruption. We need to do this. We need to design with purpose. We need to incorporate identifying endocrine disruption into chemical design and then commercialize it later. And so what we're working on, there we go, is rational design, collecting all the information necessary to do rational design. In order to do this, we have to, understand the chemistry, the structure, the structural components. So how does changing ring substitution, we have our side chains next to each other versus across 1,3 or 1,4 here, how do these structural changes systematically change the toxicity? And then on the biology side, we have to actually understand the mechanisms. Phthalate toxicity mechanisms are not well understood. We really don't know what phthalates are doing exactly. It's a whole lot more complicated than something that is a simple estrogen mimic. So we're trying to work on this too. But we put all this together, we understand how changes in structural features impact the biology, that's structure-activity relationships. So that's what we're trying to do with this information. We want to see if we can figure this out so that we can identify or design safer plasticizers. This is a screenshot from the PlastChem database. This is a really cool project that identified over 16,000 chemicals in plastics, mostly through mass spectrometry. Just taking plastics, extracting them, seeing what you get out of it. And there are loads of plasticizers in here, a small portion of which are phthalates. And so, I keep clicking the wrong way. I went through the PlastChem database to see what kind of other plasticizers are out there that are not phthalates. And here's some of the different structural features. So we have things like, this is a meta-substituted phthalate. We call these isophthalates. So this is a different ring substitution pattern. There are other terephthalates out there besides the one that looks like DEHP. We have these hydrogenated things. This is DINCH. You might have heard of DINCH. Well, not only is there DINCH, but there's other stuff that's hydrogenated. So this one incorporates 2 different structural changes from DEHP. It's hydrogenated and it's para-substituted. And then there's also these tri-substituted things out there. So there are loads of phthalate alternatives that we've never heard of, that manufacturers are putting in products and they don't tell us. They just throw them in there. They haven't tested them to see if they have the same properties as DEHP. And these need toxicity studies. But I'm also hoping that we can figure out if maybe making some of these structural changes will make them safer. And if we could find one that was safer, that would be much easier than trying to make our own and present it to a company. If we could be like, this one is better, try to use that one, or use this class over that class. But we need a lot more information before we're able to figure that out. So the future directions where we're going with this project overall is that we're trying to figure out structure-activity relationships of these different classes of phthalate-related compounds. We're working on figuring out mechanisms using non-target methods to try to figure out what the receptor genomic targets are of these compounds. And we're getting into AI a little bit with a cheminformatics analysis of structure-activity relationships. Because basically we're going to be collecting too much data to be able to analyze this manually. So we're going to get some informatics help. So in conclusion, we are overall profiling structure-activity relationships of families of phthalate replacements. We are trying to identify mechanisms by which they act as endocrine disruptors. There we go. That's the actual conclusion. There we go. We are trying to understand mechanisms of action of these phthalates and their alternatives, right, to identify or design safer chemicals, right? This is turning out to be an urgent problem because the phthalate alternatives are already out there. We're all exposed to them already. This is, I think the last line here is maybe the most important thing, which is that we need chemistry, we need chemists to do this. And when I'm around an endocrine disruption audience, I'm usually the only chemist, besides my PhD advisor, who facilitated my entry into this field. And chemists, I talked to my department about this, they don't know anything about this too. They design chemicals and they do not think at all about toxicity. They just want to start, patent it and spin off a company. They don't think about toxicity, they don't think about any of that kind of stuff. And this needs to change, right? We need chemists here, we need, more endocrine disruption people getting out there. We need more, more collaboration, really, to make a difference on a problem like this. There we go. So that is it. Thank you all for listening. I would like to thank the members of my lab who contributed to all this work, and our funding, and to HEEDS for everyone for this wonderful award and for having me out here today to talk about this.
So thank you guys again. Really honored to be this year's award winner. And I'm excited to share a little bit of what we've been up to. So I thought I'd step back. Probably most people in this room understand endocrine disruptors, although that would be a weird segment of the population probably. But there's this recent review from Michelle and Merrill and a team of great folks in this field defining key characteristics of endocrine disruptors. So the Endocrine Society defines them as any chemical or mixture of chemicals in the environment that interferes with any aspect of hormone action. And this can happen both directly at the level of the receptor. So a chemical can trick a hormone receptor into thinking that it's the endogenous hormone, or can inhibit the endogenous hormone from binding. And then there's a whole suite of indirect interactions as well. They can interfere with production, transport, distribution, etc. And of course, these chemicals are present all over, in all aspects of our daily lives. And so we spend a lot of time in my group thinking about chemical mixtures. And so a few kind of take-homes there, we're exposed to hundreds to tens of thousands of chemicals on a regular basis, right? It starts during gestation, and it continues chronically through early development and really throughout our lives. Recent estimates put this at about 350,000 chemicals and mixtures on the global market. And what we do know from a variety of groups are that the actions of endocrine disrupting chemicals and other chemicals acting through similar mechanisms can be additive or synergistic, right? So you cannot consider toxicity in isolation. And this leads into these ideas of something from nothing and a lot from a little, where we get unexpected toxicity when we start to combine chemicals. So I thought I'd give just a little bit of a snapshot of what we work on in my group. So we start at the environmental health screening, and that's where a lot of our projects start. I'll talk a little bit today about the ethoxylates, but my first PhD student is working on liquid crystal monomers. These are chemicals used in all of your favorite LCD screens. So they're encapsulated within the glass outer panel, but as we found out, they leach out of that panel and result in human exposure. And they're pretty uncharacterized in terms of toxicity. We certainly work on some of the classics, things like PFAS. We work on some flame retardants. We also do some work on artificial turf. We have a small area of work in our lab looking at hormonal contraceptives as well, more from the perspective of metabolic health than reproductive health. A lot of these projects find their way into more concrete health effect toxicity testing. So we work primarily in zebrafish as our in vivo model. We do a little bit of mouse work in collaboration, but primarily fish is our model, and most of our projects have found their way down there. We've done a little bit of human epidemiological work, primarily around the liquid crystal and artificial turf. So we have a small cohort of kids in Detroit that we've followed around playing soccer on artificial turf and measuring the chemicals that they're exposed to on turf versus natural grass. Trialing interventions and seeing what chemicals show up after they use one of these fields. In liquid crystal monomers, we know almost nothing about human exposure, right? So we've been going into homes through our Center for Urban Studies at Wayne State and trying to better understand how often people are exposed to these chemicals and at what concentrations. We do a little bit of work around mixture modeling and reproducibility, and we have a fairly large focus on metabolic health in the lab, but a small focus on kind of the basic mechanisms of how fat cells develop. So we have maybe too many things going on in the lab, and my lab always threatens me to not come home with new projects, but I was really good last year, so I think we're maybe due for a new one. But today I thought I'd tell you a little bit about this ethoxylate story. So this started during my postdoc at Duke. During my postdoc I worked in an environmental chemistry group, Heather Stapleton's group. And so they're really strong exposure scientists, right? So they're really good at figuring out what chemicals are present in the environment, what human exposure looks like, and not so much the biology side of things, right? So I was brought in as the biologist to say, what is the effect of all these chemicals that we're finding? And so one of the things that she was really interested in was household dust. So what you might not know is that all of the chemicals that are in your residential indoor environment find their way into your house dust. So whether that be the pesticides that you bring in on your produce, pesticides that you bring in on your clothing and your shoes, all the plastic products that you're bringing into your home, flame retardants in your sofa, your electronics, all of these things find their way into household dust. So if you do a non-target assessment of household dust, which we did in collaboration with Lee Ferguson at Duke, you find about 10,000 different chemicals in the average household dust. So we're constantly exposed to this huge complexity of chemical space. So this study collected about 140 households in central North Carolina, went to all these homes, vacuumed their main living area, and then we took the dust and we measured specific chemicals present. We looked for chemicals that we didn't know, right? We took an untargeted approach or a broad approach to say what else is present in the dust. And we looked for biological activity. So we wanted to say, do these interact with hormone receptors? And that led us down this path of metabolic health. So we started finding that these dust samples, or the chemicals present in these dust samples, could dysregulate hormone receptors that were important for fat cell development. And so we started looking directly at that process. And so there are some really nice assays. So this one shown up here on your top right is testing the ability of a chemical, or in this case a dust sample, to promote fat cell development. So we start at a pre-adipocyte, or a precursor fat cell stage, and we follow those cells through after exposure to a chemical or a dust sample, and we can measure the extent of differentiation, or the extent of development into a mature white fat cell. And so we can see that generally speaking, at higher concentrations of dust, as you increase on the x-axis here, you get an increase in lipid being packed in by these cells. So they're becoming more mature, packed lipid fat cells. And this is happening at really low concentrations. So this is microgram amounts of dust, right? So very low amounts of dust. The really cool piece of this, I thought, was that when you looked at associations, you found that the greater the ability of the dust sample to promote fat cell development in the lab, the greater the body mass index of the person living in that home. So that really suggests there's an association there, right, between the chemicals that we're exposed to in our home environment or in our broader environment and the metabolic health of those people. Now, is that causal? Is there some other relationship going on? That's what we've been spending some time trying to figure out. And so we spent a little bit of time trying to figure out what were the chemicals actually causing this. And so we honed in on these surfactants, mostly through non-target analysis work done by Lee Ferguson. They're high production volume chemicals. They're used in laundry detergents. They're used in hard surface cleaners. It's hard to find any hard surface cleaner or laundry detergent that does not have these chemicals. Paints, cosmetics, a lot of agricultural products as well. And as such, they're ubiquitous environmental contaminants. So they're found in sewage effluent all the way through to finished drinking water. They're also found quite reliably in indoor house dust. They're also found in clothing and on human skin. So there's a lot of routes to exposure to these chemicals. And so here's the Lou link. So there was some I was going to say old, but there's some slightly older work by Sumter and Jobling showing that the degree of ethoxylation, and I'll come back to this, seemed to diminish the estrogenic activity of these surfactants. But what you'll see is that over here, where you're increasing this ethoxylate tail, you do see this little bump in estrogenic activity. So there's also some work done in Lou's group on some similar surfactants used in Corexit. So DOS, not that dissimilar from some of these non-ionic surfactants that I tested, was able to pretty robustly stimulate fat cell development. So we said, this is a good potential target to look at. So we asked this question, do these surfactants dysregulate metabolic health? And so just to orient you to these chemicals just briefly, so they start with some sort of base compound, nonylphenol or octylphenol being some of the most widely described options. And you add this, what we're going to call an ethoxylate chain on the end of it. And so you have the base alkylphenol, That's summarized here on this version of it. And then you've got that ethoxylate tail. Now, what we've learned over a couple decades of using these chemicals is that they degrade into this base nonylphenol or this base octylphenol, and that's pretty appreciated to be toxic at this point. So industry's been moving away from what we would call these alkylphenols and to many different alternatives. One of those being alcohols. So here's an alcohol. We've got an alcohol base, and the same concept, we can add, that ethoxylate chain tail. All right, so we were curious, if we see activity, what portion of the molecule will be responsible, right? Is it this base compound, or is it this ethoxylation, this tail that we're adding to the molecule? And so we tested both things. So first we looked at just the base chemical, and we found, of course, all of these diverse base ethoxylated surfactants induced fat cell development. So that's the short version of that. So then we said, well, which portion of it? So this is just the base. This is without any of the tail added. And they have pretty limited effects on fat cell development, nothing for most of them. So we said, well, there's something about that tail that we're adding on that makes it, quite good as a surfactant. And so here we just held the nonylphenol base constant, and we varied the length of the tail, and we see actually this really nice inverted U, where we see this increase in the ability to promote fat cell development up to a certain tail length, and then a decrease. So, we were curious, will this have the same effect in a vertebrate animal? And so we use zebrafish. And why do we use zebrafish? Well, there's a few great reasons, but the mechanisms governing fat cell development are very highly conserved between a fish and a human. Things like adipocyte development, lipolysis, endocrine function, all very highly conserved. Adipose distribution into visceral, subcutaneous depots happens the same way in a fish as it does in a human. They have a heterogeneous cell population, and unlike mammals, it's possible to do very easily high-resolution internal imaging of adipose tissue. So what does this look like? So we treat during early development. So we spawn our fish, we collect the embryos. At 1 day post-fertilization, we start to expose them to chemicals. We expose them for 5 days, and then these larval fish we subject to energy expenditure testing, behavioral testing, gene expression testing, and then we age them out without further exposure until they're 30 days old, which is kind of a prime time point to look at the internal adipose. All right, so here's a brief picture of a fish, and I've just highlighted some of the adipose depots. So there's 38 distinct adipose depots in the zebrafish. They've all been cataloged painstakingly by John Rawls' group at Duke. And they're all partitioned quite cleanly into subcutaneous or visceral, much as they are in humans. All right, so I'm going to skip a whole bunch of what I just said that we did for the sake of time, and just come to the take-home, which is these chemicals also have these same effects in fish. So we see an increase in body weight of the fish that were exposed to these chemicals, and a vast increase in the adipose, or the fat in the fish as well. When we segment that out into visceral versus subcutaneous, we see a big increase in both visceral and subcutaneous, but a preferential increase in the visceral depots. So you can see these are the nonylphenols, these are some of the alcohol, these environmentally friendly replacements. They do the exact same thing. So as you increase the length of that tail, you get an increase in visceral adipose, which then comes down again as you increase further, and you get that exact same pattern in these presumably safe replacements. All right, so a few final notes to close out. I want to highlight that we still don't know a lot of the things that are present in a matrix as complicated as household dust. We still don't understand a whole lot of that, both identity of a lot of the chemicals present and then interactions between those chemicals. These alcohol ethoxylates, these alcohol surfactants are maybe another example of a regrettable substitution. As industries shift away from these, what I would call more legacy, nonylphenol-based products. Industry is also finding many creative ways to shift, including PFAS ethoxylates, which Heather Stapleton at Duke has been working on, which degrade into a PFAS, which doesn't seem safe. We find pretty reliably that when we test for effects on fat cell development in a cell model, that carries through quite well and did for these chemicals to whole organism responses, with effects on energy expenditure, activity, so our fish move either more or less depending on the specific chemical, gross body weights, and adiposity. And what I didn't show you is that we have a potential mechanism through thyroid hormone receptor. Although we have a bit more to do on that if we get further funding for this. So I'll stop there and think I have just a tremendous group of people in my lab. Matt LaFaux has departed, but did a lot of the early work on this project. He's now working for EPA. Roxanne Berube, another postdoc who's currently doing some work on this. And then some of my PhD students, Samantha and Kyle, have done a lot of work on this as well. And yeah, I just have a terrific group. And we've been funded through, NIEHS primarily and EPA as well. And there's a nice picture of a fish there, the fat in a fish. And thanks, I appreciate it.
Okay, thanks so much. So we're going to end the day with a panel discussion led by Nancy Denslow. And the discussants are Satomi Kono, Woody Woodward, and Matthew Hales. And after we let them talk for a few minutes, then Nancy will open it up for anybody else in the audience who wants to come up and say a few things.
I'm Woody Woodward. I work for the Fish and Wildlife Commission in Florida, and used to be the Game and Freshwater Fish Commission when I first met Lou. And our relationship went back to probably around 1988. And at that time, we had been doing a project in Florida to look at sustainable use of alligators. And one of the things was to collect alligator eggs for ranching. And ranching is collecting eggs, hatching them out collecting wild eggs, hatching them out in captivity, and then growing them out to harvestable size. And we were working on 3 lakes, which was Lake Apopka, Lake Griffin, and Lake Jessup, where the eggs were coming from. And none of those lakes had good hatch rates. They were probably in about 50% when we first started out. And so they were supposed to be up around 80, 90% based on research that had been done in Louisiana. And during that 6-year period that we were doing the study, the the Lake Apopka hatch rates went from about 40% or so, 40 to 50%, down to 3%. And so we knew something was going on out there, but we didn't know what. And of course, Lake Apopka we knew was polluted, and there had been some chemical spills there, and so we immediately thought there was some sort of acute toxicity going on with environmental chemicals, and the chemicals were agricultural, and there was also a chemical plant that nearby there that had spilled, and those things were trickling in, those chemicals were trickling into the lake. So anyway, we wanted to find out a little bit more about what could cause these problems, and one of the obvious things was something going on with female alligators, adult female alligators. And so we knew nothing about female alligator reproductive biology except for what we'd read in the literature, and that was somewhat incomplete. So we went to the University of Florida zoology department, and we were introduced to this professor, Lou Guillette, and his credentials were interesting. He's a professor, of course, and a good educator, but he also had done some work on reproductive biology of lizards, and that was good enough for us. So we entered into a 3-year contract and study with Lou to look at the reproductive cycle of female alligators. And we were interested in both morphology and endocrinology on that. And so Lou and I spent a lot of time going out and collecting adult female alligators for specimens, and we needed a robust sample. So we spent a lot of time on the road for about 1.5 years, and it was 2 hours one way and 2 hours back. So Lou and I had a lot of opportunities to talk about a variety of things, and everybody knows he's always interesting to talk to. And so, but one of the things that came up during our conversations was how to measure success in our jobs, and there was a number of metrics that Lou kind of threw out there, but a couple of them that stuck with me. Number one was he measured a large part of his success in the success of his students, collaborators, and cooperators. And looking around this crew, I think Lou would be pleased to see the fruits of his work. The other measure of success that Lou imparted on me was the contribution to science. And Lou was kind of a stickler that you weren't contributing to science unless you published your work in a peer-reviewed journal, book, or proceedings. And so he kind of stuck that with me, and I think it's done well, you know. And he would also help. And Lou was a great writer, so to him, he could just churn out publications one after another. A lot of us aren't that good at it, so it took a lot of work, and it took a lot of prodding by Lou sometimes for us to get things out, but he was pretty good at that, and he would help in any way that he could. Anyway, those were two things that kind of stuck with me over the years, and I think, you know, with this crew, there's been so many publications out and so much success that I think, like I say, Lou would really be proud of what he's done. And I'm proud of Lou, too. So.
Thank you. So now, if anybody has a story they would like to share, please come share it.
How about you?
My story? Yeah. Sure. I'll tell my story. I was going to tell a little bit of it tomorrow, but I can tell it today. So, my background is in biochemistry and molecular biology. I know nothing about environmental anything. I do a little bit now, mainly because, well, back then, so I got my degree here in biochemistry and molecular biology, and I'm still here, right? So I'm still here. So I had little kids, so I decided that a good career was to be a core director. So I was at ICBR, that's the Interdisciplinary Center for Biotechnology Research, and became the director of the proteomics core. Even that, you know, when I started that, I knew nothing about proteomics, so I had to learn that first to do that, but I did. And Shelley Schuster was the director of ICBR at the time. And he and Lou became good friends. And I think it was Lou's suggestion to start a new program called BEACS. BEACS stood for Biotechnologies for Ecological, Evolutionary, and Conservation Science. And so that established 4 different laboratories. And one of them was the molecular biomarkers laboratory. And I said, sure, I'll do it. I always say yes, so that's a problem. But I always say yes. And there were 3 other laboratories. One was a genetics lab to look at phylogenetic differences among ecological animals. One was a hormone lab that Lou was in charge of. One was a hybridoma lab, or an antibody lab, to make specific antibodies for environmental organisms. Anyway, so I said, sure, we would do it. And so what we decided to do in my lab was to do 2-dimensional gels. That's sort of a new technique, and not too many people could do that. It had a lot of technology involved. So we did it, and we had beautiful gels, but no clients, none. And so one day Lou says, oh, I have a friend visiting from EPA, Leroy Fomar. He says, and I'll bring him over and he can talk to you. So he did. And Leroy had these samples of fish plasma that he had collected from a river in Ohio that was highly polluted. So these fish were collected downstream from a coking facility. And he wanted to know if we could find him a biomarker for the cancer. And I was thinking, oh, Lord. And I said, sure, we can do it. We'll do 2-dimensional gels, and we'll look at the proteins. So we did. And so the proteins that came out, they were beautiful gels. And sure enough, there were differences in the plasma of the fish downstream from the coking facility compared to control fish that didn't have any cancer. And there were a lot of proteins that were different. We published on a few, but one that was very important was a large protein at the top of the gel that we could identify by sequence analysis, because a proteomics core can sequence proteins. So we identified it as vitellogenin. And so to me, I said to him, well, there's this egg yolk protein. I don't know what it means, but it's different. And he was so excited, and he couldn't imagine that we'd really sequence it. So he sent me all kinds of other samples and lots of other projects. Well, it turned out that it wasn't maybe necessarily a biomarker of cancer, but it was definitely a biomarker of pollution, of endocrine disruption that was also in that river. So because we were able to sequence it, we could make peptides that matched the sequence. We made antibodies. And then that apparently was a big deal in the US, because already in Europe, Charlie Tyler's lab and others had found that vitellogenin was a very good biomarker for estrogen contamination in rivers and other places. So, Leroy invited me to take my poster to CTAC meeting. I'd never been to a CTAC meeting. And I knew no one except Leroy. And I was just so amazed when there were 20 people deep in front of the poster, and I was like, oh my goodness, I didn't know. And so that's really what changed my career a little bit. And now I'm doing, environmental work. And then, what I need to also mention as part of that was because we were setting up the lab, we needed someone to help us with vitellogenin. We were making antibodies right and left, most of the antibodies that are now commercially available, we made. And we made them available to the community. And so one of the people that came by to apply for the job was Kevin Kroll, sitting here. And so he said, okay, he was interested in the job. And I said, well, we have to work on this protein, vitellogenin. And he said, like, what? He said, oh, no, this is what I know everything about. And so Kevin joined the lab, and because of his knowledge about fish husbandry and fish toxicology and fish physiology and reproduction, we've been able to get into that area a little bit. And so that's my story and how Lou really changed the career trajectory that I was on. So, other stories, please.
My name is Camille Fiani, Dr. Camille Fiani. I was actually, Buzzy was my chair. I was trained under her. And the reason I wanted to speak is because everybody else has been speaking about Lou as a scientist. I'd like to talk about Lou as a communicator. I came to University of Florida after having spent many years as an environmental scientist working in communications. I was always interested in science, meaning the connection between humans and their environment around them. It was very important. So I was very interested in just kind of learning a little bit more about that and how we can teach scientists really and students how to communicate their science to the public. When I came, I was doing my anthropology, learning my anthropology, and very interested in that. And Lou brought this idea, what about creating a course on communicating complexity in science and showing students as well as scientists how to communicate their science to the public more effectively through the media. It was something that was extremely important to him. I was very proud to work on the course with him, to help teach it with him, and Thea Edwards and others who participated. But what really was most, what impressed me the most is that as wonderful a scientist as he was, the communication aspect, making sure that you communicate your science without losing its integrity as you communicate it through the media, and making sure that you maintain the control over your narrative was extremely important to him. So even though I didn't prepare anything, the reason I wanted to come up here is letting you know that especially now, it's more important than ever. And ensuring that as you communicate with other journalists, some of them who probably don't understand, didn't come from a science background, don't even understand your science, it is critically important to make sure that the narrative is controlled by you and that the messaging, that key take-home message is brought clear, made clear, and moved forward, moves the communication, moves the, not just the messaging, but moves the public to actually adhere to that message and form their movements based on that message and making sure the message that is communicated is the right one. It was an honor to work with him, and I just wanted to share that since everybody talked about the scientific aspect of him. The communications aspect was so important to him as well. So, thank you.
So, speaking about communications, I actually want to stand up here right now and represent Joe Case, who had to leave earlier, but Joe is the Director of Communications for the University of Florida. And he and Lou had become very good friends as well when Lou was still here, and of course that continued on. But one of the publications that Joe oversaw is the Explorer magazine, which really highlights and features some of the key work that's coming out of the university. And so of course, obviously, Lou is featured in this copy, but Joe wanted to read this line because he just found this to be the most hysterical thing. So, for most people, the business end of an alligator is the one with the big teeth and an even bigger mouth. But for U.S. zoologist Lou Guillette, the size of an alligator's penis is more important than the size of its teeth. And it goes on. I have several copies of this. If anybody would like one as a keepsake, please let me know and I'm happy to share.
Great story.
It's one of those things that you'll regret later. So my story is I had the opportunity to go to Australia with Lou and Ashley Boggs, and I was so excited. Can you imagine being with Lou in Australia, in Darwin, Australia? So I was like, this is going to be insane. And so, true to fashion, we ditched the conference like 2 days. And so at the end, just to go and take pictures, which was amazing. And so we were going on all these back roads and doing all these amazing things. And as we were driving, like Ashley was like seeing some really cool things and Lou was seeing some cool things and taking pictures. And I remember looking way out and seeing some cars and some vehicles that were upside down. We're talking like a quarter of a mile into the nothingness and like upside down. And I'm like, what idiot like drives a car into the middle of nowhere and tips over? So anyway, fast forward, we're driving down the road and all of a sudden, I don't know where it came from, but there was just water across the road. A little bit. And so we were driving and we're in like, I don't know what kind of car it was, but just imagine like a Toyota little kind of car. And most of the cars that are going by us have those like snorkely things. So, and I was like, oh, that's interesting. Wonder what that's for. And so we're driving and, you know, some water is coming across the road and it's getting a little bit higher and a little bit higher. And I'm like, oh, this is interesting. And then it gets to a point where, you know, he's like, maybe we need to turn around. But then all of a sudden it got so high, we couldn't actually stop. And the water would have gone over the tailpipe, and then so we couldn't stop, we couldn't back up. And so I'm like, oh, and I even think at this point I took a picture, you know, and took a little video with, you know, Ashley was in the back, and because I was like, this is it, might be how we die, and I don't know, it's true. And so we're driving, and all of a sudden the water gets higher and higher, and we can see way up ahead there's a bridge. And then it flashed back to the car that's way off, quarter of a mile over, and I'm like, this is how that happens, right? So cars get, you know, flushed off, and, you know, here they are, end up. And this is, by the way, like crocodile territory. And so I'm like, I'm freaking out a little bit, but at the same time, Lou is driving, right? So Lou is driving, and I'm like, we have to make it out of this. I mean, this can't be how we all, this just doesn't make any sense. And so then up ahead, there's like a bridge where then you can see where the water is just rushing over. Now, it hasn't rained that I've seen in days, so I'm not knowing how this is happening, but the bridge just kind of is just going like crazy. And then I'm like, that's got to be the deepest point. And so we drive, and you can see where the road comes up out of that. And so we're driving, and just as we hit the bridge, you can feel the car, all four wheels are off the ground. And you can feel that we are just floating. And, you know, I'm white-knuckled, Lou is white-knuckled, and I looked at literally white-knuckled on the steering wheel, so I'm like, okay, this is bad. If he's worried, I'm worried, but here we are. And then just as we were floating and you couldn't even see the rails anymore of the bridge, just as we were floating toward, like, what would be off the road, you could feel it just catch. And then we slowly caught and then just got up out of the water and, you know, and kind of went off. But to me, like, I look back and I'm like, that was so symbolic. Like, you know, it was just so symbolic of, you know, here we are in this sort of, you know, dangerous, but also just sort of treading through life like people do, and he pulled us out of it. You know, and I kind of knew he would, but today, you know, this, you know, Ashley and I, you know, we still talk about it to this day, and so hopefully she's kind of watching this, and yep, can be like, that was a really cool memory. So, anyway, thank you.
Hi, I'm Thomas Rainwater. I work with Lou. I'm giving a talk tomorrow late in the afternoon, so you'll hear more about the details about my relationship with Lou, but my talk's, I think, like 12 minutes, so I'm not going to have enough time, so here's a story I wanted to tell you tomorrow. So while we're talking about international trips with Lou, as Matt mentioned, in 2013, so I did my graduate work in Belize, Dream Project. I'll talk about that more briefly, but and you'll see tomorrow, in a very indirect way, Lou made that possible for me, even before I met him. And you'll see about that more. But anyway, I met Lou later on, and we became friends and worked in his lab. And I wanted to take him back to Belize one day and show that this is where I used to work. This is where I kind of cut my teeth on crocodilian, so to speak. And it's a cool place to go. And so I'd love to take you down there and show you where I used to work. And you're going to love it. And so Matt and Russ and I and Lou, we went down there, and we had lots of adventures. And one of the things we did was we had this long-standing American crocodile survey project going on the coastal zone of Belize, and including this atoll. It's about 30 miles off the coast of the Belize mainland. And every year or two, we would go out there and we survey crocodiles, do eyeshine counts, and catch crocs and mark them and stuff. A really cool project. And so that's the first place we went. We landed in Belize City, hopped in a taxi, went down to the wharf, caught the boat, and took the hour drive out to the island. And we spent a week there. And at the time, what they did was this outfit would sometimes let tourists come in and live. And then this is an outpost. I mean, it's just a couple of little cabanas on the edge of a coral atoll. You have a couple of trees, and it's beautiful, but it's very, very isolated. And but people can pay to go out there and live and go out with researchers if they want to. So we had a handful of folks with us for a week. We didn't know them until we got there. They went out with us for the week. And of course, Lou was, in his element, because not only were we doing cool stuff, and of course he had his camera and all this incredible photography options and opportunities, but he had these families, including these children, that were he just was so excited to include them in everything we did. So at night, we would have to have a quick lunch, and so what are you doing tonight? We're going out and look for crocodiles. Oh, can we go? Of course. So, Lou needs to put himself to explain what we're doing. My job's easy. I just go out there and organize things, and Russ and Matt and I will do most of the capturing. And Lou is just on. He's just teaching these adults and kids. I've got pictures of all this. And it's great stories, but and every night we do it for them. We go catch crocodiles. One night we would go look for iguanas, and there were boa constrictors everywhere, which is a really cool place. And the kids were really into Lou, and he was so engaging with them that really made a difference. And we caught a huge crocodile behind the place, and they got to come check it out and look at it, get pictures with it, and just an amazing experience for everybody, but especially these two young women. I think there was a woman, and she had her daughter and her niece, and I think they must have been pre-teens or something like that, maybe, 10 or 12, maybe a little older. So we were about to leave to go back to the mainland and go to our next destination. We asked them, so what's your trip? What are you going to be doing next? Well, we're heading down south to some place to hang out for a couple of days, and okay, it was so fun, great to spend time with you guys, and hope you have a great time. It's been wonderful. And she said, well, where are you guys going? So we're going to this place in the jungle called Lamina Outpost Lodge up in northern Belize. It's on a freshwater river, and we're going to be doing similar things. And that sounds really fun. And so that was it. So we were about to get out on the boat to go back to the mainland, and she said, hey, by the way, I just called that lodge place you mentioned. We're going to go with you guys again. And so they totally changed. They changed their entire plan so they could stay with us for another week. Because you guys had all these incredible adventures, and so Lou was delighted by that. So well, just follow us. So we took the boat back to the mainland, hop in a cab together, a little van, go to the airport, drive 2 hours north to the New River, and then we get a boat that met us from the lodge to drive up. It's like a 1 hour drive up this river to the lodge, which is up in the middle of nowhere also, but on a freshwater river. We get on the boat, start going, and then, we're maybe, a mile or two from the lodge. We get up this river, and all of a sudden it opens up to this big giant lagoon. It's like 25 miles long by a mile wide. In the distance, you can kind of see this hill, these Maya ruins, and next to those is this lodge. You can see it over there. It's like, this is so cool, Off we go, across, and all of a sudden the heavens open up, and we just get absolutely just destroyed by rain, just drenched, soaked, everything. We're just like, oh. And it's just, but hey, this is an adventure, this is part of the experience. You're gonna love this stuff, And so they smiled, and everybody's cold. And we get up there, we get into the lodge, and we walk up the hill, and there's this big open veranda. We get up there, and of course I worked there for 4 years. And I knew the staff and the people. I was so excited to see them. I hadn't seen them in 10 years, and I wanted to introduce everybody to everybody. We're talking, and so. We're on this veranda, and it's kind of the off-season, so it's just us. It's just like Lou, Matt, Russ, and I, and then this woman and her 2 daughters, and they rush down to their cabana because they're cold and want to get into some warm clothes. And so we're standing there talking, and I'm introducing, hey, Lou, this is so-and-so, and this is so-and-so, and we're talking in it. Hey, nice to meet you. And I kind of noticed, kind of over my shoulder, there's a couple, like 4 people walking up the veranda and kind of sit down right over here by the tables where you eat dinner and stuff, and we're just talking. I didn't even look at them. They've been on a tour. The Maya ruins are right next door in a jungle complex, a beautiful place, and they had been on the tour, I guess. It happened all the time. I didn't pay much attention. So we're talking to Lou. This is Lou. I used to work here, and this is da-da-da-da-da-da-da. And Matt's kind of looking over my shoulder, kind of looking over there, and, you know, these people, I'm like, man, what's he looking at? And we're just talking, and we're talking everything. And this is Lou from Finally, and I look over there, and it's Leonardo DiCaprio and Bradley Cooper sitting there, and their girlfriends or dates. And they had actually, I guess DiCaprio owns the property, or maybe an island off the coast of Belize, and they had flown in by helicopter to have lunch, to do the ruins tour, and then to go back. And we were sitting there, and I was like, you know, we just kept talking to Lou, and just, we totally ignored him, because Lou was he was the focal point. All the people there wanted to see Lou more than they wanted to see those guys. It was really cool. And then they ended up walking down, and we heard the choppers coming across and came and picked them up. Off they go down the steps, and I think Matt was hoping that we could have talked to Leonardo and gone out with us and talking a little bit of conservation, but they had to leave. But and then I'm almost done. But the next day we go do the ruins tour. So we're working on crops at night. So during the daytime we have time to ourselves. So we do the ruins tour. We go walking around, and there's all these beautiful, Mayan temples, kind of reclaimed by the jungle, and there's trees everywhere, and vines, and the howler monkeys, and there's all kind of cool stuff, and excuse me, and then we finally come to the high temple, and I had photographs of this in my talk. I might work them in tomorrow if I can. I pulled them out because I was scared it's going to go too long, but anyway, it's this mountain, this huge Mayan temple, and as everyone's mentioned, Lou, he's into photography. He loves his sunrise and the sunset photos especially. And so we climb up there. I've got videos. It's almost straight up. There's a rope hanging down. There wasn't a whole lot of safety protocols there in Belize at that time. And so there's this rope. You climb up to the very top, and we get there. And I wanted to see it. It's breathtaking. You can see over the entire. You're way above the canopy of the forest, and it's just lagoons and pine savanna across the river. It's just fantastic. And Blue said, we're coming back tomorrow at sunup to get that picture by God. So next morning we're up, 4 in the morning, get all our gear on, and walk through the forest, and get up, and we climb up the top, and we have some spectacular photographs of the sunset. And of course, the woman and her daughters are with us, one of the most important parts. Shortly after we saw the movie stars and they departed, we sat down and had dinner, and we're kind of still wet, but then they come back and they're all warm and dressed and feeling good, and they sat down. And so, guess you just missed. And we told them, and the two young girls were just devastated that they had missed Bradley Cooper and Leonardo DiCaprio. It just broke their hearts. They didn't believe us in the first 10 minutes, but anyway, I felt bad for them, but they left at the wrong time. Anyway, so we did all that, had a great week, and as we were leaving to go back, and hop in the boat to go back down the river to go back to the airport to go home, we said our goodbyes to all the staff. You're there for a couple days, you make friends with everybody. It's almost like a family, which is kind of Lou's thing. And we had a great time. And we get in the boat, and we got all our luggage and everything, and we're saying goodbyes. And we get pulled away from the dock and have to go across the lagoon. And just like on the way in, the heavens opened up again, and we get absolutely just drenched. And she's like, "Good God, guys, does everything have to be an adventure with you?" So anyway, that's kind of how I feel about with Lou. Everything we did was always an adventure. It wasn't always unsafe. It usually was safe. It was always a fun adventure.
Thank you.
I actually was originally only going to speak at the end and said most of what I needed to today, but I just wanted to thank everybody again for coming, and I know we all miss my dad, his laugh, his smile, his presence, knowing everything would be okay no matter what because he was there. Thank you for your stories today. I just want to thank everybody for making his life special, full of adventure, laughter, love, and learning. And we continue on in that today in the same spirit, and thank you for carrying that with you into the years to come. All right.
Yesterday was just unbelievable. It was so interesting, so exciting. Had me have a couple of tears. The memories and stuff, it's really something else. So we're going to just continue in this vein today. And I'll just start by introducing our first session chair, who's Ethan Sheely. And Ethan is a graduate student in the Savannah River Ecological Laboratory at the University of Georgia.
Hey there, everybody. For those of you that don't know me, I'm a PhD student in Ben Parrott's lab, and I just got to say, I've heard nothing but good stories about Ben's time with Lou and everyone he worked with while he was at MUSC. So it's great to be able to put some faces to the names that I've heard about. But yeah, I'll get us started with our first session here today. First up, we've got Dimitris Baropoulos.
All right, well, thanks. It's great to be here. There are some incredible storytellers here, and I'm not a storyteller. I'm a data slinger. And let's see, how do we move? Okay. Forward. Oh yeah, so is that a good question? It's like, not just it's sort of like, what's the question, but immediately after that, is that a good question? And I'm using Comic Sans. Teresa told me about this, but Lou said, it's the easiest to read, so use that. Okay, so lessons that I learned. So number one is speakers are data slingers or storytellers. And the largest audience is won by the storytellers. And he was like telling me this because it's like, try to learn how to tell stories, you know? And I'm like, no, it's like, and like Matthew Hale's talk yesterday, he was saying about, you know, graduate students and data, you know, and that I never got out of that. But okay, so I'll tell you a story, the origins of near destruction of mankind, which is appropriate now. Kronos and the Titans, they created mankind, okay? And then Zeus and the Olympians came along, and Zeus thought humans were pitiful, rudimentary, and highly experimental, and wanted to wipe us off the face of the earth and start afresh with a more advanced form. Okay, but Prometheus gave us fire, and that gave us something that kept us on, and it gave us a little bit of a step up. And what people don't know about Prometheus is that his X-Men power is he knew everything that was going to happen, okay, for everybody. So when he was chained to that rock and having his liver eaten out, he had Io come by and he told her whole story. She said, "Would I be tortured forever?" And he says, "Like me?" And he was like, "No, you won't. It's going to end." And gave her detailed examples of how to go through. And so I kind of, I saw that, and my wife's a classics major, Callie, and she was like, "Yeah, well, that's a great story," you know. And Lou was kind of a Prometheus. He seemed to know you through and through after 5 minutes of talking to you, and he knew your strengths and weaknesses, you know. And that's the end of my storytelling. Okay, so let's see. Okay, so where was I before I met Lou and Buzzy? So I was trained in the biolabs of Harvard, and I went through like 3 Drosophila labs. So I was a developmental biologist in Drosophila, and then I went and, let's see, is there the pointer? Oh, yeah, okay. So this guy, before I came to MUSC, he was my mentor, and we notched it up from Drosophila to mouse. That's Mario Capecchi, and his mentor was Jim Watson, and there's Wally Gilbert, maximum Gilbert sequencing. And so we were interested in this Hox gene, which is conserved. It defines the cervical thoracic boundary all across many different lineages across vertebrates. And what my first graduate student found was that estrogen and progesterone can perturb the fundamental patterning genes. And we thought, well, wait, these are hardwired. They're conserved throughout metazoa.
So how could that be? Because, locked and loaded, pre-programmed. It's like, no, nothing is off the table as far as endocrine disruptors. Okay, so, and that's where I was. Hello? Oh, okay. And then, so there's Lou and Buzz. That's Buzzy and that's Lou. And that's a photo of Lou. Yeah. So actually, I didn't meet Lou first. So they came and interviewed at MUSC, and I met Buzzy. And I was just amazed at, the simple elegance, paper, a stack of paper and some crayons, and hand it to the kids in the, in the Industrial Valley. And you look at the writing of the kids in the foothills and in the valley, and her work prompted the Mexican government to change policies about the handling of these pesticides and herbicides used in the valley. It's like, what was the budget for that? $10? So great. But then Buzzy said, you need to meet my husband. If you are impressed with this work, you need to meet my husband. So then I met Lou, and that's the end of the story. Hello. Oh, whoops. So, we're on to lesson number 3. I thought we only had the first. Okay. So it's teamwork. So he had this magnetism to attract the smartest, the most capable folks. And it's like, you've got a team here. This team can really accomplish things. Just plant the idea, and if you convince them, then you got something going. So yeah. And you all know who you are. I won't go through each. Look at all the hair I had then. Yeah, so just an incredible group of people, talent on so many different levels, and all the levels you need to accomplish a task. Just amazing. The idea is to recapitulate that and emulate that team, all the time. Okay. Oh, and everybody's showing their son. So this is my son Anthony, helping us with the mud snail project. And another lesson is if it's not good for the critter in your backyard, it's not good for you. Okay. And so let's see. Okay. Yeah. So one thing Lou instilled in me was, what is a good sentinel species? So it's something that's long-lived. So these mud snails, Tridia obsoleta, they live in our backyards, and they live up to 40 years, and they stay in one location. Okay, and they undergo imposex in the presence of, for example, tributyl tin. So they were dredging Charleston Harbor and making it deeper for the super tankers. So we thought all that settled tributyl tin was going to be kicked up and we'd have imposex. So we had the plan. My son Anthony worked for that first graduate student down in Mazatlan, and they had Plecopurpura panza, the purple dye producing, and they're real easy to identify for imposex. So what I found out was, after a while, sounds like a great package here. Well, these guys are almost impossible to identify imposex in. So, boom, okay. Well, it's a work in progress, right? So, yeah. So that was the lesson that we learned there. But where is your backyard? Well, it's the whole planet Earth, because in these lower latitudes, we have pollutants that are put up in the air, and they come crashing down at the poles. So what we consider pristine environments, like you go to the ice shelf in red, white, and blue land, that's the new name for it, and you'll see these black specks of pollution that are kind of burrowing holes in the ice because the sun hits them and they heat up and they melt through. So you take a lot of those particles and that adds to the melting process. So this is a project, we have this with the Norse Polar Institute, Marmot Detox Project, whales and polar bears. So I just show some examples of biomagnification and bioconcentration. And what's very interesting is that the killer whales are at the greatest risk for demise because they're apex predators. So not only do they eat everything and everybody, but they go for the liver first, and the liver will have a concentration of pollutants and contaminants. So our main focus now is on the killer whales, and we're using the fin whales as a control for that. But also, this one guy in that Marmot group, he swims with the orcas, and when the fishermen collect in the net the herring not in the net will stay with the school in the net, and so the killer whales will come along and they will actually they'll pick the ones on the outside of the net and eat them. But the way they eat them is incredible. It's like a baseball player eating eating shelled sunflower seeds. They'll take the whole herring in, and in a second or two, they'll spit out the filleted vertebrae. So they'll just like, and it's just like, really, that takes a lot of control between your teeth and your tongue and to do that. But they love those snacks, Okay. So, and then, so we do these dart biopsies and collect the samples. And so the last time, so when I went, the first time it was like, you fly into Oslo, then you get onto this small turboprop, and you fly 1,000 miles north to Tromsø. Then you get in a boat and go north from there. So Tromsø is 350 miles north of the Arctic Circle. So I say, I don't like cold. Whoops. Let's see. Oh yeah, and here's the whole thing. And our part of it is the. Let's see. I think there was one more picture. Oh yeah. So we have this special sauce, but we also are doing the IPSC part of this project. So, but marine mammals are really, really difficult to induce into pluripotency. So we're trying all sorts of different things, and conversion of producing MSCs and producing conversion from fibroblasts to neurons, et cetera, et cetera. So it's a real challenge, but hopefully I live long enough so that we have some success in it. Yeah. But, Lou is here. He is in this project. He's right in here, and he's with me. And he's sort of like, I don't like the cold, And I when we went out on the boat, I said, what's the weather going to be like today? And they said, everything, So sunny and warm. A snow blizzard that tears right through your coat and your face. I came in off the deck and they said, here's some goggles, but you can't protect the rest of your face, which is going to be torn to shreds. So just stay inside during this. Yeah. So I heard Lou say, toughen out. Pull yourself together. Okay. So, and then, where have we got? So, this project was started, and we got this Gulf of Mexico oil spill grant,
And we wouldn't have gotten it if it weren't Lou's name on it, you know. So it was great to get. And we were charged with identifying endocrine disrupting chemicals in oil. And so what's shown here is Corexit dispersant, which is 30% DOS. And this is a mix of Corexit, and this is MC252 crude oil from the wellhead. And so it does a great job of dispersing the oil. It's an emulsifier, but Corexit will kill the oil-eating bacteria. So, and if I took this vial with the Corexit and I just opened it up here, within 5-10 minutes, we'd clear the room because people would be coughing because the surfactants in your lungs would not work and you would be filling, your alveoli would be filling. So they dumped 2 million gallons of this in the Gulf, and it's sort of like, maybe they should have done that little pilot study about the oil-eating bacteria, you know, that sort of thing. But yeah, you know, Lou said, you gotta grab the audience, because you say emulsifier. He says it's in the definition of homogenized milk. So you you buy a gallon of milk, it's good for 2 months in your fridge. Does it ever separate out? No, it doesn't. You know, so. But then one of the BP Gulf oil scientists said, "Well, you're telling us this is dangerous, but we give it to pregnant women." And I was like, what? You know, and it's funny because it's, you know, it's the method of choice. So constipation, because your estrogen and progesterone levels change during pregnancy, food doesn't move through the digestive tract as fast, and so you absorb more water. And so they take the Corexit, Doculax, and so and I said, well, that's a good idea for a project, you know, let's feed pregnant female mice. But you know, that's all published work. I'm going to jump ahead to, so we were doing all this stuff, and I came across this paper on this, so we have this PPAR gamma luciferase reporter mouse, okay? Well, PPAR receptor luciferase. So, so there's cycluc-1. So that is a luciferase substrate that passes through the blood-brain barrier. So we said, well, let's see what it's doing, and you know, and I guess the heading for this section is, the question was, why are people so effed up? You know, and I'm including myself. And the idea was, you know, there's some neuroendocrine dysfunction going on here. So what we did was we orally gavaged male and female mice, and we gave them the cycluc-1, and then we looked at them over time. And the females, their brains will max out after, so the female brains will max out after 20 minutes, and they're still chugging away after an hour. The males max out after 75 minutes, but are a lot weaker, okay? Well, age-related neurodegeneration, dementia, is more prevalent in women than in men. So that kind of was interesting. And so we did histology and immunohistology, and we found this, we found it lighting up within the within the dentate gyrus of the hippocampus. And if you look at the human atlas, brain atlas, and down below here at the bottom are different patients, donors of brain donors, and you could see the red is PPAR gamma and PPAR beta delta expression, and it's hippocampus, frontal regions, parietal lobe, everywhere where that's affected in dementia. So this has got us into that mode. Also, we did metabolomics, and just to focus on histamine, so DOS treatment really ramps that up. And that's one of the major upregulated metabolites. And if you go to the literature, histidine, look at the p-value on that in what's called the undernourished Alzheimer's brain. So there's some excitement in there. So we started in on behavioral testing, and so these are mice fed during, their mothers were fed during pregnancy and up to weaning, and then they weren't fed DOS again. And they have, they're slow learners, and they don't remember as well either. So fear conditioning is where you give them a tone, beep, and then you give them, you take a 9-volt battery and you touch it to their tongues, and they freeze. Well, not exactly that. It's to their foot pads, But it's like when you touch, it's a very slight electric shock. But they don't learn to freeze with a tone, So, and then this other one is Barnes maze. And what you do is you show them the goal box, and they have food in the goal box, or you have opposite sex bedding, and they're attracted to it. And you change the position of the goal box, and that there are these colored triangles and squares and whatnot, and you can reposition it, and you see that with dosage, and also these pictures are actually from a receptor mutant that we generated, and with gene dosage of the mutant, they kind of lose their way, and they don't know the concept of, okay, I'm on this table and I've got to find the goal box. In fact, a lot of times they'll get to a corner, an edge, and they'll back themselves off the edge and fall off the table. They're just sort of like, I don't want to do this. Okay, so, and then there are other, with the ALS side of things, we have this catwalk, and so you can measure all these different things. And so here is a control, and you can see the very regular movement of the control down here in this pattern. But then in those mutants, you see that they have irregular patterns. Okay, so, and then of course the DEXA scan, and you can look at different body mass measurements. So you have bone fat cartilage, and you can measure fat percentage, total weight, bone mineral density. So we've really kind of ramped things up, and it's all because of that start with Lou that we've gone through that. So, final lesson. Okay, so there's a couple lessons here. So field scientists they're tough stuff. They're survive. It's sort of like, what do we need to do to get the job done? And it's like, you don't hear a field scientist say, oh, I'm not going to do that. It's like, no, you're going to do whatever it takes to accomplish the job. And so we just have another challenge here, and it's like, okay, let's figure out what it's going to take to keep things going and get, progress with the project. So don't Lou was, he was the positive thinker. I was the one who was thinking of every which way it wouldn't work, and he was the one who was like, this is great, this is exciting, this is going to go forward, we're really going to sink our teeth into this, and it's going to just evolve through levels and levels and levels. And I know somewhere that he's seeing all this stuff that we're doing as a result of what we did together, and he's saying, be happy, be confident, you're doing something worthwhile, just keep plugging away, don't lose heart. So, and that's the end. Okay.
All right, thank you so much. Next up, we've got Matt Milnes from Georgia College and State University.
All right, thank you.
Good morning. I don't always wear shorts for my presentations, but I found the shortest pair of khakis I own. Doesn't totally do it justice, but here we are. So I'm going to talk a little bit about Lou's early work, and I mean some in here have known Lou longer than I have. But a lot of people know Lou as the endocrine disruption guy. They know him as the alligator biologist. And there's probably some folks in here who don't really know him as the lizard biologist. And to be fair, I can't say that I do, that I know him that way. When I met him in 1995, I joined his lab. It was actually one of his graduate students, Andy Rooney, who was a graduate teaching assistant in comparative anatomy. And he brought me into the lab while Lou was on sabbatical. He was mostly in Tulane that year, working with John McLaughlin. So I heard a lot about this Lou guy, but I really worked for Andy. But eventually I did get to meet Lou and went from undergrad in Lou's lab, working for Woody Woodward at the Florida Game and Fish Commission, and then back to Lou's lab for a PhD. So I had the privilege of working with Lou for about 10 years. So that's, yeah. So anyways, yeah, so I'm going to talk a little bit about his early career. Let's see. There we go. So I just thought I'd start off with, we'll call it the statistics, right? By the numbers. I'll give you a second to kind of take all this in. If you look at his lizard work by publication, it extends from, as far as I could tell, and I found one of his old CVs online still, and it seems like 1979 was his first published paper with lizards. And the last one I could find was 2007. So, which, one of the interesting things was, during the 10 years that I feel like I was working pretty closely with him, I had no idea that he was publishing so many papers on lizards during that time. Where did he find the time? So based on Google Scholar and his CV, I think he's got about 70 papers on lizards. Nothing to do with endocrine disruption and alligators. Don't anyone throw rotting fruit if I lump tuatara in there too, because we know, they're not really in the same lineage, but they kind of look like lizards, right? So we could probably add another 5 or 10 papers on tuatara to that list. And the one thing interesting about his work is there was this transition. I mean, when Lou started, he kind of met more of like the classical definition of a herpetologist. He was really into his lizards. And so if you just go to like JSTOR with the Herpetologica and JHerp and those type of journals, you'll find 24 what you would think of as like herpetology papers, which was kind of interesting. He directed a number of graduate works, theses and dissertations that the focus was on lizards. And just to kind of now take some modern perspectives, so some of you might be familiar with the series that Dave Norris and Kristen Lopez have edited, The Hormones and Reproduction of Vertebrates. So they recently updated that, and just going through some of the chapters that aren't about endocrine disruption and aren't about crocodilians, he's got about 24 papers, different papers that are cited throughout there. So even today, you can see kind of the impact of his work. And then on the right side is kind of like we'll call it his lizard CV. He was associate editor of JHerp. He was the president of the Society for the Study of Amphibians and Reptiles for a year. I don't know how many people knew that, but kind of interesting. So anyways, all right, so there's by the numbers. So when you look at this, like if this was just like the retirement dinner, you'd say like this person had an accomplished career, and when you break it down by the numbers, this is kind of only like maybe a fourth of his career. That's impressive. There's his first paper. That is not Scoloporus eurovi. Scoloporus eurovi is a viviparous species. That's Scoloporus ungulatus in my lab. You notice to the right there's a little nesting box. About half the time they don't use the nesting box. I don't think Lou had an iPhone in 1979 to record that, but anyways, I thought you'd get a kick out of it. All right, in the conversations I had with Lou in the 90s, you know, talking about like what he did before he got into endocrine disruption in alligators, a lot of times, like if he was just giving a short answer, he would say, I studied the evolution of viviparity. That would often be kind of like his go-to describing his early career. And so I thought, well, let me just throw up some papers that kind of, you know, the titles are representative of some of that early work. And then below the green line, I wanted to see, you know, I looked at, well, how impactful is this work? You know, not just how many citations, but what about today? And what was really interesting is that, you know, we've got some relatively contemporary papers using the latest and greatest in phylogenomics, right? Like thorough sequencing and that sort of thing. The hypotheses that he proposed, sometimes in collaboration with Richard Schein, like they hold up, right? And they're still impactful. So, you know, he did study the evolution of viviparity, but he also was a mechanistic physiologist. And I think some of these papers just sort of represent some of the core work that he did over, you know, the, say, postgraduate studies, and then some of his early time at UF. And if you look at the names of some of the folks, and if you recognize them, or you know where the species are from, you know, he was already an international scientist before he got into endocrine disruption. And if you said, how do you train a biologist to be an expert on alligators and an expert on endocrine disruption, without studying alligators or endocrine disruption? You tell him to have a career like this. You know, he understood the biology of reptiles. He understood reproductive physiology. He understood endocrinology really well. And I think that's what set him up for some of his later successes. The paper in the upper left, right? Well, you can see, you know, almost cited 1,000 times. So even if we count for the times he cited his own work, that still leaves about 750 citations, right? So pretty good. And as we've discussed, in order to understand what's disrupted, what's abnormal, you have to know what normal is, right? And so while he was doing this endocrine disruption work, he was still pursuing the basic biology of the organisms that he studied. And so I think, you know, we've all kind of appreciated that. And so again, you know, below the green line, we've got one of the most recent papers from Ben Parrott's lab. And it's amazing work. It's really complicated, but it asks an important question. And it does an amazing job of addressing it. On the right side, some chapters I was involved in writing, you know, where in those chapters, I'm leaning heavily on both the basic biology as well as his studies of environmental influences on reptiles. So I just kind of thought, well, how do I convey his impact to the scientific community? And so that's kind of part of the way I went about it. You know, like everybody, Lou's personally had a big impact on my life. As I mentioned, you know, I worked with Lou in collaboration with many others for about 10 years. And then I went to the San Diego Zoo and had the opportunity to do some work with some interesting animals. Still mostly in the realm of endocrine disruption. We're interested in phytoestrogens and how they affect reproductive success in rhinos. There are populations of California condors now that regularly feed on marine mammals, which you can imagine potentially that's a problem. You're not going to be able to get a captive colony of any size of rhinos or condors and do experiments on them, but through things I learned in Lou's lab and then postdoc opportunity that was largely facilitated by Lou and Tyson Iguchi, I learned how to do some molecular endocrinology so we could clone receptors from these highly endangered species and we could screen them for interactions with various EDCs. And yeah, so I'm I'm proud of some of the work that we did, and Chris Tubbs, who we're going to get a little video message from him, he's kind of carrying on at the San Diego Zoo with some of that work. And the funny thing is that I'm not blaming Lou for my 7 years out of research. I had this crazy idea that I was just kind of disappear to this little college in the mountains and become a teacher, and Lou saw that at that moment in time that's what I wanted to do, and he supported me. And he wrote letters of recommendation, and I got the job that I wanted. And that was fine. It was the right thing in my life, but I missed the research. I missed this. And so I decided I wanted to get back into it, and I found Georgia College. It's kind of, I call it my Goldilocks job. I'm still doing a lot of teaching. I teach 3 courses a semester, but I've got support to do research. I've got some excellent students. But I got there in 2018, 7 years out of research. I was like, okay, 7 years is a long time to be out of research and try to pick up. So I had to figure out what to do. So Matt Hale. So I got there in 2018, and I've been thinking a lot about Sclerophorus, in part because of Lou's influence. And he did some really cool stuff looking at elevational gradients and the evolution of viviparity. And in the eastern U.S., we've got this species that goes from Florida up to like New Jersey. And I was like, man, there's a huge latitudinal gradient there. Even in Georgia, we've got coastal populations and mountain populations. So I kind of had this idea, but I hadn't committed to it yet. And about a year after I got to Georgia College, Matt Hale sends me an email, and he's like, hey, we've been doing this study, done some testosterone implants on some lizards, and we were mostly interested in the livers. And I've got these repro tracks, and there's something interesting going on here. Looks like there's some hypertrophy in the accessory organs. Are you interested? I said, well, yeah, what a coincidence, right? And so about 2 weeks later, he drops by the lab, drops off a jar of lizards, and, well, little parts of lizards. And I was like, okay, I'm gonna take a look at these. So I start looking at them. It wasn't necessarily this linear, but you can see here's a male and a female control, right? So these are immature animals. These are like a couple months old. And we've got like the kidneys, the testes, the mesonephros, which is the precursor to the male reproductive tract, and it's the embryonic functional kidney in amniotes. And so I'm looking at males and females, kind of orienting myself, because I'm not really a lizard biologist. And then I look at the ones that got the testosterone implants. I'm like, whoa. The way it projects doesn't do it, but like the hemipenis is almost like poking you in the eye. It's like really, it's like, and the mesonephric kidney now looks like an epididymis, and the mesonephric duct looks like a ductus deferens, and I'm like, okay, but these are males. They were immature, but they got testosterone. It's like, okay. And then I looked at the females, and that was kind of one of the things that definitely. So I'm trying to put it together. I'm like, there's too many reproductive tracts. Like, what's happening here? And so the oviducts in these females, like, as a result of the testosterone treatment. And so that was kind of unexpected. And let me see. If you look at the kidney, so that's the metanephric kidney, the definitive kidney. You can see some passageways that are filled with like a white opaque substance. They have this accessory, I'll say, structure called the sexual segment of the kidney that is probably not homologous, but maybe analogous to seminal vesicles. It produces these seminal secretions under the influence of androgens. But apparently, you can wake that up in females too. So I'm gonna go through this kind of fast, but just at the histological level, so MD, that's Müllerian duct. So think of this as like a vas deferens that's not yet developed. Here's testosterone treatment, right? So basically, you're getting like precocial maturity. Here's cross-section of the kidney in a control. I've got some collecting ducts labeled. So the sexual segment of the kidney is basically a modified collecting duct. And so over here you can see what the androgens just like, they got big, they're full of all these secretory vesicles. Here's the Müllerian duct down at the kind of posterior end. Females, the oviduct is just this completely like regionally undifferentiated, very thin tube, and in testosterone treatments, you've actually got glands even. Like, so it didn't just even get bigger, but there's some regional differentiation. We've got glands. Yeah, it's really funny, like a cross-section of the female, because you've got, you know, essentially ductus deferens or vas deferens as well as an oviduct, right? And so all kinds of interesting things there. We said, well, you know, we assume this is happening through the androgen receptor, but we wanted to be sure, so we did some immunohistochemistry. AR is everywhere you'd expect it to be in the males. AR, I think the most interesting thing here is that AR was present, although kind of weak in the oviducts, but when you treated with testosterone, you got a lot of expression of androgen receptor. And so the theme here seems to be there's a lot of autoregulation there where testosterone upregulates the expression of its own receptor. And so instead of conclusions, I have a take-home message as far as the research goes. And so one of the interesting things was there's this asynchrony in the developmental pattern here. So the gonads differentiate first, and then the hemiphallus seems to commit, right? So if it's male, without the treatment, it wouldn't have necessarily grown, but it retained, or it has that sensitivity where under high androgens, it's going to grow. But in the females, that was lost prior to the treatment. So there was no hypertrophy of the hemiphallus in the females. Also, the Müllerian duct, the precursor to the oviduct, that had already regressed in the males. So we didn't see any hypertrophy of the Müllerian duct in males because presumably it had already regressed to the point that it was no longer sensitive. But what was really interesting, for those of you who maybe remember like a comparative anatomy class, you probably learned about this in the context of mammals, that this precursor to the male reproductive tract in females will regress so that it can't develop, and this happens like in utero or before birth. These were like 3-month-old lizards. And so what's really interesting is that in the females, the precursor to the male reproductive tract, not only was it present, but it still had androgen responsiveness and basically became, for all intents and purposes, an epididymis and a ductus deferens, 3-4 months after hatching. Which, so the chronology of these events is pretty interesting, and maybe haven't fully appreciated how complex it is. But like when I think about my interpretation of this all goes back to the training I got from Lou. And thanks, Matt, for bringing those lizards to my office. Anyway, so we have some really interesting questions to ponder now with that hypertrophy of the oviducts. Was that just driven through androgen signaling AR, or was that testosterone being converted to estrogens through local aromatase activity? So we're trying to separate out the AR-dependent effects from possible localized aromatase and ER-dependent effects. We had kind of a swing and a miss where we tried to use an androgen receptor antagonist, an estrogen receptor antagonist, but we weren't able to totally knock it down, and estradiol in young lizards is apparently very toxic. If anyone has experience with that and can help us understand what's happening, please talk to me later today. And then the last thing I'm interested in is when does the mesonephros lose its ability to respond to androgens in lizards or non-mammalian vertebrates? So some interesting stuff. So now that I'm kind of established, or getting there, establishing my lizard lab, we're working on, we've got Lizzie and Connor working on various aspects of, we just want to do some foundational information. Again, understand the biology of the beast, might be what Lou would say. And that will set us up for future success in asking some interesting questions. So they're working on male maturation and seasonality. I've got some students working on, female. I've got some students working on male aspects. And then I thought I would end with, so there's Andy. He couldn't be here. He was supposed to be here today, but unfortunately couldn't. This is a lot of the cohort from my time in Lou's lab. It's not everybody, but it's a good chunk of the people, and I appreciate that. We got Satomi on the right, which again, he couldn't be here, but I wish he was. Brandon is, I mean, we're 10, 15 years on, and he like took my camera when I wasn't looking and created that selfie, and I try to haunt him with that any chance I get. And then we've got me and Woody. At the time, I think that may have been the biggest female that that we had ever caught, and that was a lot of fun. So, anyways, thank you all.
All right, next up we've got Chris Tubbs from the San Diego Zoo.
All right, I'm trying to share my screen here. So first of all, I'd like to thank Jerry and Thea for inviting me to present this talk. I'm really sorry I couldn't be there in person. The sun is just rising out here in California, but I couldn't think of a better way to spend a Saturday, and I really enjoyed hearing all the presentations so far. So the title of my talk is A Career in Imagining How Introductory Biology Changed My Life. I think a big theme of this talk is my naivete in all things science, going into college, and how Lou really shaped my career and led me down a path I never could have thought that I would have gone to. I wasn't even going to actually put together a PowerPoint, much less show data, until John Wise in his talk yesterday said Lou wouldn't be very happy if we didn't show any data. So I'll show a little bit of data, but most of this is just really about how Lou his impact on my life and how I got to be where I am. So I grew up in South Florida. I went to the University of Florida. The reason I went to the University of Florida was because I knew I wanted to work with animals, and the only way I knew how to do that was to be a veterinarian, or as Lou would affectionately say, a dog plumber. UF had the only vet school in Florida at the time, so I went there. I signed up as a microbiology major, which lasted, it's not even actually documented on my transcript, so I switched out of that pretty quick. And ended up becoming an animal science major, which put me on this course trajectory that I wasn't really happy with at the time, mainly because my first semester I was supposed to take introductory chemistry and not biology, and I really wanted to take biology. So anyways, I took the chemistry class. I spent way too much time playing pickup basketball on the courts outside Jennings Hall, and quickly earned a C, the first C of my career in high school or otherwise. And as they say, C's get degrees, but they don't necessarily get you into vet school. And so I realized I had a tough hill to climb at that point. But continued with my coursework, and the first semester of my sophomore year was when I was supposed to take introductory biology, and I did. And I remember walking into the lecture hall. It was a pretty large lecture hall, and seeing this guy sitting in the back row of the middle section of the lecture hall with lots of white hair and a big white beard, thinking, "There's no way this guy's in the class. What's going on here?" And it turns out he was the professor. He was just watching us file into class, and I firmly took my favorite position of, in the back of the room. And then he got up to start lecturing, and I think we can all appreciate when you take a class with Lou, just how captivating he was. I mean, he was telling stories about jumping on the backs of sea turtles, I believe in Indonesia. Of course, he would teach us a ton of biology. We didn't really know he was teaching us a ton of biology, but through these stories and everything that he had to offer us, it was a transformative experience to be one of his students. And thinking back on it, Lou didn't teach introductory biology every year. He taught it every other year. And if I had taken biology that first semester, who knows what would have happened. So that's kind of the first way that he kind of influenced me and basically set my life in a direction I hadn't thought of. So he talked a lot about the work that he did and undergraduate researchers, and of course I sat there and listened to these stories about driving airboats through swamps and catching alligators with your hands, and I was like, I want to do that. And so I asked him if I could be an undergraduate researcher in his lab, and he said no. he didn't have space. And so I asked him again later in the semester, and he said, no, I still don't have space. He offered a class the next semester, which was a biology reproduction class. It was comparative vertebrate reproduction. It was an upper level class, and somehow I managed to get into it as a sophomore and made sure to plant myself in the front row of the lecture hall for that class, and just do everything that I could to do the best I could in that class, and continued to pester him to work in his lab. And eventually he invited me to his office for a meeting and said he had a spot. He would bring me in and explain that there would be like a probationary period where you basically had to show that you weren't going to make mistakes, this kind of thing. And this was concerning to me because I had immediately prior to that meeting just ended up paying like an $80 bill to the chemistry department for all the glassware I had broken during chemistry. I thought, oh no, I really have to buckle down and make sure I make this work. And I did, and sort of, again, it was just one of those opportunities that was life-changing. So I was able to work with Lou and his grad students. I was there with that cohort that Matt just showed, and got to do a ton of work with alligators. I also got to do a ton of work with fish with Ed Orlando, who was in the lab at the time, because fish were really what I was interested in. And it was just, it was an amazing experience that just opened my eyes to research. So I didn't realize research was a career. It was something, in hindsight, I should have. I was kind of one of those Jacques Cousteau type kids who watched all the nature programs and that kind of thing, but didn't realize you could actually have this as a job. And to hear Lou's stories about the work that he did in the places that he traveled, and just the freedom to ask questions and figure out how to answer those questions just was incredibly inspiring. And I quickly decided that school wasn't for me. Not that I could have gotten in with my grades, because I still managed to climb quite a few C's through college. Just had a great experience working in his lab for a couple of years, and then decided to take a year off from undergrad in between grad school or whatever the next step was. And then, of course, was looking for work, and Lou put in a good word for me with Woody Woodward, who we heard from yesterday. And I got to be a part of this lineage, which was an incredibly fun part of my life. I got to work with Matt, Dee, and Woody, doing alligator work for a year. It really was the best job I've ever had. It was so much fun. But then, I needed to figure out what the next step was. And I had seen students come through Lou's lab as undergrads that ended up as grad students. Matt just told you that was his experience. And I summoned up the courage to ask Lou if I could be one of his graduate students, if he had space for me. To which he responded, no. He didn't have funding for another graduate student. He had already accepted one, and at that time the lab was on tougher financial times. And so he said, I can't take you. I wish I could, but let's find a spot for you to go. And he asked me, I think probably one of the quintessential Lou questions is, what is your dream job? And so we talked a lot about that. And I told him I was interested in endocrine disruption, and I was interested in fish, and he gave me some names of some folks to work with, and actually paid for me to go with him to the E-Hormone conference to meet Peter Thomas at the University of Texas, who was a professor who worked on fish and endocrine disruption, and essentially took me out there for a meet and greet. And we went out there, and I met with Pete, and I ended up working with him. And it was a really, really cool experience in that it taught me a lot about how hormones and hormone receptors work, and how to biochemically assess that and to measure hormone receptor interactions. And I worked on fish, and I studied how fish sperm were influenced by progestins, and how that affected sperm motility. So pretty specific area of research. And as I was wrapping up my PhD, I was sitting in my office. I think I had a little less than a year to go, and the phone rang. And there was in San Diego a nature Nature Conservancy that had maintained a subspecies of steelhead local to one of our rivers. And one of the last males in their collection was about ready to pass away, and they were going to euthanize it. And the San Diego Zoo was interested in collecting sperm from that fish and preserving it in their frozen zoo, which is a large biobank that we maintain with over 1,300 species now. And somebody at the zoo was asking if anybody knew anything about fish sperm, and at the other end of that call was Matt. And Matt's like, I know a guy who works on fish sperm. And Matt called me and asked me, what do you know about sperm cryopreservation? I knew absolutely nothing. I was zero help to him. I think I said, maybe put some glycerol in it. Because I had not worked on cryopreservation of sperm. I worked on breaking sperm apart to try to figure out biochemically how they worked. And at the end of the call, which I think was probably the worst, the least helpful call maybe ever for him, he offered me a postdoc. And it was not a good interview in terms of knowledge for what he was looking for. But it was one of those things that at the time I didn't take seriously, because again, being naive, I didn't realize that research at a zoo was a thing. Like I didn't know that was a job. But obviously Matt was working there, and it was a couple of weeks after he called and offered me the postdoc. And at the time I had another offer for a postdoc at a big biomedical lab in Stanford that I was going to take, but I realized the opportunity to work at San Diego Zoo is pretty incredible. And I thought back to some of the things that I had discussed with Lou when we were discussing graduate programs. I had thought to apply for a program that was straight PhD, no master's, and I was a little nervous about that. And Lou was like, hey, if you don't like it, just get your master's and then figure it out and go somewhere else. Like you can always just get there, and if it doesn't work for you, do something different. And I thought the same thing about San Diego Zoo. I didn't realize what the opportunity was at the time, but did know that if it didn't work out, I could always go somewhere else. Well, it ended up working out. I had a great time working with Matt. As he mentioned, we worked on a bunch of different species, and ended up there, and just this really unique opportunity to work comparatively across all sorts of different species and do all sorts of fun different things, and to do so in an environment that's very different from a lot of the research environments that are out there. So we heard some talks yesterday where folks are just doing this super elegant work, with knockouts and tissue sampling and that kind of thing. When you work on rhinos and condors, that's not necessarily something you can do. As Matt alluded to, you're not going to have a research colony of rhinos or condors to do those types of studies. So we had to get creative in what we did. And I joined Matt shortly after he arrived there, and we built really the first molecular endocrinology research program in a zoo setting. And to this day, that's still the case. We're the only people that do what we do in this particular arena, all with the focus on conserving endangered species. So here's the data I promised. So as Matt mentioned, he did a postdoc with looking at endocrine disruption and doing in vitro receptor-based assays. Of course, I had that experience not in the endocrine disruption field, but doing mechanistic hormone receptor work. And we started by looking at addressing a problem that had been going on for decades, where southern white rhinos born in captivity failed to reproduce. And we keyed in on diet and phytoestrogens. And I remember in that biology and reproduction class that Lou taught him discussing phytoestrogens in sheep, which was a huge problem back in the 1940s. And so it just was kind of cool to come back that many years later and basically try to tackle a question centered around something our mentor had taught us, to do so with some new techniques in a completely different arena. So we did a lot of cloning of receptors. I think Tyson said that they cloned a receptor in a week, which at one point we actually did that. Took us a while to get going, but we got pretty good at cloning receptors. We started by looking at southern white rhinos and comparing them to greater longhorn rhinos. which are a species that lives in the same environment, eats the same diet for the most part, but breeds relatively well, actually quite well compared to southern white rhinos. And what we were able to find, if you look at these two graphs, is we treated these hormone receptors with different concentrations of phytoestrogens. And in this top graph, this is a phytoestrogen genistein. We saw no significant differences between the two receptors, but in most cases, what we were able to see is what is represented in the lower graph, where southern white rhinos have higher activation or higher maximal activation of estrogen receptors when treated with phytoestrogens and greater one horns. So for endocrine disruption guys and nerds like us, I mean, this was incredibly exciting. Like we have this species level effect that we can then associate with this receptor level effect. That did not necessarily convince zoo veterinarians or nutritionists to make a change. We got this work published. It was really cool to have it published. We got it published in Endocrinology. We had a super nice commentary piece written by Heather Pattisall, who I believe is in the audience. But still, it wasn't able to convince folks to make a diet change, even though it made total sense to us. I had to go at that point into full Lou mode in terms of communicating effectively with people. And I spent probably, at this point, 6 to 8 years as we gathered more and more data, just trying to explain this to folks as simply and clearly as possible to try to make a change to help this species out. Of course, we're not feeding rhinos purified phytoestrogens, but what we did end up doing was looking at diets from 9 different institutions across North America, basically extracting those diets, treated cells, expressing rhinoestrogen receptors in those diets, and then relating that to fertility of rhinos that were fed that diet during gestation. And it was this data here, after those 6 to 8 years of heavy politicking, where we were able to show that diet estrogenicity has a significant negative relationship to fertility, that convinced folks to actually make a diet change. So we were able to convince our zoo to formulate a new feed that was free of soy and alfalfa-based ingredients, and make that change, and then actually see what happens. And it was around this time when this diet change happened that I had my last conversation with Lou. He called me, him and Ben and Parrot were on the call. I don't know why they called me. I can't remember that. But I remember, well, I assume it was probably to get some tissue from some rare critter, because when the Gillette Lab people call me, that's usually what they're looking for. It's, hey, you work at a zoo that has access to all these species, can I get a sample of this? And of course the conversation went well beyond that, and I was telling him about sort of our work and where we had been and what we were doing. And I just remember him saying to me, he's like, he occasionally called me Chrissy. Chrissy, you're doing great things. He's like, and you're gonna make a difference. Like, this is really, really cool. And we had talked about me coming to visit him in South Carolina at the time to give a talk, and him coming out to visit and see the rhinos, and unfortunately that never happened. But what did happen after we made this diet change, and I told him that we had done it, and we were really hopeful. We had this calf born, and this calf was born to a female who had bred for years unsuccessfully, and then after the diet change, was able to conceive and produce this calf. And we've since had 4 calves born to 2 females that, same situation, bred for years, were never able to successfully reproduce and did the same. We've shared this information, obviously, and we know of 6 other institutions that have reduced soy and alfalfa in their diets and successfully had calves being born. And so, again, it's a different situation when you work in a zoo. We have the luxury of being able to translate our work super quickly relative to other fields like the biomedical fields and things like that. We just have to convince the right people, and it takes that communication that Lou has taught us all, and the good science that Lou has taught us all to be able to do that. And when we started this project, Matt and I, the southern white rhino population in captivity wasn't self-sustaining, and that's different now, and that's in part because we've had females that have been able to reproduce post-diet change that weren't previously represented in the population that now are. So that's kind of a cool thing, and something that Lou and I talked about in that last conversation about being able to use science to make a real difference. You know, the other thing that I really take from Lou is his appreciation. Matt hit on this. He always framed things in the vein of evolution and the comparative nature of vertebrate reproduction. And while species are nuanced, it's incredibly fun to be able to translate what you do across a bunch of different species. So this is just one representative slide of the species that I've been able to work on and appreciate and kind of like, you know, have an interest in and engage with in large part because of what Lou taught us. And I'm incredibly grateful for everything he's given us. As I said, I wasn't going to present data or actually put together a PowerPoint, so I don't have an acknowledgement slide. I do want to thank Matt for taking a chance on me as a postdoc. He taught me a lot. He like Lou, was a great mentor. And I appreciate that when he did go off to teach in the mountains, he handed the keys over to me, and we've been able to continue the work that we do. And then, of course, the take-home message here is I wouldn't be where I am without him, and I appreciate everything he's done. Thank you.
And next up, we've got John Bowden from far-off land of University of Florida.
Yeah, thank you, everybody, for inviting me to present today. It's an honor to talk about the impact that Lou had on me in my professional career, not as a researcher, but also as a person. So when I got into thinking about research, I knew nothing about research. And so my presentation today is going to be talking about what Lou did to help me become who I am today. And I really struggled with the title for this talk. Probably had 30 or 40 different types of titles, and usually when I'm stuck, the first thing that happens is, in my head, I said, what would Lou do? And usually I come to a solution right after I think about that. And so that's what ended up being the title of my talk today. So if you want to just briefly talk about where I started in Wisconsin as a little boy, I had a healthy fascination with alligators. So it really wasn't a surprise that when I decided to go to college, I chose the University of Florida. Bless you. When I got to the University of Florida, I wanted to be a biology major, but back then, they didn't have a biology major. They do now, but they didn't have a biology major, so bless you again. Good, okay. I had a counselor at the time when I first got to UF, and they said, well, why don't you become a chemistry major? Chemistry is involved in life, and you can pivot out of that. And so I became a chemistry major. And I really liked chemistry. I was really good at it, but ultimately my passion was the environment. It was doing something biological. And so at some point when I started to get towards the end of my undergraduate career, I started thinking about career paths, and I wanted to reach out to people and have discussions about what I could do. And I met Lou then. And I remember meeting Lou, and I was very intimidated. You read, he's a very big person in his field. He's got lots of papers, and he wrestles alligators, right? He's a very intense person, but when I met him, he was so gracious with his time. And we had great conversations about life and career goals. And I also remember at one point, he took me up into the top of, I believe, Bartram Hall, and I got to see the alligators on the roof. And it was just a really cool thing. Hey, we can do research on stuff like this. This is a really exciting idea. So when I decided to do my PhD at UF in chemistry with Rick Yost, I made it a point that I wanted Lou as a co-mentor. I really wanted to do something where we could use innovative chemical techniques to apply to environmental and biological questions. And the first time I met with Lou and Rick, I just remember that it was just an amazing meeting, and I left with pages and pages of ideas and notes. And I feel like that's always what happened when I talked to Lou, is that there was just a ton of ideas. And that was really exciting. And Lou was always teaching me. He told me, I don't want you to be a chemist who just has samples show up at your desk, right? I want you to actually go in the field and actually see how hard it is to get one data point. And that's something that really rang true to me, that I now pass on to my students, and that it's really difficult to do a lot of the work we do. And And I also remember on all these trips, my favorite part was actually the ride to there, where I had maybe a couple hours with just me and Lou, where we could just talk about just life and philosophy and other things, and those are some really great memories. When I was actually defending my PhD, Lou came up to me afterwards and said, "Great job, and you know, obviously you know the chemistry very well, but if you want to get in the space that I think you do, then you need to learn more about biology, especially if you want to be dangerous, right?" And so after I left my PhD, I went to a lab that was completely orthogonal to my current background. I went to a biochemistry and molecular biology lab in St. Louis University. And I have to say it was probably the two hardest years of my life, but also probably the most important 2 years of my life, because a lot of the things I learned there, I now take with me now in my current position. At the time when I was actually doing this postdoc, I was still in contact with Lou over a paper where we were measuring the levels of metals in the water outside the launch pad at the Kennedy Space Center, pre and post-launch. And during this time, I found out in the end of 2010 that he was leaving the University of Florida and going to South Carolina, and my mind was like in shock. And not too long after that, I get an email from Lou that basically said, "Hey, I have these new people I'm working with at NIST, at the Hollings Marine Lab, and we're looking for somebody who's interested in doing measurements on human and wildlife samples." And I wish I had a moment where I could see what my face looked like when I read that email, because it was just mind blown, right? I was like, this is exactly what I want to do. And I spent a lot of time preparing for this talk, going back to a lot of old emails, and one that I really just made me smile a lot when I looked at it was when I told Lou that I was coming and he was excited that I was going to be there. He sent me this email that says, "We have enough to keep you busy 80 hours a week. No kidding, but I promise I won't kill you." And it was just that great attitude that he just had invested in all the people he was working with, that he just really was excited to work with us. And that was something for me that was it really meant a lot to me. So when I worked at NIST, when I was at the Hollings Marine Lab and we worked with Lou, I mean, obviously I don't have enough time to go into all the things that we did, but I just remember this time being a very positive one for me, because it transitioned me from being a student of Lou to a collaborator, a colleague, somebody that, you know, he would ask me questions for advice and I would actually provide some ideas, and it was just a really great kind of period for me to develop as a scientist. And also I feel like I became Lou's friend, and I know this because at one point when he found out that I was the third, he started calling me JB3, so I would be getting messages that JB3 or hey JB3, so I felt like that was just a really great time. And I have very fond memories of having meetings with the White House and also continuing to go in the field with the lab. One of the really great experiences that I had with Lou was being able to go to South Africa. So there was an environmental disease called pancreatitis that was impacting crocodiles and other aquatic wildlife. And we were putting a team together to see if we can actually analyze and figure out what's happening. And this is probably one of my favorite photos here on the left, because it's the very exact moment that we were both looking at the disease for the first time. And it captures a moment that I always kind of look back to, and it was just a really great memory of like the beginnings of a really great project. Also, I have these two photos down here, and I like these photos because for some reason I'm very serious in this, and Lou is not. And we're in South Africa, we're in the bush, we're knee deep in fish carcasses. This is probably like third day of doing this from sun up and sun down. And here I am, I don't know, giving some kind of like stoic photo, and Lou is giving me bunny ears. And it was just, it's a great reminder of the levity and just his atmosphere around you, that it kept things really fun. And I also really appreciated that Lou let us be creative and think outside the box. One, I just had a crazy idea. I say, hey Lou, what happens if we take these point-of-care analyzers that do blood chemistry measurements? What if we take them in the field, and what if we can take a drop of blood and assess whether or not an animal has the disease or not without actually having to kill it? And he was like, it's great, let's do it. And he supported it and ended up working, and we published it. And it was just a great example for somebody early in their career that it's okay to think outside the box, and having somebody supporting that was just a great influence on my development. So one of the things that, looking back, and I think a lot about Lou, in my mind, the way I think about Lou is he was this train that was going 100 miles an hour. And I was just trying to hang on. I was just trying to be part of the whole process. And when that train stopped, I actually found myself in a place where I didn't know what I wanted to do. I didn't really know what kind of researcher I was going to be. I didn't know what I wanted to, I was just lost. And it wasn't until we had the memorial services for Lou that I saw how much impact he had on so many people, and the types of things he did for people to help them grow as scientists, and just the overall help that he gave everybody, and how much they appreciated everything they did for him. And it was at that moment that I realized that's what I wanted for myself. That's what I wanted for my career. I wanted to have a legacy like Lou, where I had people that I leave behind that really appreciated everything I did for them. And so when the opportunity came up, and there was a position here back at University of Florida, so I'm back here again, I took it. And I wanted to start my own research group where I could have the same impact on students and people that Lou had on me. And kind of like a parent who, a new parent who now realizes the things that their parents did for them. I look at a lot of things that Lou did for me, and I use a lot of that to drive how I interact with my students. And he continually teaches me as I think about things that he did, and I'm trying to be a better mentor. Some of the things that I really take to heart about Lou is, and you're going to hear a lot of these things today, just surrounding yourself with great people. And that's obviously, the students are the lifeblood of the lab, and if you really put a lot of time and attention to them, you can really do some cool research. I also try to inspire people like Lou does and educate, always educating. And probably the word I like the most is investing, because I feel like investing is giving a piece of you to your students, right? They see something that you're giving into them, that they can see that you're bought into them as a person. And so Lou did that to me. I was a chemist. He could have just flicked me away and said, I don't need to deal with this little chemistry guy, right? But he said, hey, I see potential. I see a way that we could use his enthusiasm and we could do some really cool research. And I want to do that with my students as well. And also, credit is infinitely divisible. So we, keep that tradition carrying on, that we always give everybody credit. Another thing that I feel like stems from Lou is that, again, he gave me a chance to work with him, and he saw potential in somebody who was outside of his realm of expertise. And so I try to do the same thing in my lab. So I try to make my lab available to everybody. So whether or not, whatever your major, your background is, whatever your knowledge level is, if you're on campus or off campus, we make our mass spec, our workflows available to everybody. And so our work, and I'm not going to get into a lot of my research today, but our work is focused on using chemical methods to assess emerging contaminants in the environment, and we try to study their life cycle. So once they're released, where do they go, right? And we have students from all sorts of majors and all sorts of levels, undergrads to postdocs, that come in our lab. We teach them how to do, and they actually do the analysis themselves. And I feel like that's something that Lou would have supported if he were still around. So just a few things that we do. Again, I would love to talk a little bit more about my research, but one of the things I really want to push forward is being a steward of the environment. Obviously, more now than ever, we need to be stewards of the environment. And so a lot of the work we're doing right now is on PFAS. I'm sure everybody is familiar with PFAS in here and why they're bad. And we do a lot of work measuring PFAS in the environment. So this is a study we did where we looked at over 2,000 water sample sites in Florida. And we actually have an interactive map online that you can go into, and you can see a dot wherever you want, and you can see the concentrations at those sites. We also do a lot of work with environmental samples. We were doing body burden studies. When Lou passed away, I actually have all the samples. So I saved all the work he had done and all the samples. And so we have samples from the 1990s that we are currently having students analyze, so we can do retrospective analyses of a lot of the contaminants that we're interested in today. And so a lot of the work he did back then is still relevant to a lot of stuff we're doing now. And we're also doing a lot of work that I think you'd be really interested in, like looking at maternal transfer of contaminants and unique environmental sentinels. I'm also still working in South Africa. So Lou lit an international research bug in me, and I've since been back 5 more times since the trip with Lou. And so we are in the middle of a lot of projects focused on water quality, both drinking and surface water. We have a big road dust study where we're looking at metals and contaminants in road dust and its impact on air quality and health. And I'm also continuing the tradition of bringing students with me as well so they get that idea of how hard it is to do research on an international setting. And hopefully those experiences will help them get better as a scientist too. So my last slide is, I have to say that I probably would rearrange and change slides probably 100 times for this talk, and I just kept feeling like when I would go through my presentation, I wasn't fully encapsulating the impact that Lou had on me professionally, both as a scientist and as a person. Hopefully what I showed today exemplifies that, but just know that it means much more than what I was able to show today. So thank you for your time.
All right, great stuff again. Last up here we have Patrick Delaney.
All right. Well, I'm Patrick Delaney. I'm a wildlife manager for a company called Deseret Cattle and Timber, and it's basically a big ranch. And but I used to work with FWC in the Gator program, but I just wanted to talk about Lou and his influence on me, and the influence on a lot of folks in this room and basically around the world. But when I got the email from Jerry, I just thought I was going to get up and tell a story, and then he asked me to do a presentation. So I started putting something together, and it really was kind of like what you were saying. I went through so many different times looking at pictures and trying to decide what to say, and the influence, it is hard to encapsulate like the influence Lou had on my career and life and just as a person. But one of the things I thought was interesting is when I first met Lou, you didn't know how to take, there's this guy, he looks like a wizard. I mean, he looked like Dumbledore, basically. He has this huge beard, larger-than-life character, and I was this new person working with FWC, but I'd always dreamed about working with alligators and got there, and I got the dream technician job, and then he starts talking about all this high-end science stuff, and it's almost like a different language. I don't understand any of it, and I still I appreciate everything you guys do, but it's and so I started working with him, and just fortunate to work with him, and Arnold and Woody brought me around, and it was great, and the fieldwork side is what I just excelled at. I love being out in the field, and some of the folks I've been catching alligators and doing fieldwork with a lot of you in this room, and that was something I was really good at. And then I just remember Lou, we were working up samples, and he's like, "You ever thought about getting a master's degree?" And I'm like, "I don't know." It seemed like such a hard thing, and I joke that I have like a math and statistics disability, but it really, that was one thing, and Arnold can attest for that, where he drug me along my master's degree, where it was a very difficult thing for me. But the fact is, I did finish eventually. But it was one thing that I remember barely getting into graduate school, and it was Lou's recommendation letter that was the deciding factor. I remember sitting down in the interview and taking the GRE a couple times, and then finally got in there, and then what was interesting is he goes, "Well, you do have a recommendation letter from Lou, and it was a really good one, so we'll let you in." And which is I don't know, it's hard to describe what that meant to me. But so through the years, the influence he had on me was I wanted to be involved with anything that he was working on, but it became clear that I wasn't going to be the best scientist in the world. I could do the field work, and it was fun. I could coordinate big efforts, collecting the samples, but when it came down to crunching the numbers and publishing my work, that's not what I was good at. And so he would have conversations with me, and one of them was like, you can still be really successful, but you don't have to be the best scientist. And he said, one of your skill sets is you're a good communicator. You're really good working with people. But if you judge yourself based off how many publications you get out, you're probably going to struggle. And I remember I've read a variety of leadership books, and one of them, there's a quote, I think it's by Einstein. I found it on the internet, so that's what it said. But it said, if you judge a fish by its ability to climb a tree, it will live its whole life believing it's stupid. And so what was interesting is I remember when I was working for the state, there's times where I just couldn't get a publication out. I couldn't get my work finished, and I just was trying as hard as I could. But then if you took me outside of that setting and said, hey, will you describe what you're working on? I could just talk to a whole crowd for a long time and be very passionate about it and describe it. And I just remember that was something that was a shift. And there's a few people that helped with that, but Lou was very influential. And so that was something that kind of made me do the shift. And so the job that I have now, I mean, most people don't even think I'm a biologist anymore. Most people think I'm like an attorney or somebody that deals with contracts and human dimensions, but that's my core, and I think, and that's worked out really well for me where I'm at now. But he was such a big mentor for me and most people in this room, but I appreciated what most people have said. But what I noticed was he was a world-class scientist, but it was the way that he communicated that really set him apart. I've seen multiple presentations that he gave in front of the heads of NASA. I've seen him give presentations to a variety of different things, and he could take the most complex topics and make them understandable to the general public or to decision makers, and I thought that was such a skill set that you didn't see very often, and it really set him apart. But then it was also the energy that he had, and it was it was just a big magnet. And so working in a big ranch setting now, I was thinking about topics for what this would be, and I heard the term hitching your wagon to a star. And so that's where I came up with the idea for the topic for this, but it really was. And I know a lot of people in this room hitched their wagon to Lou, is because he had such an energy about him, and then you just felt so good being in his presence that you knew if you were in his presence, you could do more. And so that was something that anything that Lou was doing, and if I was able to squeeze my way and be a part of it, I would do it. To the detriment sometimes where almost like I'd play hooky to go hang out at NASA all the time with Russ and Lou, but it was such a great time. So we had a lot of really fun times, but it's probably some of the best times of my life were going out and catching alligators with a lot of the folks in this room, but probably most of all was Russ and Lou, and so we had some really good times with that. And so there's a variety of stories, and so that I could say, and we were talking about them last night, but one of them was there's an email after one of the times I played hooky, and it was just Russ, Lou, and I, and it was when, I think was this Atlantis? And so we were going around, and it was just a perfect day. Where I was there for a couple days, and we were hanging out, catching gators, doing a variety of work. But then afterwards, Lou sent this email, which I, for the life of me, could not find this email. But it said something along the lines of, and he sent 2 photos in the attachment. It said, in this first photo is this world-class scientist in awe of this once-in-a-lifetime opportunity in front of the last space shuttle. And you can see this is a great photo, and it's right in front of this, and just the fact that we had access to be able to be in this location. And then he said, in the second photo are his colleagues making the most of the same opportunity. And so that's Russ and I. And it was one where it just made me so happy to be part of the team, but it was funny because we had a lot of fun where some of the research was great, but I mean, it was hanging out with your friends and doing great work. And I think that was one of the things that we really liked. And so the other thing, like Russ mentioned it yesterday, but Lou mentioned the dream team a couple times. And so for most of the field work, they would separate Russ and I, where we would have different boats. And so everyone would be on different boats. And so not to say that we're competitive, we were pretty if you know me and you know Russ, you know a few of us, we're pretty competitive. So you wanted to catch the biggest alligators, you wanted to catch the most alligators, and you wanted to catch them faster than anybody else. And so, but we'd have all the separate boats up, and so a lot of times they never put Russ and I together. And so but eventually one day we're on Woodruff, and Lou said, okay, I'm going to put you and Russ together, we'll put the dream team together. And I think we had to catch, correct me if I'm wrong, I think it was 24 adult alligators in that one day, and I think there was 4 boats. Russ and I think, caught somewhere between 14 and 16 of the 24 alligators, and so we were very successful. But it was one where it was just such a good thing to have, but we were those were some really good memories. And but it was something I think Russ told me, that the amount of times I think I was just so lucky to be out there and just spending time with Lou and Russ. And Russ had mentioned it was almost like, for Lou at the time, it was like therapy going out to NASA. So you got a glimpse of a window of just a lot of personal time, riding in the truck, riding on the airboat, and so you got to spend a lot of time. And so I was so fortunate where there was a period of time where I had the availability, even though I was kind of playing hooky from work sometimes, which basically work, but I was able to be there, and it really helped shape me for the person I am today. So if you see this photo, that's Russ right there, and that's that fancy hair right there. And so this was one of the, I think this was a field tour we did one time, and a lot of the folks in this room were there, but that was a great time. So the other part was just being a storyteller, and so you could just sit and just listen to stories from Lou, and he had such detail about the different things and his travels, and I thought that was incredible. And so I listed a couple stories that some of the folks know about in this room, but we could tell stories for a long time. And so I think all of us became better storytellers by being around Lou. And so we had some where, Arnold and James, we were on Lake Apopka, and they had an 11-foot alligator on the boat and sunk the boat, and then it became known as like the submarine airboat. So that's a fun story. It talked about the dream team. Cameron Carter, who's one of the biologists that used to be with FWC, there was a time where he turned into like a human torpedo. And so there's a lot of great stories, but I'd say one of my favorite ones, which I had PTSD over for years because of this, was Russ for years was working on getting this brand new airboat. And if you've ever seen it, it was this really nice big airboat, and finally got it, and he goes, "Hey, I'm going to be up in the helicopter, but I'll trust you to drive this airboat, but can you take it into these places and go check these nests?" So then I went out and proceeded to, I don't know how the airboat didn't sink, but it almost sunk to the point where it filled with water, went under the water, and then Eric, what's that his name? Eric? Yeah, Eric jumped on the bow, and we filled the boat with water accidentally because we were coming off like the edge of the water and went in, and it was such a big boat that it just torpedoed into the water, and luckily had enough power to pull out. And then went like this, and then I thought it was going to roll over, but somehow it didn't. And then so we proceeded to drive for a while, and bilging the water out and sweating, and then it eventually kind of righted itself. But it was one of those where, come to find out, Lou basically did the same thing a few days before that same day. So it's almost like he gave me a cursed airboat, but yeah. So the other thing is I've read a lot of these leadership books and other things, and I remember a couple years ago I read this book, Multipliers. And as I was reading through it, this was a few years ago, and I kept thinking about Lou when I was reading this, because you see a lot of these words that are repeated in people's conversations and stories and how his influence was, and he really was a magnet. He was a magnet for really talented people, but it was also, he was an amplifier, where he seemed like you could just get more done being in his presence. And it could have been because you wanted to make him proud, but it could have also been that he could just hone your skill set in such a way where you could do more because of just being in his presence, and he'd try to clear out some hurdles so you could be more productive. And that was just something that I noticed with that, and that was just, I know it's a good book if you haven't read it, but literally Lou could have been one of the case studies in that book. So anyways, it just, his legacy will live on, and I know even personally, I think about Lou all the time still, and it's one where the influence he had on me was tremendous. And so these are both my kids, and what's funny is I thought it was the same photo. I was wearing that same shirt in the delivery room for both my kids being born, but I actually just wore a different shirt just because it looked like it was so similar. You can see I used to have hair. I don't have hair anymore, but because it's a cool story when you go into the delivery room and all of a sudden looking at the shirt, you're like, hold on, alligator OBGYN. So it raises some questions, and it's something that I was always so proud to talk about because I knew Lou, and he was one of my friends. That's all I got.
So the chair of our next session is Lori Cruz, and she's an associate professor of biology at Wofford College. So welcome, Lori.
I was one of the last cohort of graduate students here at the University of Florida, and I think about Lou almost every day. I'm teaching a comparative biology of reproduction course right now, and when I was preparing it, I went back to my old notebook from when I took this class here. And it just means a lot to me. Also, I don't need them, but I wear my purple camo pants. As often as possible. So our first speaker for this session is Dr. Roger Newman, and his talk is called Dr. Lou Guillette Jr., Professor of Obstetrics and Gynecology.
Thanks, Lori, and thanks, Jerry, for inviting me to be part of this. So glad to be. From the hand raising people did yesterday, it's obvious that everyone knows about Lou's Darwin Day parties, and that to Lou, there was no such thing as a theory of evolution, there was just evolution. So what I wanted to talk about today, from my perspective at the Medical University of South Carolina, was Lou's evolution from being a lizard man that we heard about earlier to an OBGYN man, and we were proud to have him. In late 2008 and through most of 2009, I was asked to be on a search committee for a COEE endowed chair of marine genomics. It was probably the worst job I ever had because we had a pile of application packets and resumes and journal articles, and I learned more about zebrafish and ice fish and fish that glow in the dark than I ever really wanted to know. But the application that I kept coming back to was Lou's, and specifically to this particular paper that's cited here that he was really the primary author on, regarding female reproductive disorders and the role of endocrine disrupting compounds and developmental timing. It was a fascinating paper where they reviewed reproductive biology, the effects of endocrine disrupting agents to disrupt aspects of reproductive endocrinology, and the timing of when those insults might have occurred, and the role of endocrine disrupting agents in all of these different areas that in OBGYN are things that we see every day. We see a rising prevalence of polycystic ovarian disorder and menstrual irregularities and endometriosis and fibroids. On here it's listed as early breast development, but breast cancer as well was talked about in this article. In my mind, and this is the conclusion of the article, and you can read it for yourself, the last line is that the review of the EDC literature as it relates to female health provides an important platform on which women's health can be improved. To me, I read this and it was a manifesto. It was like a Unabomber type of thing. I had been an obstetrician for 25 years. I was involved in American College of Obstetricians and Gynecologists. I was a president of the Society for Maternal Fetal Medicine, and I don't think I'd ever heard a single lecture or been to a conference or even a grand rounds presentation about endocrine disrupting compounds and the role they could have in women's health. And after being on this search committee, I knew that we needed to have Lou as our COEE chair, and he needed to be in the Department of OBGYN, which we were able to do. So we welcomed Lou to Charleston as a professor of obstetrics and gynecology. I don't know if how proud he was of that title, but I thought it was pretty cool. and he was the director of the Marine Biomedicine and Endowed Chair of Marine Genomics at the Hollings Marine Laboratory. And as people have talked about already today and yesterday, Lou had an incredible ability to bring stakeholders together, to involve multiple different people in multiple lines of research. He really had boundless energy and curiosity. And these are just some of the many other things he was involved with, some of which we've talked about here, some of which we haven't. We talked earlier about his work with the crocodiles in Africa. Dimitri talked about the Gulf of Mexico oil spill. He continued his work with alligators at the Yawkey habitat, natural resources habitat up around Georgetown, South Carolina. We'll talk about infertility in a second. Satomi Kono became involved in looking at the pharmacogenetics and pharmacokinetics of 17-hydroxyprogesterone, which was being used at the time for prevention of preterm birth. We initiated a study that Laurie Cruz is very familiar with. On here, he called it the MOM study. We called it the reproductive development study, where we had a cohort of women, pregnant women, where we collected urine and blood samples during their pregnancy to look at a variety of phthalate metabolites. When the babies were born, we collected cord blood, we collected biometric measures on the baby. Laurie wandered the hospital measuring penile lengths on newborns and frankly was never stopped by security. So I was always amazed by that. But Lou was a kid in a candy shop. I know that MUSC creates frustrations because it does for everybody, but he was excited to have the opportunity to work with investigators from the College of Charleston, laboratories, at the medical university, working with NIST investigators, working with NOAA investigators, working with Department of Natural Resources folks right there on the Hollings Marine Laboratory campus. So he was involved in everything, and he cut a wide swath through the OBGYN department. Also, as everyone's already shown or talked about, Lou was all about his students. Besides the ability to organize stakeholders, it was his ability to inspire students, and he continued to do that at MUSC. There are a number of different students that he worked with. I won't mention them all. A couple of his PhD candidates that he and I both shared time with on their thesis committee, one of whom was Jessica McCoy. She could not be here, but as you can see, she was very successful in carrying on the research that Lou did. One of the reasons that I wanted to mention Jessica was a story that I had to hesitate. I didn't want to tell yesterday because I knew I'd tell it today. Lou came to all the departmental OBGYNs, or almost every one when he was around. And my thought was, there is nothing here that would be a remote interest to Lou. And that was because I failed to appreciate that Lou found everything interesting. So we were there one day when one of our Rico Suave infertility specialists was giving a talk. And as soon as the talk was over, Lou went up and talked to him and said, "When you aspirate those follicles, what do you do with the follicular fluid?" And the answer was, "Well, we throw it away." Well, by that afternoon, Jessica was already over at Coastal Fertility Center scoping out the laboratory and what's going on, and began a project where, as they were harvesting eggs for women getting ready to undergo in vitro fertilization, Jessica and the PhD lab person there in the, Coastal Fertility were collecting the follicular fluid, which resulted in the study, one of the studies that was referenced yesterday, about the presence of various EDC compounds in ovarian follicular fluid. We had a whiteboard of subsequent studies that needed to be done looking at the relationship between these concentrations of EDCs in the follicular fluid and the quality of follicles, the estrogen levels that mothers were having as they were being stimulated, the quality of the eggs, the fertility of the eggs, the ultimate implantation rates. And I have no doubt that those studies, which unfortunately weren't done, I have no doubt those studies would have shown that there's a relationship between the success and failure of in vitro fertilization, depending on the levels of follicular fluid EDCs. Abby Wenzel was another PhD candidate we had who's also carried on Lou's work looking at exposures. Under Lou's guidance, she was one of the first people that I know of who was looking at phthalate alternative that were emerging in the early 2010, 2015 time period. One of my partners, he had an influence not only on PhD candidates, but on our other OBGYN faculty. Rebecca Wineland's a sonographer, never had any interest in reproductive endocrinology or reproductive effects like that. But in a conversation with Lou, she said, and this is when we were doing the reproductive development study, and Lori was measuring penile lengths. Rebecca said, I can measure those in utero. Would you be interested in doing a study where we looked at the effects of maternal phthalates on male genital development in mid-gestation? And of course, Lou was always interested in everything. So we did another study, a very, very nice study, looking at the effects of anti-androgenic effects of different drugs on male genital development measured in utero. He influenced our fellows. Mary Sterrett has now gone on to she's an obstetrician, gynecologist, maternal fetal medicine specialist, but is doing reproductive environmental science studies in Africa. Although he was not one of our fellows, Barrett Welsh from the NIEHS contacted us several years ago and said, your reproductive development cohort is a beautiful cohort, and I'd like to include that in a pooled cohort study to look at urinary biomarkers of phthalate exposure and preterm birth. And he's now published, I think, 3 different studies using that cohort showing the relationship between certain phthalate metabolites and preterm birth. The point of all this is that Lou's effect, he liked to talk about intergenerational effects of things. And Lou's influence passes down from person to person to person. One more to show is the picture here of Michael Bloom, who's a PhD from George Mason University, who came to us again after Lou had passed, but wanted to pick up that work. And Michael he's published multiple articles using some of our biosamples and based on the studies that Lou helped us put together. And I showed this slide specifically because of the last paper mentioned here, which was referencing the ECHO fetal growth study. Anyone here are familiar with ECHO? So a number of heads nodding. Lou used to love to kind of make fun, tongue-in-cheek, of the Human Genome Project. Billions of dollars over a decade or longer that showed we had far fewer genes than we thought we had, and we didn't really learn anything new about health and disease. And his slide that I think someone showed the other day with genes and a big giant Earth for the environment and epigenetics, ECHO has Lou's fingerprints on it. It is a congressionally funded study called Environmental Determinants of Child Health Outcomes, and the goal of which is to recruit about 50,000 maternal child pairs, follow them through their childhood, through their adolescence. We are one of the sites that are part of ECHO. we would not be part of ECHO if it weren't for the work that Lou did. Michael is has multiple research projects ongoing in ECHO, looking at phthalate effects. So again, it's the fingers of Lou reaching out and touching other investigators and other generations. I didn't have a picture of Magana Varde, so you have to look at Michael's ugly mug again. But he's her PhD thesis mentor, and as you can see, she continues to be interested in endocrine disrupting compounds. So it's, again, an intergenerational effect that passes down along the line. It also crosses specialties. Everybody got to show their kids yesterday, so this is my daughter as a high school student out with Lou and riding an alligator. She's now a neurosurgery resident at Emory University, but to this day, we'll talk about the time that Lou tricked her into eating alligator chili at one of his Darwin Day parties. I was just one of many, but one of the things I'm very proud of in my career was having the opportunity to write a letter of nomination for Lou to receive the Heinz Award, which, as everyone in this room knows, is really kind of the Nobel Prize for environmental research. And my take-home memory of Lou, which I'll illustrate with a number of his pictures, was simply, besides being the Pied Piper that he was, the genius that he was, the communicator that he was fearless environmental warrior. And just a couple of pictures, some of which you've seen before. I love this picture. He somehow had the ability to antagonize animals that he didn't have with other people, so that was... That's a great picture, too. This is a shameless plug. The other thing I do besides medicine is I write novels. My third novel is called What's Become, and it was a novel about the reproductive health impact of environmental dumping being done by the paper company in Georgetown, South Carolina. I'm surprised I was never sued by them. But a prominent character in the book, and part of the book's dedication, is to Lou, who's the guy who really figured out what was going on. And I used for the cover of the book, I used this picture that we've seen before of Lou's eyes at night, alligator eyes at night. That's all I have. I appreciate the opportunity to talk and tell you a little bit about some of the things that Lou did while he was in Charleston at MUSC. Thank you.
Okay, cool. So I was glad to hear that other people sort of struggled with their title, because I wrestled with what this actually meant and sort of went back and forth a few times. And I'm actually really glad that I went after Roger, because he actually summed this up in a way that I wasn't really able to, which was that I joined Lou's lab and came to MUSC when Lou was really starting to do all this translational work on human health. And I think I actually knew Lou as the OBGYN man and not really so much as the lizard man or even the alligator man. But so it was a lot of that work on those cohorts and the translation of actually identifying chemicals in people and then trying to think about what that actually meant for their health that I think really influenced my time in the lab and then also what I do now. And so
I was really grateful that Teresa was uploading all of the 4 terabytes of photos because she found this really amazing photo of everybody in the Gillette lab when we were at Hollings in 2014. And I mean, so many of these people are in the audience. People that aren't, we're all really still close and in touch with one another. And it was just an awesome group, and it was really fun for a few years. I was there, started in about 2013 to 2015, and so I only kind of got a couple of years with Lou, to be perfectly honest, but I feel like the mentorship that he imparted on me was really also just through, obviously, the legacy that he had with other people and constantly being surrounded with everybody. And so one thing, while I was in the lab, and so Dimitri also got to talk about the DOS project in my dissertation work, so I didn't have to do that here. But I wanted to talk about specifically one aspect of that work, which was that while we were working on chemicals that were found in the Deepwater Horizon spill, there was, an industry talking point that was, well, all the chemicals that are used in Corexit have been used in consumer products forever, and they're used in all these things, and they're totally safe, and they're having no impact on people. But there is still this question of like, well, where are they used? What does the exposure actually look like? Is there a reason to look into these chemicals? And so I came across the Environmental Working Group, which is where I work now, which is a nonprofit organization, because they had this database of ingredients that were used in consumer products. And at the time, and I think still sort of is, one of these ways where you can actually figure out where chemicals are being used and how people might be exposed to them. And it sort of got me interested in, maybe I want to be a little bit more connected to sort of that aspect of the work, thinking about exposure and then thinking about what it might actually mean for individuals, and then really how to communicate that to the general public. And so that's where I am now. And you'll see one of my lessons from Lou is about teamwork, and obviously everybody here has also talked about how amazing Lou was at building a team. But this is the team of people that I work with now at EWG, and it's this group of really wonderful scientists that are really focused on research projects that I think drill down into exposure science and then also trying to communicate that out. And I noticed, even earlier, John Wise was talking about his hexavalent chromium work, and I realized that there's overlaps with some of the work that people are doing here on looking at hexavalent chromium removal in drinking water facilities. So there's ways that Lou has actually influenced where I am now that I wasn't even aware of at the time that I was a grad student. Okay. And so there are a few lessons from Lou that, these are not groundbreaking. A lot of people have talked about this already. But I'll talk about sort of my personal story about, I think, where in my time at MUSC and a little bit of time after, they've impacted me. And then I also want to tell you guys a little story with some data that because I think people here might be interested in it, and I think Lou would think it was kind of a cool project too. Okay, and so we've heard this expression from Lou that the science do the talking. And I think that is true and safe when you're with a bunch of scientists. But if you're trying to talk about the science to other people, you have to also be good at talking about the science. The science won't just do it for itself. You can put a graph up there and people won't know what it means unless you actually talk to them about it. And Lou could do this, excuse me, Lou could obviously do this for really any audience. So, Lou could talk to kids, and I didn't even know about this, but I was digging through some of just news articles and things like that of Lou, and evidently, Alligator Man was talking to 500 elementary school students, 500 elementary school students, which honestly sounds terrifying. It's really impressive who he could talk to. And then he also talked to the public. So, there's numerous articles of Lou, but to be in a documentary that's reaching people's homes while they're sitting on their couches, watching TV and getting people excited and understanding about endocrine disrupting chemicals was pretty amazing. And then he could also talk to lawmakers, and I've been thinking about his congressional testimony that he gave because this is just news coverage of it, where really the foundational researchers in EDC science got up in front of Congress and were like, we have to do something about this. And it actually ended up, I think working towards passing some of the legislation around how pesticides are regulated in this country. And it's work that, I spend most of my days looking at pesticide risk assessment documents from the EPA. And all of this work is so foundational into how those assessments happen. And again, I kind of didn't know and wish I had more opportunities to actually talk to him about it. And then if we talk about science communication, this is his iconic quote, which at our staff meeting this week at EWG, I was mentioning I was giving this talk, and people in the room were like, I know that quote. I remember that, testimony. And so it speaks to his legacy there as well. And I just put on the left, this was, the wording that was used in the abstract of the 1994 EHP paper. And so to go from the wording on the left, which to be honest is already very well written and well summarized, I think people could understand that. And his scientific writing was already really accessible, but then you translate it into something that is gonna have really lasting impact in a way that people understand. Okay, and so science is a team sport. We all know this, but Lou built different teams that I think different people interacted with. And so at the center of these logos and figures is the molecular structure for DOS, which is the chemical we were working on and identified as a putative obesogen. And when we started sort of working on that project, it was just kind of us at first, and then once Lou started where it was going, he started to bring in a lot of people. So, we were obviously at MUSC, but then we worked with people at NIST. John, we spent a little bit of time with Teresa as well, figuring out if we could get an analytical chemistry method up and running and actually measure this chemical in breast milk. He brought in Bruce Blumberg from UCI, who was, foundational in understanding molecular mechanisms of adipogenesis and how chemicals would interact with those receptors. And he was like, take a look at this, what do you think? But then he also, which I think this is the piece that helped set me up for where I wanted to go, was, he knew that I was interested in communication, that I'd probably want to do some news coverage on this, and so he got me media training from the Science Communication Network, which is not something that every graduate student gets to do, and it made me really prepared for where I was going later in life. And he connected me with Pete Myers at EHN and got a news coverage article. Dimitri put it up on the slide. So just all this incredible work, he brought in Mary Selmafini, who does consulting work at FDA to understand, how does this chemical fit into the regulatory space, which I know a little bit more about now, but I didn't know when I was 25 and just learned about, EDCs for the first time. So it was really amazing, the team that he built around you so that you were supported and you got to go where you wanted to go, even if you didn't know what it was. Lou maybe knew what it was before you did. Okay, and then the third lesson was sort of just keep moving forward. I think people have alluded to this, that Lou, I think, could appreciate the wins, but if you wrote a paper, it was on to the next thing to some degree, and you were always drilling down into the next questions. And I also think about this keep moving forward in a way of sort of keeping up the fight. There was just so much work to do, there was so much investigation to do, and you needed to get it done, and so you kind of had to keep moving. And in the spirit of that, I wanted to talk about some research that we started a couple years ago in collaboration with Dimitri. So team building in effect right here. But this is a publication we put out it came out last year, and it was probably a work about a couple years ago where we were looking for this chemical chlormequat in food and urine in adults in the United States from 2017 to 2023. And I think people will find this study interesting, and I think I hadn't realized it until I started thinking about it, but there are so many aspects of Lou and the training that you get in sort of developing this research project. So this chemical chlormequat, or what our communications team called it, was just chlormo what? They were sort of we've never heard of this. How are you trying to pitch this to us? And it has some really interesting biology and aspects about this chemical. So it is a plant growth regulator, which is regulated as a pesticide in the US and abroad, even though it doesn't actually do any killing of weeds or insects or things. But it actually is an inhibitor of plant hormone synthesis. So it basically prevents growth in these crops, and it's used because essentially if crops get too big, like wheats and oats, and then you have really windy weather, rains, they're more prone to falling over, or what's called lodging. So if you add a plant growth regulator, you keep them short, they don't fall over, and you can theoretically improve your yield, and I'll come back to that. The other thing that was interesting about this chemical was that it's actually really widely used in the UK and Europe, but it's not used in the US, and there aren't too many examples of that, so there was something kind of interesting there. And the reason we got involved, or just kind of curious about chlormequat, was that, as I had mentioned, had been used in UK really widely on food crops. In the US, it was only registered as a ornament for ornamental plants. So, you know, like your azaleas, you don't want them too like big and bushy, so you add this plant growth regulator and you keep them sort of small. But in 2018, the EPA approved this import tolerance. Like it was kind of sneaky. You know, there wasn't any news about this. Like there's going to be a new chemical in your oats or wheat or things like that. But they had approved this import tolerance, and then in 2023, they actually, you know, suggested maybe we will apply this on domestic plants here. And so, again, I wanted to figure out if I should care about this chemical or not, and so I was interested in the toxicity. And there actually had been some studies published like in the early 90s in Denmark where pig farmers were noticing that there was basically decline in the reproductive success of the pigs that they were raising, and they determined that this came from chlormequat-treated grain that they were feeding their animals. So, I mean, the phytoestrogen story, like there's so many connections here too that I had never even thought of before. And basically, there's all this peer-reviewed literature suggesting that there's impacts on fertility and development that's not considered in the regulatory space. And so, you know, there's no epidemiological studies, there's no updated EPA reproductive toxicity. And so what we wanted to do was actually just ask this question first of like, well, what is the prevalence of chlormequat in food and people? You know, can you measure it? And then maybe should we think about looking at it? So I was like, I can't do this. I don't have a lab anymore. So I called up Dimitri and I said, do you have some urine samples we could test? I found a lab in the UK actually that we had to send urine samples to, and I was like, well, John taught me how to look at analytical chemistry and determine if these methods were good or not, so I can at least figure out if this is going to be legit. And so we sent these samples off, and basically we found, you know, that chlormequat was prevalent in US oats and food, oat-based foods, but not wheat-based foods. And then that it was also prevalent in people from the US. So we had samples from 2017, 2018, 2022, and 2023. And you know, we weren't expecting to actually see this much detection, but you know, it was there. And it was kind of, there's so many more questions that this has opened up. Might be an increasing trend, things like that. And so we published this, and then to come back to sort of this communication piece, this was sort of the EWG press release that we put out on this, which was that we found, you know, a little known toxic chemical in 4 out of 5 people, and we had our 3 takeaways that we wanted people to get out of this, which was, this is the first time it's ever been looked at in the US population. Federal rules allow this to be imported, but it's not used here. And there's some potentially concerning toxicity, so this might have interesting impacts on human health that needs to be investigated further. And so there were just so many pieces of this work that I felt like I wouldn't have been able to do had I not had the training in Lou's lab, or the teamwork and communications that I had built from that. And so I just wanted to close with, you know, it was nice to see that other people went and dug through their emails in terms of remembering Lou, because you know, sometimes like those words are the things we have and you don't really imagine how impactful they are at the time, but to be able to go back was really special. And so this email was from Lou after Teresa and I think 7 other graduate students, many of the ones that Roger mentioned in his previous talk, took our journal club, and we decided we didn't want to just discuss papers every week and how they impacted our work. We wanted to actually set up this journal club so that we were doing a review of the literature on phthalates and their impacts on human health, and then also review the literature on intervention studies and think about how we could give some advice to, I think, probably Roger, I can't remember who the audience was, but I think it was probably a group of healthcare providers or some people in the OBGYN department. The presentation was also attached to this email, which was awesome, so I got to go back and look at it. But like these words from Lou were just so amazing, and I think it you know, solidifies how he was saying, like, I know you each work harder doing the science, right? He trained people that were really good in science. But it was amazing to see the passion you bring to making a difference. The world, my world, is better because you guys are in it and I share it with you. So there were so many things, you know, I think that, like I said, Lou embodied all of those lessons. He didn't necessarily just teach them to you. He led by example. And then, of course, at the end, he mentioned, let me know what I can do to help you move this forward, including turning this into a review paper. So, that was his closing. And yeah, so that was Lou. And yeah, thank you. This was a photo I took at one of those Darwin parties as well, because that dock was an amazing place to sit and just look at the sunset with friends. So, thank you.
All right, next we have Thomas Rainwater from Clemson University, a long-lasting influence. One man's experience with Dr. Lou Guillette Jr.
Hi, everyone. For those of you who don't know me, I'm Thomas Rainwater. I just wanted to say I feel very, very honored to be part of this symposium. I'm going to go through this quickly, surprise, surprise. So try to buckle your seatbelt. So, I could just stand up here and drop the mic and say what they just said, because everyone today is pretty much, it's going to be a lot of overlap with photographs and the sentimentality and the message, but we know that Lou has profoundly impacted science, so many different areas of science, from evolution, ecology, reproductive biology, endocrinology, herpetology, ecotoxicology, all those and many more. And also he's impacted individual people and groups of people. So today I just want to quickly, as best I can, go through and just describe how he's influenced my life and my career. So my little story starts in the early 1990s. I was a master's student at Clemson University in environmental toxicology, and I was studying the exposure and response of wild birds to turf grass insecticides on golf courses on the coast of South Carolina, which is a really cool project, and I was enjoying myself, but the problem was everywhere I looked there were alligators, and I was much more interested in alligators than I was in the birds, and so I would go back to my advisor at the time and say, can we change the project, or can we add this into the project, or can I do another project? And every time I would do that, he would kind of feign interest and not really give me the time of day about this. He just changed the subject. And finally, one day I cornered him and said, why is it you always just kind of blow me off when I bring this up? And he said, to be honest with you, Thomas, because he's an avian toxicologist, he said, I don't think you're going to find any way to get this work funded. It's just not, where the money is and where the interest is right now is in avian and mammalian ecotoxicology. And so I went back to my corner, and I finished up my degree, and I ended up doing I went into an internship at a chemical company, actually, in Princeton, New Jersey. And while I was there, something happened that changed the trajectory of my life. And it's this paper you all have already discussed. Lexi just had it up there a minute ago, but his big, first paper, 1994 EHP paper, that described all the reproductive abnormalities of juvenile alligators in Lake Pocket compared to Lake Woodruff. And you all know that story, but the cool thing was that immediately just changed the script. Whereas birds and mammals are everything, but now alligators were on the front page of the newspapers, and Lou Guillette's out there, preaching the word, and everybody's listening now. And so, fast forward a year or two later, and I changed advisors, and somebody would give me a shot with crocodilians, and we actually wrote grants, and we actually got a 3-year grant from the US EPA for me to do research on not alligators, but actually go to a country where some of these contaminants like DDT or other organic chlorine pesticides were still being used to some degree for malaria control, and that country is Belize. And so I ended up going to Belize. I spent 4 years there working on the ecology and ecotoxicology of more or less crocodile. It's a freshwater species that lives on the mainland of Belize. And the idea behind this our proposal was just we're trying to kind of mimic what Lou did here. We just wanted to go to a place where we have a different species, but these chemicals are still being used, and look at some of the same endpoints. Hormone concentrations in the blood, phallus size in males, patelogenin induction, stuff like that, plus looking at contaminant concentrations in their eggs, and all these different things, while also looking at the ecology of the animal, because not much is known about this species. So I went and did that. It was a life-changing thing for me. I met my wife there. It changed me professionally. I was able to kind of get a foot in the crocodilian door and began to go to meetings, and was able to kind of get my career kind of jump-started from that. So I get back from Belize, and now I've got 4 years worth of samples I got to analyze. And I had no clue how to do a radio immunoassay. And so we went in there, we tried to figure it out, and of course I failed. But I'd known Drew from Clemson University, and I called up Drew, and he just graduated from Lou's lab, and he was in Maryville College, and I said, Drew, I'm having this trouble. Do you think you might be able to help me out? He said, I'm actually going to Lou's lab in a couple of weeks to run some thyroid hormone assays, and I'll talk to Lou and see if you can come down when I'm there, and we'll introduce you to people, and we'll see if you can learn the assay. I said, man, thanks so much. So there I went, and I spent a week in Lou's lab, my first time in Gainesville. I got to meet Lou. It was like meeting, super, your biggest rock star. I was in the same room with Lou Guillette. And here was this gracious guy. We'd go have lunch and we'd chat, and he was very busy, but he spent time with me. But I met, I met Matt Mills, I met Thea Edwards, I met Tam Barbeau, all these different people in the lab at the time, and they all were very generous and helped me, taught me the assay as best they could, showed me what equipment I needed. And I went back with a notebook full of notes. And I went back and we got what we needed to get the assays going, and 3 years later, everything got analyzed and I was done. By the way, there's this mask Tyson was talking about, the worldwide Lou's mask he took back from Japan. Anyway, an incredible experience for me. Got my project done, and then fast forward about, I guess after a postdoc at UC Davis, and about 5 or 6 years of being a mercenary scientist, I would just kind of go different places and do different projects, including Matt Mills, actually, when he was in San Diego, having met him in his lab, he sent me to India for 3 months to work on a project, which is incredible. Different story for a different day, but I worked on a bunch of different projects, bounced around, but I ended up coming back to South Carolina, where I'm from, and there's Woody on the left and the bottom, Phil Wilkinson on the right, and they were doing a long-term study on the nesting ecology of American alligators in South Carolina, and so I've talked to those guys, they invited me to come help them out, I just want to get in the field with them. So I volunteered for the first year and had a great time with those guys, learned a lot, and was really kind of enjoying it, even though I didn't have a full-time job. But I came back home one day from work, and somebody sent me an email, hey Thomas, by the way, I don't know if you know this, but Lou Guillette's going to be in Charleston next week to give a seminar. Oh man, that'd be so cool to see. I haven't seen Lou in a couple of years, and it'd be cool to see him. He has these great talks. I have to go do this. And so I went over to the Marine Lab, where Roger just was talking about, and I walked up to the door to go to the seminar, and I saw this flyer on the door, and it kind of caught me in my tracks when I looked at it. And what caught my eye was this candidate from chair of marine genomics. I was thinking, wait a minute, this can't be right. Lou Guillette's even considering leaving the University of Florida. This guy, he is the University of Florida. He's Mr. Florida, at least in my mind. When I thought of Florida, I thought of Lou Guillette. I couldn't imagine, he would leave there, much less come to my backyard where I'm already living. This is too good to be true. And so I went to the seminar. It was great. Talked to Lou a little bit afterwards. He was very busy, but it was an awesome experience. Went straight home and emailed Matt Mills, and I said, Matt, do you know much about this? Is he actually really considering leaving the University of Florida? He's like, I think he might be. I don't know for sure. Lou, he's in demand everywhere, but apparently, as Roger mentioned, and I think Roger, you were searching for several years to fill this position, and finally found the right person. I didn't know that the backstory that Roger told there, but they offered him the job, and he took it. And sure enough, he showed up in the next, I think 2010, showed up in Charleston. So the first thing I wanted to do was get Lou up with Phil, because Phil was that South Carolina alligator biologist, and we've been there for many years working too. And I wanted to get these two guys together to talk about collaborations. And so Lou came up, we spent a couple of days in the field together, and that collaboration was born. And the best part was, so here comes Lou with all this incredible, everything that Lou brings, right? The people, the knowledge, his personality, all these things. And yet Phil, his 40 years of knowledge of alligators in South Carolina, but Lou also brought this, all the students and the postdocs and all the lab know-how. It just exploded into incredible research that we did, and the students started doing their projects in South Carolina. And and what was really cool was after a year of working together, so I think in 2011, Lou brought me on as a postdoc in his lab at the Marine Lab. So I actually was getting paid. I'm not working in the lab with Lou and his students, which was pretty much heaven for me. And so, what the other cool part was, I got not only working in South Carolina, because Lou kept working. He was working in both Florida and South. I get to do that thing John mentioned about. I get to hop in a truck with Lou, and for 5 hours, just the two of us. And we would just drive down and go meet Russ and go meet this group. It was amazing conversations that we had about everything, but it just changes you. And amazing to have him, because it's hard. He's very busy. Back at school, you can't get. He worked hard to spend time with you, but it was hard. Having 5 hours with him there and back was amazing. So I got to meet all the players, all the FWC guys we talked about today. Arnold and. Cameron, he's not here. Patrick, God, there's so many. Anyway, so we spent time in the field, and it was a great experience. And then I also got to go to work on some of the projects at the Kennedy Space Center, which is obviously a really cool experience. And in doing so, got to meet Russ. And got to learn how to, the right way to catch alligators, right? The correct way to catch alligators, and also the correct way to collect samples and the correct way to process samples. So you get back to Russ's house and pull over the centrifuge and get everything done. But anyway, long days, but we got it done. So back over. Oh man, sorry, Matt. But the bottom line here is that I also felt part of a family. When you work in Lou's lab, and this is obviously part of that family, you felt part of this huge academic family, postdocs and former students and people working in government and academia and industry, all these different people are together. And it was just amazing. It was amazing to be a part of that, and it still is amazing to be a part of that. I was in heaven, and I was working in Lou's lab. I didn't want to go anywhere. But all of a sudden, after 30 years or so, the U.S. Fish and Wildlife Service in Charleston, their toxicologist position came open. The woman that was in that position retired after 30 years. And it came open, and I talked to Lou about it, and I said, man, I really don't even want to apply for this. I love working with you guys. I can't, other than making some more money, this is my dream job. And he's like, listen, he said, just apply for the job. If by chance you get the offer, then we can talk about it. But don't pass this up. These jobs are hard to get. And you might be able to get it. So I did. And I applied for it. And long story short, I got the offer. Went back to Lou, and I said, I'm not sure. He said, listen, he said, if something happens to me, I can't promise you don't have a job. He said, just, I'm just giving you my advice. He said, but I'll consider taking the job. Your office is going to be 5 miles from where we already are. We can still collaborate on the side on projects and all these kinds of things. I said, and after consulting with him and others, I decided to take the job. So I went to work for Fish and Wildlife Service, and I was there for a while. But his wisdom and his encouragement was vital to that move. And then while I was with Fish and Wildlife Service, I still was able to keep projects going in Belize, and even work with Lou in his lab, so it worked out great. But one thing I mentioned this yesterday, I'm going to go through this kind of quick, but it meant a lot to me to be able to try to invite Lou to go back to Belize. And I want to show you where I used to work and the things I used to do and meet the people I worked with. And you kind of, indirectly, you got me there, and I want you to see this. I know you enjoy it. So Matt and Russ and I went down there. And while we were there, of course, I told you the story yesterday. I'll get through this quick. Lou immediately, found these folks, and they just glommed onto us. And he spent the entire week, working with this is the woman and her daughter, and her niece was there too. And we had just a great time with this family. And we went back inland. Of course, I told you about the Maya ruins and so forth. Indiana Jones got nothing on Lou Guillette. So we were there, and we went walking around. And here's the high temple. There's Lou in the foreground. You can see him there. I think he's holding the machete. That's his camera, actually. And there's Russ up on the first rung of the temple. But we climbed up the top. I think that's Russ and Lou going to the top and going to the very top. And then, Lou we got up there. It's a gorgeous view of the canopy of the entire river system and everything. And Lou's like, "We're coming back tomorrow morning." So we came back the next morning before dawn, and Lou was setting up his camera at the very top, and of course got one of those really nice sunrise shots. It's kind of a little bit hard to see, but it was a really cool moment. So we got back. I think Matt mentioned this yesterday. While we were there, my son was about 2 weeks from being born. I'd missed the Africa trip, I think the year before, because he was, well, I think it was the same year I missed the Africa trip because he was born. They went to Africa just after they got back from Belize. Anyway, 6 months later, in the early part of 2014, Buzzy and Lou show up with this beautiful quilt that Buzzy had handmade for my son. And, just very, very, very sweet and meant a lot. We still have it. And, get you thinking, I'm a father now, obviously changes the way you think about things. And I have to admit, having been around Lou a lot, you can't help but to watch the way he and Matt interact. And I just thought he was an incredible father, and meant a lot to me to be able to see that. So I thought, there are some things about Lou I wanted to emulate as a dad. Unconditional love, support, all those things. And another important thing is, early exposure to the family business. Somewhere here, there it is. So I tried hard to do the same thing. But anyway, it meant a lot to me. It still means a lot to me that my son, even though he was little, got to meet Lou, and he knows all about Lou and Buzzy to this day. So that was another influence he had on me. And then in 2015, I finally got a different job. I work for Clemson now. I work for South Carolina DNR. It's kind of a hybrid job of doing wildlife research in the coastal plain of South Carolina. So I'm back into research again. The Fish and Wildlife Service job was mostly regulatory. It was a good job to have, but it was not, really my wheelhouse. I found it was just not as much fun to me as research was. So I'm doing this job now, and the best part of that job is being able to work with students. And again, we've heard a lot about Lou's mentoring prowess, and he's a legendary mentor, legendary teacher, legendary researcher, and I'm none of those things, but I try my best to give students the best. I can't have my own students because I'm a research scientist. I'm not faculty, but I can be on committees and I can actually, I can take them in the field and help them get their samples. So the cool part of that is I get to collaborate, right? So I can collaborate with all my family members from Lou's lab and elsewhere. And so all these students, I'm not advising them, but I'm working with them in the field, which is very rewarding. And so we've been doing that for I guess this summer will be my 10th year in this position, which I just, I feel guilty saying this because I know people's jobs right now are in jeopardy, but I love my job and I feel fortunate to have it. And in addition to that, I'm just going to mention this real quickly, it's collaboration. These are just folks from Lou's lab that I met while I was with Lou that I have collaborated with since Lou's passing. Including Matt Mills, who sent me to India for a while back, but Brandon and a variety of different projects, lots of different papers, and we could talk at length about what we've been working on, but there's not time for that. But the bottom line is there's a family here, and these collaborations hopefully will last a long time. And I'll finish with this. I think I bounced over one, didn't I? One of the things that was cool about Lou is that when he was at Hollings Marine Lab is that we would, because we had one in South Carolina and in Florida, there was so much going on. A lot of it was collaborative. We were doing different projects that kind of overlap. That early spring, about this time every year, February, March, we'd have a meeting, either in Charleston or we got to Nashville somewhere. We get all of our collaborators together, and we'd have like a day and a half, or maybe just a day meeting to talk about what we had done the previous year, share the results, and what's the plan for this year? What are the questions? What are we trying to achieve this year? And it was really fun to get together with the group and kind of plan and be organized for the upcoming field season. So after I got my job and after Lou passed, we decided to do a similar thing, but to cast a little bit wider net. So what we've done is we've created this thing called the Palmetto Research and Alligator Research and Management Symposium, or PARMS for short. And we have it once every other year, and this year we have it in 2 weeks. This will be our 6th one. And we pretty much just, any people working on crocodilians of any kind from anywhere can come, but mostly people from the southeast come, and we have generally anywhere between 70 to 85 people show up, and we just talk about the latest science on crocodilians, whether it's alligators, crocodiles, caiman, whatever it is. And just in case you're interested, the next one is in 2 weeks at Savannah River Ecology Lab, where Ben's lab is, and registration ends on Wednesday. We've already had close to 80 folks coming, but I'd love to have anybody else that didn't know about it that wants to come over. And so I'm just going to end with this. I kind of warned Tammy about this, but this is back out on the coast on the atoll in Belize, where we work together a lot, and some of Lou's just breathtaking sunrise photos. And you may have seen some of these before, but Lou's the kind of guy that I think I've said this before, that you could be eating a sandwich or walking down the street or reading the paper, and he comes in and he gives you a phone call, and 2 minutes later you're ready to take the next hill. He just has that ability to really, really energize you. And he's had an incredible influence on my life, both professionally and personally. And I want to thank him for that, and thank his family for having us. Cheers.
All right. For this last speaker for the session, we have Russ Flowers from NASA, talking about Lou's legacy, amazing science, and the family team.
All right. So it's kind of be tough on me, but I'm Lou was a very amazing man that just inspired all of us to do what we do today. He basically took hundreds of people and put them together. We're all part of his family. The thing that people talk about right now and the way that he's been affecting their lives shows you how valuable the man was to us. He, let alone how much science he accomplished. He always had time to talk to you, to email you, call you. He always took care of all of us. And that was what was really cool about Lou. So, sorry I have a cold, can you tell? Anyway, Lou basically inspired all of us to do a lot of different things, and I was just very happy that I was a part of this team. Basically, the family members that we all have. We have Buzzy, who was like the instrumental part, or the background of all of us. She allowed us to be with them, to be part of all of us. So, and Matt and Teresa, they actually met in Lou's lab. We got to see all that happen during the time we were catching gators, which became a very big thing for all of us. We all acted as a big team, and that's the thing that was most important with all of our research. It wasn't just one person doing one thing, it was hundreds of people accomplishing a goal to basically provide more information for science. And that was the goal for everybody. Whenever you were with Lou, you were a piece of the family, and that went through everything that he inspired. Whenever you joined his team, basically we were all safe because we all watched out for one another as a family. Here's more of the family members that we actually had symposiums where people could actually come out, and the family would actually become one as a whole, and we'd actually go and have these symposiums to basically do information exchanges just like Thomas was talking about. We tried to have them every third year if we could. We actually ended up doing 4 or 5 of these international symposiums. We actually had people come from Japan, from South Africa, and all around the world. And with that thought, we started to work at NASA. NASA was a big problem for Lou to get off the ground. I actually proposed this information to him and Ashley Boggs in 2003. We sat in a restaurant, got a napkin out, kind of drew up what Lou saw as what we could do or how NASA could provide information with the animals that we had on the center. With that information, Lou said, okay, we can do this, we can do that, we can do this. I've got the students, I have the availability, we can make this work. I said, all right. I said, I've never caught an alligator. I had no idea. I'm a Colorado boy. He said, don't worry about that part of it. We got people, Woody will take care of that. I'll show you how to catch an alligator and we'll be on with it. So, is that all right? So we wrote a proposal to NASA in 2003. It took 3 years to actually get that off the ground. We got off the ground in 2006 and finally started to go try to catch alligators on the Space Center. You've all seen this one a lot. But anyway, NASA became kind of an instrumental part of Lou. He loved the fact that NASA was embroidered within his soul. He could tell people that he worked with NASA, which was awesome. Because once he got inspired with NASA, he incorporated more people into his family to run more samples. But what Lou liked the most is when he came to the Space Center, he actually could fly in helicopters, he could go on the VAB. We had full rights to do anything we wanted. And most of all, we could do it with family. But Lou was kind of a little embarrassed, sometimes. And but he could have assembled teams of people to actually do the work. As you can see on the far top right is Brandon Moore. That was our first alligator we ever captured on KSC. And all the rest of the people that actually provided support during our stuff on Kennedy Space Center. So now when Lou needed more support or family members, all he had to do was point his finger. And hundreds would come running. As you can see, we've got a lot of heavy hitters in here. We have Graham Webb, Xander, we have John, we have Patrick. You can see him right behind me. Patrick's over there. But anyway, all these people actually supported us to get the job done. Oh man, I hit the wrong button. Okay. So anyway, we started our studies. We actually got to go to Belize, went to South Africa. We did a lot of work out at we basically worked the entire world to get these samples done and processed. And this wasn't done by just one person, it was done by the family. Each family member had a different thing that they would do. Some people would catch the gators, some people would process the animals, and some people would actually store the samples. We had stuff going on everywhere, and to have Lou as a center person was amazing to be able to do all this stuff. People don't realize how much Lou had under his belt and how many people he had to communicate with different processes within the human body. So with all these people, we had different things going on. At NASA, we did egg collection work, we did hatchlings, we did big gator captures in Belize. We actually did some live captures, and we did a lot of nest work, trying to find out how many nests there were. And then at South Africa, we did a lot of work with the large animals. So Lou would go out and show you how the work was done on the left. Then after he figured out that his family knew what was going on, he'd sit at the bar drinking his peach tea, and he'd send his crew out to go do all the work. That's an intelligent man. So, okay, so they sent all the people out to work. And then, so at NASA what we did is we actually did thermistor work on the right, we actually did egg collections in the middle. And we also had some problems during some of our surveys. Basically, a wheel flew off one of the airboats once. We had a lot of other stuff that happened. But on the top left is my wife. When I first met her, it was funny because a hurricane was coming, and we took 360 alligator eggs and shoved them in her garage. How did I meet this guy? So Lou was always taking pictures, no matter where you went or what you did. That was, I think, his relief. It's one thing that he could do to actually get away, which kept his sanity, because he had a lot of family members to provide for, like these guys up at Yaki. And then we had all the people that worked around the Space Center. Now, as you know, Lou is the photographer machine of the world, and he has family members, and some don't learn very well, like Arnold Brent on the top right. As you can see, his lens cap is still on. But people love to come to NASA. It was a big thing. You know, you got to go where the space shuttle was. It's a big, epic area that nobody ever gets to go to, but Lou could take people there. We could check nests, we could catch gators, we could do what we wanted when we wanted to, and he also brought international people. We had Hannes Botha and Jan, who were actually learning how to catch gators and how to process samples. This is at NASA before we went to Africa, so they knew what was going on. And then he also brought Tai Sinaguchi from Japan. And Tai is cool, man. He's great. He always has his camera with him. One time, Lou didn't have his camera, and Tai got the best picture of a bobcat you've ever seen. It was hilarious. So anyway, Tai got us to Japan. We went there, and we saw the biggest Buddha in the world. As you can see, Lou like that. He's having a good time. And then we also went to Africa. Now, Africa was a big ordeal. We had to take a lot of gear. It's gear intensive. I was kind of the gear guy, and Lou knew that when I got all the gear there, we'd go out and catch our crocs and get the job done. So we went out on a kind of a front venue where we went to go see if the crocs were there and what we could do with Jan and Hannes. And, well, we couldn't catch crocs yet, so we had to have a little fun. So then we went back, we brought Matt, we brought all the gear, and assembled all the cages for these crocs. And catching crocs in Africa takes a lot of diligence and a lot of work, because you have to take these cages and hike them. You have to hike these cages for miles. And as you can see, we have 3 cages set up. You can see the distance from the focal length. There's 3 cages that are in the water. There's another one we're assembling. Then you have to put all the bait and everything else into these things. Sometimes you have to hike them way down these hills and up the hills. And there's a cage. There's a cage down the hill on the left. And that was to catch crocs. So we'd actively catch crocs with rods and reels, and we'd also catch them with cages. These guys are pretty elusive, especially in the daytime. But when we did catch them, it was kind of a great thing. I mean, it was a lot of work. Lou is, of course, showing us how to get blood samples out of crocs, and then we set them up on the bank. And everybody worked them up. As you can see, we have a team on the right working these crocs up as fast as we can to get all the information and things taken care of. One night we went out, we caught 5 crocs at one time on the bank, and one of the gentlemen in the picture is basically wanted to go catch another one, and he said, Russ, you see any more crocs? And there was a big one right in front of me. And I said, no, there isn't more. Just so that you could just do our job. Anyway, so we taught the guys how to catch crocs and take urine samples with a catheter. And the guys on the right, where Jan is holding the catheter, we captured about a 3-foot animal in the U.S. and flipped it over and catheterized the animal. It took us about 30 seconds. Well, they thought that you did that with every animal. So when we got there, Lou and I caught, you can see my face. Lou and I caught this animal and said, okay, you guys get the urine out. And then we turn around and these guys are picking this 8-foot croc up, trying to get a urine sample, when normally you just pick up their tail and you just get the urine sample. They thought that that's how you did it. So Lou and I were just, it was pretty funny. And then we have the bad people. You always have to have a couple of the bad kids and family members. Hannes was driving the car and the cop pulled us over and he was trying to write us up for a ticket for the rental car we had. And he finally got out of that because I thought we might be going to jail. So we have the dream team here, as these guys talked about. It's kind of the dream team in South Africa. And then during all these procedures, you always have to have helpers. Matt was always a big helper for all of us when we were in Africa. He's always helping his dad and me. Because we were the guys that went out at night, and then Lou and Hannes would do the samples in the daytime. And also, what you have to realize is when we go on these Africa trips, we have very limited resources. Some of these boats are very small. This boat we took 7 people on this boat to catch crocs. And not only do we catch crocs, but if you look at all the trees around us, it's very tight. So when we catch the crocs, we always have some family members that are kind of lazy in the back, as you can see, because everybody always works hard, and Lou always took his turn every once in a while too. So it wasn't just a couple of the other people. As I said, it was tight, but we all kept working hard. It was a lot of work. It was a lot of time. You know those glorious moments when you finally do catch a croc, you can sit back, relax, take some photos, and then you release them, and Lou always took part in that. It was a great time. And that was one of our biggest crocs we caught. It was 16 foot 2 inches. That took us probably 9 hours to process. It was a monster. And then Lou's favorite time was always letting the crocs go and getting cool photos. Meanwhile, back in the lab, we set these labs up anywhere. I mean, that's what was cool, is we made do with what we had. So this lab was set up in the middle of nowhere. So they process samples at night. We'd actually go catch crocs at night, catch crocs in the day. We were working probably 18-hour days. But when it was all done, we all finally got to relax, and we go out down to Cape Town, and we'd enjoy some shark diving. Now, when our preliminary runs one time, we went down, and Lou was taking pictures of this hippo, and this hippo pulled this move about 4 times, and Hannes said, Lou, you take one more picture, and he's coming on the bank. And that's when Lou packed up his camera and walked off. So anyway, Lou was always an educator. He always came through with a very good message. It was always a take-home message. There was never just a message, it was a take-home message. That's how he gave it. My take-home message today is this. And he'd get right to the point. But he always learned from people and also taught people at the same time. It was a very instrumental language that Lou spoke, and he could teach anybody anything. I was the smartest man in the world for probably 5 minutes of my life every time I saw Lou, and then I was the dumbest guy 10 minutes later because I'd always forget it. But he'd always teach it in a way where you understood it, and you were like, God, I'm so smart. 10 minutes later, oh, it's done. And anyway, he always took great photos. This is when we were at the hatchery. There's always people watching him take photos. Sometimes he had best friends watching him. Some of the photos, some of these are a mix of mine and Lou's photos. I just threw some together because it was just amazing what he did. And then we also got to go to Belize. And there's the house that Thomas showed. That's where we stayed. That was our hut. We went to Tenerife and Lamini. It was great times. Thomas was instrumental in organizing all of that, those trips, the CITES permits, all the background information that happens. It's amazing the amount of people that it takes to get this stuff done, especially international. So there was, again, always teaching, and everybody's always enamored because he goes to what your venue is. If you have an interest in some certain thing, he'll know it. He'll say, oh, here's how you do that. teaching us in the car. Of course, the other students in the background think they know everything in the back of the car. And then here is where we wake up every day. That's Thomas getting his teeth brushed, of course. Then there's Lou. You'd always catch him in the morning, getting the morning sunrise photos. And we go out and we work for all the crocodile nests along all the atolls. We go to each island and just run the islands to try to find these nests. Once they're found, we document what was there, what was happening, and then at night we go out and do active captures. We jump off the boat, go snare the crocodiles. So we did a lot of work in Belize also. We hiked a lot of trails, saw a lot of cool stuff, saw a lot of neat people. Of course, these guys run around and pick up the dogs as they're running around doing their work. And then we ate a lot of food. And within eating the food, there's Lou. He's, the teacher. He's talking more, and I don't know if Thomas didn't finish the story, but this lady and her daughters actually followed us to the next island. They actually bought a different ticket, bought another flight, and flew to the next place to go with us on our next adventure. That's how enticing that Lou was. Not only do you have to have food, but you have to have Marie Sharps to go with it. So anyway, we finally caught Cotton, the super duper croc, but we enjoyed our time there, and Lou left us with a lot of cool stuff. So, I have to give a data slide since I'm here. What the heck? And in that time frame, we caught 2.5 miles of alligators. If you stack them nose tip to tail, and drove 2.5 miles, that's how many gators we caught. Not only that, we did 19 VHF transmitters, 10 satellite tags. We were instrumental in putting thermistors in 91 nests. We actually hatched out 96 nests with 3,000 animals with a 75% hatch rate. So we did a lot of work. It was an amazing time. There was a lot of people involved with all these projects. I can't even imagine how many people Lou had to correspond with all the time. But yeah, time for you. We had at least 21 publications. We did 4 international symposiums, and 8 graduate students came out of this program. But just so you know, see that waiting on the raindrop? That was Kinkajou, and Thomas actually got bit by that thing. And so when he got back, he had to get rabies shots. And everywhere we were in Belize, if you look at the top left, it says waiting on the raindrop. That was for his son. And finally he had his son. That's who that was about. And there was also, when Thomas and I were talking one night on the right, this bat came flying down. Thomas said, "What was that?" I go, I don't know. And he pulled up the sheet, and there's a bat in the sheet. But it was some great times. There's more. There's some great times. We accomplished a lot of stuff. See, there's a lot of slides that I didn't show because I went through. If I didn't go through 10,000 pictures just to put this together, I didn't go through one. It was just an amazing time with a lot of cool people that I consider family. Anyway, that's it.
So we're really privileged this afternoon to have a presentation by Charles Tyler from the University of Exeter in the UK. So Charles is an internationally leading environmental biologist and ecotoxicologist who focuses on the mechanism of endocrine disrupting chemicals in fish and all kinds of different wildlife. And his work many years ago with colleagues at Brunel University was really the first research to show that effluents from wastewater treatment cause feminization of male fish. And that got a lot of press when that came out. He's been awarded numerous international prizes, including the Founders Award, which is the highest award offered by CTEC. And he's going to tell us a little bit about his interactions with Lou
and how they influence the work that he's going to present. And the work he's presenting is called Endocrine Disruption in Fish, From Mechanisms to Population, a Journey of Discovery Over 3 Decades. Welcome, Charles.
Thank you very much to Jerry, especially in terms of for inviting me to be here today. Basically, it's a real honour and a privilege to be here. And there's very few people I've travelled half around the world just to come and talk about their career, basically, and passage of time and the wonderful science that they did. But Lou would be one of those, basically. He was a fantastic chap. I feel a little bit of like a fish out of water here, basically, because we've heard some fantastic talks and stories in relation to Lou and your interactions with Lou and the fantastic field work that was done. And my journey, I've never did any science with Lou, basically. He was my best friend in science, but I never did any science with him. We had lots of discussions about science, and we shared each other's ideas and supported one another in terms of job applications and positions, but we never actually did any science together. And I'm going to tell you my story, I suppose, in terms of into endocrine disruption in the last 3.5 decades, but as Jerry says, I'm going to talk a little bit about Lou, of course, as well. So to some extent, Lou and I have to say, and people have said this, a bit kindred spirits, because I'm absolutely a nature nerd. I'm all about wildlife and wildlife sort of protection, and everything I do is sort of focused towards that. And I actually started, as Lou did, as a reproductive biologist, physiologist, and then quite early on we moved into sort of endocrine disruption as it started to emerge in the UK. And then, similar to Lou, we then tried to get on and try to understand in terms of what it meant for fish in terms of populations. And then the last sort of decade and a half, I suppose, I've been really focused on sort of mechanisms, particularly on the molecular mechanisms of these sort of compounds. And that's the sort of story I'm going to walk through today. But as I say, I'm going to start off by just giving a little personal tribute to Lou first. So it was, untimely passing, of course, of Lou so early on. I'd actually just been out to visit him and went out on one of his trips in 2015, literally a month before he passed away. It was a very, very sad time, and actually I really couldn't believe it when I heard the news. It was very difficult writing to help him to write some of these obituaries about him, basically, because he was, a huge figure. He wasn't that, when he stood next to me, he wasn't a very big man, he was quite a short man, but he was a big, big man in terms of his persona, in terms of what he did and what he put out there, basically. And my major connect with him sort of scientifically was that his good friend Tai Gucci and I invited him to be an international advisor to a committee that Lou and I, Tai and I oversee between the Ministry of Environment in Japan and our UK government for facilitating research and policy development around endocrines and endocrine disruption. It's a meeting that it's a system that's been running now for 26 years, and we meet annually either in the UK or in Japan. And Lou would come to these events, and he always had a big grin, of course, big smiling face, basically. He'd always ask those pertinent questions, as he always did, and he was critical, which was great, basically. When the science wasn't up to standard, he would make sure people knew it wasn't up to standard. But what he was a fantastic advocate, as many people have already said, really, is in terms of supporting the ECRs, the early career researchers, basically. He was always there talking to them. He often wasn't talking to the big fish or the big people, he was talking to the students, basically, and I always admired him and recognised him for that. Tai asked me to put a couple of slides in when I was coming over, but of course he's given his presentation. He gave a wonderful presentation. But as people know here, basically, Lou and Tai were great friends. And I think that Tai benefited hugely from Lou in terms of getting the bigger picture in terms of the environment, how wildlife systems work, how ecology works. And of course, Lou got the major benefit in terms of really being able to get into molecular biology and have probably the most efficient, effective labs in the world in relation to molecular biology, certainly in the environment side, basically in Tai's lab, basically. And it was a wonderful marriage, basically, wonderful partnership. And of course, it was hugely productive. I think about 50 papers or something like that between them. And of course, you couldn't interact with Lou and not go on one or two of his field trips. And it really was an absolute highlight for me, really, going on his field trips. And I did down in Florida, and also when he was down in South Carolina, egg collecting, basically, for those wonderful sort of painting egg studies that he did, and of course, catching alligators too, basically. And I have to say, though, in terms of, I was a little bit disappointed when I came back from South Carolina last time, because I could see people smiling now. The alligators we were catching were pretty small, and then Ty would send me pictures of these huge things that they'd been catching, basically. So a little bit one-upmanship between Ty and I, but it was a wonderful time. But to some extent, that where Lou and I sort of linked most, without doubt, was our love and passion for wildlife and wildlife photography. I've been a keen wildlife photographer for 40 years, and so whenever I came down here or we went trips away together with Lou, it was always, can we find time to get out and do a little bit of wildlife photography? And of course, here in Florida, you have some pretty wonderful wildlife and some pretty amazing sort of lighting conditions as well for great photography. But one of the most wonderful trips I ever did was when Lou and I were down in Australia, went down to give keynotes in Melbourne, basically, and the organiser there very kindly organised for Lou and I just to spend 3 days together on Kangaroo Island. We just wandered around the island with our cameras, photographing all the fantastic wildlife. And as has been mentioned, Lou at heart really was a physiologist and an evolutionary biologist, so he loved being immersed in these sort of evolutionary bottlenecks, basically, the marsupials and the things like the echidnas, these monotremes, these egg-laying vertebrates, and he would chat and talk about them all the time. He was so enthused about being in that sort of environment. We had a fantastic time down there. And it was really at this time I sort of recognised really in terms of, that, some of the meds really were taking a bit of a toll on Lou. In the afternoon he would always have to have a little bit of a kick like this, basically. But he would get up and he would be re-energised after that and he'd be ready to go again. But he always had his camera next to him in case something popped up. And my family loved him. They loved when he came to stay, basically. And everyone's showing their family, so I thought I'd show mine, basically. And this is my daughter, that one on the far left at the top. That's my older daughter. That's my son at the back right, and my younger daughter second from the right, basically. But Georgina, one on the left, basically, particularly like Lou, and they got on super well. And Lou was super talented at many things, photography, science, communicator, but he wasn't the best artist in the world. And that's an original of an alligator that he drew for my Georgina, basically. And she still treasures that. So if you haven't gathered by now, I'm an absolute nature nerd, and all the science I do, all the science that my team does, is about trying to understand how organisms work, how they fit into the environment, basically, and how the pressures we place on them affect their physiology, affect their behaviour. And the whole ethos of this is trying to get the information required to help better protect organisms in the natural world. And of course, when Lou came down here to South Carolina, basically, went to South Carolina, basically, he expanded very much into the human health side, basically. He was one of these big picture people. And I think he recognised early on, before many others really, in terms of if you want to really protect wildlife and get it really into the public domain, you need to have the human angle, the human element in there as well. And nowadays, everyone's talking about One Health, basically, but Lou was doing this 20 years ago, One Health. I don't need to preach to this audience, basically, but we absolutely have to wake up to planetary health in terms of we really are facing a biodiversity crisis. If we look at the rate of decline of biodiversity, in freshwater systems, populations have declined over 80% in the last 30 to 40 years. That's twice the rate of the other two sort of environmental compartments, the land-based systems and marine-based systems. And there's no doubt that chemical pollution is a contributing factor to many of those declines, basically. And if we're not convinced in terms of we're changing the chemical milieu across our planet, you just need to look at satellite images. Satellite images will tell you in terms of the concentration of nitrogen dioxide around urban conurbations. These are above levels which we know are causing harm to human health. You can look at ammonia concentration across large agricultural and areas where there's a lot of burning, basically. Soil concentrations are causing harm to the soil. And then, of course, we all know about ocean acidification, low pH now driving some species which are mobile away from their resident homes and actually causing localized extinction for some which aren't able to move. And so we know that we're changing sort of the chemical balance on the planet to harm. But equally, as Lou always used to say, really, in terms of wildlife are sentinels for our own health, basically. And you can look at this, and this is a really good paper that's published almost 8 years ago now in the Lancet by Landry and Batal, basically, and it just gives you some stats and data in relation to the impacts of pollution on human health, basically. And it's more than many of the most notorious diseases. It causes huge numbers of premature deaths. It's not equally dispersed in the sense that clearly low and middle income countries are particularly impacted by it, and it costs hugely to these global economies in terms of pollution on human health, basically. And of course, we all know that we're dependent on chemicals, and we're dependent on a whole variety of different chemicals, everything from plasticizers, fertilizers, and pharmaceuticals. And most of these end up, as we all know, in the aquatic environment. Some of these are harmful, and others we don't know whether they're harmful, but the increasing numbers are suspected to be harmful. And these, of course, include the endocrine disrupting chemicals. And amongst these, and it's certainly come to the fore in the last sort of decade, I suppose, really, are things like pharmaceuticals, be they human pharmaceuticals or veterinary pharmaceuticals, basically, because, of course, these are designed specifically to alter physiological processes in our bodies, and those targets generally be highly conserved across vertebrate phyla, basically. So my journey in relation to sort of endocrine disruption started around the same time, really, as Lou's, in the sense that I went to work with John Sumter at Brunel in 1988, 89, and it was on a project, it was on a contract from a government body, because there'd been some observations that some fish were looking a bit fishy in some of our rivers, in the sense that a few fish had been found which had male fish which had sort of feminized characteristics, and the thought was that it was due to sort of pollution. And so this first study set out to investigate that, and fish were placed in cages basically downstream from or in effluents from wastewater treatment works. I think about 28 wastewater treatment works, and my job was to measure the vitellogenin production, that yolk protein that's already been mentioned, basically. And it really was a bit of a eureka moment, in the sense that after 2 to 3 weeks of exposure, up to half the blood protein in those fish was vitellogenin. So it was up to 1 million-fold induction. And that showed that for the first time, the effluents from sewage treatment works were estrogenic to fish. And then some of you will know people like Susie Jobling and Jewel Harries came and did some follow-up studies from this work, placing fish in cages downstream of these discharges. And in some rivers, up to 5-6 kilometers downstream, these fish were still responding to whatever those estrogenic chemicals were within the water system, producing this sort of yolk protein. And then the next sort of chapter, really, after identifying this estrogenic nature, if you like, within these effluent discharges, was saying, does it really matter? Water companies are saying, well, so what? These male fish can produce a little bit of this yolk protein. Does it really matter? And so the next charge for us was say, well, are wild fish being affected? And we studied this particular species. It's called the roach. It's a member of the carp family, basically. It's found very commonly in lowland rivers in England and across Europe as well. And what we did here was not just measure the production of the yolk protein, but also we measured the formation of the ovarian cavity in males, which is estrogen dependent, and also the presence of oocytes within the testes, just as Lou did in relation to his work on alligators. And I think we sampled about 1,500 or 1,600 fish, so it was a pretty big study. And at 44 of the 51 sites studied, basically, there was intersex, and so there were oocytes within the testes of those fish. About a quarter of all the males that we looked at across that study were feminized to a greater or lesser extent. And then I had a whole series of poor PhD students, grad students basically, who then did a whole series of exposure studies on effluents at various water companies, and up to 3-year exposures. And those exposures established that we could induce all the feminized phenotypes that we saw in the wild fish. So we could induce clearly the production of the yolk protein, we could induce the ovarian cavity, and we could induce the intersex as well. So it was clearly that, it was, without question, it was the effluent from wastewater treatment works which were driving these feminized responses in fish in British rivers. And I put in, sorry, it's a bit data heavy, but I put this slide up because Lou always used to say, let the science do the talking. And about 15 years ago, 15 years ago, there was quite a lot of press saying that rivers are much better in England now, the situation's improved, there isn't intersex, etc. So we thought, well, we better go and have a look at this and actually get the data and have a look at that. And so we did a much smaller scale study of just 8 rivers and 9 sites, basically. But actually the situation hadn't changed much at all. At some sites it improved a little bit, and at other sites it wasn't much different at all. And in the interim, what we'd done is we'd done rad-seq to develop a sex probe. So now we could sex the individual fish as well, genetically, basically. And what we found is not only was the position in terms of feminization hadn't changed much, but when some of these fish were actually completely sex reversed. So some of the females actually were, in fact, genetic males, basically. So added a bit of a further dimension to the situation and the position in terms of feminization of roach. So feminization of roach in English rivers is still widespread, whatever the press say. The next sort of part in the sort of the chapter, I suppose, really, it was the whodunit, you know, trying to identify. And I know Lou struggled with this to some extent in terms of in Lake Poplar, really trying to drill down and find exactly what chemicals were driving those feminized responses in the alligators. And I suppose we had a head start to some extent in the sense that because that yolk protein was being produced, then it was a very high likelihood it was an estrogen or an estrogen-like compound. So we went to look for estrogens. And some groups were looking in the water. Our approach, working with a really good chemist, and if you're an ecotoxicologist, I think you can't be a good ecotoxicologist without working with a really good chemist, Liz Hill at the University of Sussex. And what we did is we exposed our roach to the effluent, and then we measured what got into the fish. We used various tissues, but bile's a super a target for that, because about 70-80% of all chemicals go through the bile before they're excreted. And she fractionated out this bile by HPLC, then used this clever recombinant yeast which has a little estrogen receptor incorporated into it, changes a chromogenic substance to a yellow to a red when it's an estrogen or estrogen-like compound, basically. And then when those estrogenic fractions were identified, she then went on to use things like LC, MS, NMR to identify the specific estrogens. And that bottom right figure is it looks like the Manhattan skyline, doesn't it? In the sense that there's all sorts of estrogens. These fish are just sucking up all these different compounds into the body. But particularly there were steroid estrogens, estrone, estradiol, deriving from the human population. Ethinyl estradiol is a component of the contraceptive pill. Dihydroequilenin, which I always find an amazing one. This is an estrogen that's purified from the urine of pregnant mares. It's put into hormone replacement therapy, given to people during the menopause, and then it will find its way back into the water and back into the fish. Incredible cycle, but there we are. And then other things like non-alphenol, polyethoxylates, and some phthalates as well. But the drivers in terms of the strongest estrogenic fractions here were the steroid and steroidal components. And so again, another series of poor PhD students doing controlled exposures now in the laboratory with our roach. Up to 3 years, we could show that for ethinyl estradiol, which is exquisitely potent as an estrogen, basically, they could induce all of feminized phenotypes at environmentally relevant concentrations. And that was the thing that Lou was always saying and got so frustrated about. There's lots of toxicology studies out there, especially during that early time in terms of endocrine disruption, where there were lots of exposures to excessively high compounds, basically, inducing changes in terms of phenotype, but translating that to what that really meant for sort of wildlife populations was always a big question mark. So here we're showing that this compound at environmentally relevant concentrations can actually induce all of these feminized sort of phenotypes. And of course, the press, again, the water companies and the government came back and said, well, so what? As they already, so what? So you've got a few feminized fish in the rivers, basically. You may have quite a few feminized fish. Does it really matter? So the next chapter for us was then to try and find out, well, can these fish breed? Can these feminized males actually breed? And again, to do this, we really had to come back into the laboratory. And what we did here was we set up a breeding station, basically, a series of large tanks. And we collected fish from the wild on populations where we know that there were some intersex fish. And we put in what we think to be 3 females and 6 males, some of which would have been feminized to a greater extent, and allowed them to breed. And then what we did, when a postdoc in my lab did, he developed a series of DNA microsatellites, and so we could identify, we could genotype the mums and the dads, and we could genotype the offspring, so we could find out how successful those individual fish in those tanks were in terms of siring offspring. And the bottom graph shows reproductive success at the y-axis and the intersex index. That's just the degree and feminization of those male fish, because we were able to identify that when we killed the fish, the parent fish at the end of the study. And you can clearly see with the increasing degree of feminization, there is a reduced reproductive success. So for the individual males, the individual fish, it does matter in terms of being feminized, in terms of your reproductive capability, your ability to pass on your genes. And there are all sorts of studies which clearly have emerged over time now to show that variety, not just estrogens, or a whole variety of endocrine disrupting chemicals, Lou's work, of course, but many others in terms of that lots of wildlife at the level of individuals are being affected adversely in terms of their ability to develop, grow, and or reproduce. And so there's no doubt that lots of wildlife is being impacted at the individual level by these endocrine disrupting chemicals. The biggest challenge that I think that Lou faced in terms of work in alligators, and it's certainly the biggest challenge that we faced in terms of our work on wild fish, was to illustrate where there's a population level consequence. Rightly or wrongly, we protect at the level of the population for wildlife, whereas clearly for human health we protect at the level of the individual. So trying to identify whether it's a population level sort of impact is a really challenging thing to do. And one attempt that we did to do this, and this was done some time ago now, was to again use our microsatellites to see whether the effective breeding population size had changed. So you can imagine if there's lots of male fish which have a reduced reproductive capability, if you look at that population genetically, you would imagine that actually the offspring are derived from fewer individuals and fewer males. So you can derive something called NE, that's the effective breeding population size, out the y-axis across the x-axis, which is the amount of estrogen in the river study sites. This has done a whole series of different rivers, and you can see there's more than a tentative trend there in relation to, certainly once you get at the higher estrogen concentrations, then the effective breeding population is relatively low, but there's lots of variation in the data, and actually when you analyse that data statistically, basically it's not significant. So we failed to some extent, I think, really, in terms of being able to show a population level effect. And we've done all sorts of other genetic studies, doing things like genome-wide SNP analysis now to look at estrogen-responsive genes to establish in terms of whether there's difference in terms of different SNPs basically in the estrogenic populate the polluted rivers compared to non-polluted rivers. And there are, but nothing that we can really pin down to say, well, that's really a population level problem. But of course we do know that there are associations between specific chemicals, some of which are endocrine disrupting, and population level impacts. Just for a few species, very, very few. The classic ones include, of course, your Florida panther down here, although there's a big issues there, of course, in terms of it's a pretty small, limited gene pool, basically. The orca population is an interesting one, that was mentioned earlier. The orca populations on the west coast of Scotland now are so polluted with PCBs that they can no longer reproduce. So that population will now go to extinction. And then, of course, probably everyone knows about the white-backed vulture and the slender-billed vulture populations in Asia is exposed to diclofenac, basically causing localized extinction. So we do have examples of some chemicals, some of which are endocrine disrupting and population level sort of impacts. And that's really where I finish in terms of our studies on the sort of wild populations in relation to endocrine disruption. But the next sort of chapter, I suppose, in my journey has been really in terms of trying to better understand the mechanisms, the way in which these chemicals drive and cause these effects in wildlife. And estrogens, which have been our primary focus, if you look in mammalian literature, they do all sorts of wonderful things. They're not just involved in terms of growth and development, of course, they're involved in things like calcium homeostasis, defense, the immune system, and it's incredibly complex in terms of that signaling pathway. In mammals, of course, you have 2 nuclear receptors and 2 membrane-bound receptors. In fish, you have 3. In different fish species, you have different splice variants. So it's super complicated in terms of how those estrogen signaling sort of works. And of course, there's lots of crosstalk as well between things like the thyroid system as well. And it's probably the case that every cell in the body expresses an estrogen receptor at some time during its ontogeny. So estrogens are fundamentally important in relation to all sorts of processes, not just reproduction. And I wanted to try and get a handle on whether fish, our roach and other species of fish, were being affected by the estrogens beyond sort of reproduction. And the way we did this, so about doing this, was I tuned up with very good molecular biology techs who could, and we developed a very sensitive transgenic fish. It was a double construct for anyone into the molecular side of things. Estrogen, 3 estrogen response elements linked to a GAL4, and the second construct is a US with it linked to a green fluorescent protein. And basically what happens here is you expose the fish to an estrogen, it triggers the cascade response, and then you get the fish to glow. It glows green in the sites where that estrogen receptor has been activated. And so then you can look at things like the relative tissue responses, you can look at the autogenous responses, you can look at the effective concentrations to drive those responses. So we have a sort of a wonderful sort of biomonitoring system now to be able to look into the bodies of our exposed fish to be able to say, well, where are these estrogens interacting? And we've used this estrogen transgenic now to develop all sorts of body profiling, so all sorts of different types of estrogens, steroid estrogens, alkylphenolic compounds, phytoestrogens, and of course all the bisphenols. We did a lot of screening of bisphenols probably about 20 years ago. Bisphenol A, F, AF, S, all of them, and a lot of them estrogenic, weakly estrogenic, but estrogenic. But the power, I think, of some of these transgenic systems enable you to real drill down to find out, because in some of the body blueprints, what you find is that you get differences with different types of bisphenols or different types of environmental estrogens. And for bisphenol A, the heart kept coming up. And we thought, well, this is an interesting one. So I've got a PhD student to start working on this. And when he looked a bit more closely, it wasn't the myocardium that was responding to the bisphenol A, but it was the heart valves. So you can see the heart valves being highlighted there. And when you look at the effective concentration of drivers for bisphenol A, you can see at the pretty high concentrations, well, 100 micrograms per litre is far higher than you find in the environment. But once you start to look at the metabolite, MBP, now look at the concentration. Now you're a part of a microgram per litre, which are inducing a clear and strong response in those heart leaflets, basically. So clearly it's a nice illustration in terms of how that transgenic enables us to really drill down and find out what was going on with that particular target. And you could do all sorts of wonderful things in terms of identifying the signaling pathways in which way that bisphenol is being mediated. And you would do CRISPR-Cas now, of course, but this is a while back when we did morpholinos to knock out the different estrogen receptor subtypes to be able to show that the signaling was working through estrogen receptor sort of alpha. Then another PhD student then went on and then looked to see how the heart valves developed over time and found that the leaflets didn't develop as well, and did all sorts of challenge studies with those fish to show they couldn't perform as well in terms of swimming activity. So it's a really nice example, I think, in terms of where a transgenic really allows you to establish an adverse outcome pathway in terms of from the primary initiating event right the way through to inhibited performance in terms of swimming capability. But the wider landscape, beyond the estrogens, we all know, and we've seen some presentation now in terms of it's not about individual compounds so much, it's about mixtures. It really is. And if you're an alligator living in a popka, you're not too worried about one or two individual compounds. You're worried about the whole mixture that you're facing, basically. And as the area has grown over the last few decades, 3 decades, basically, that plethora has increased as more people have come to the fore to research it. There's been more screening going on and testing going on. And now, of course, we have thyroid hormone mimics and disruptors, we have androgen mimics and disruptors, we have glucocorticoid mimics and disruptors as well. And so we have this huge complex array of chemicals which are getting in the bodies of our wildlife, into ourselves as well, and interacting and interfering with all sorts of signaling pathways to affect these processes of growth, development, and or reproduction. And just as you can look into the body of an alligator, as Lou did, or look into the body of a fish, look into some of those tissues, and they're a blueprint for what we're putting out into the environment. I mean, and some of these chemicals are in target sites that really matter. I mean, we often look at the liver, but we accept that really the liver is a processing station for detoxification. But once you start to see these compounds concentrating in areas like the brain, for example, then that really gives us, an area for concern for sure. And again, as Lou would always say, we're not much different than wildlife. You can look at in terms of concentrations of some of these compounds in the cord blood, for example, in terms of the most vulnerable life stage for human development, and find lots of these compounds accumulating within our body. So it really is about mixtures. And I'll just skip this slide, but I just wanted to say in terms of when people are talking about mixtures, we tend to generally focus about the mixtures here and now. And certainly when we've done studies with our fish, we put estrogens together with anti-androgens, we can increase response. We can get an additive, maybe a bit more than additive response in terms of driving some of these feminized responses. But sometimes I think what's overlooked is in terms of the sequential exposure and in terms of exposure over time. What's your history? What were you exposed to before? How does that affect you? How you respond subsequently to a further challenge? And so this is something I wanted to try and look at with these transgenic fish. And so what we did with that estrogen transgenic is we took out the green fluorescent protein. We put in something called kaida, and kaida is Japanese basically for maple, and it's also green in terms of it fluoresces green, but if you expose it to UV light, it goes red and stays red. So now we have a model whereby we expose our fish to an estrogen, we measure the response through imaging, we then expose the fish very briefly to UV light, it fixes that green now red, and now we can do another challenge and we can measure the green response again, and we can see how that first exposure affects the responsiveness to that second challenge. And I won't go into much of the data here, but you can see in terms of these combinations of ethanol estradiol with genistein, with bisphenol A, in different combinations, you get an potentiation of an effect, whatever combination of these estrogens you use. And for my mind, it's a big gap, I think, in terms of thinking about risk and risk analysis and risk assessment. We talk about dose-response concentrations, but in fact, we talk about dose-response concentrations for animals we maintain in the lab. What does it mean for your alligators out there, which have been exposed for 20 years to mixtures of these compounds over time, basically, when they have that sort of re-challenge down the line? So, again, I think it's an area that needs much more focus and much more attention in terms of the research area generally. And the last thing I wanted to sort of come to in relation to these transgenics is that estrogens are also fundamentally important for brain development. In fact, if you look into the brain, there are more estrogen-responsive genes in the brain than any other tissue in the body. And this is some work recently done by one of my postdocs. She identified a brand new cell type. She's called the estrogen receptive olfactory bulb. And you can see it's the first responding cells in the brain of the zebrafish. You can see them just glowing up there now, basically. And they're the first responding cells. And if you interfere with that signaling process in terms of that estrogen signaling process, that alters the ability of that fish to then smell down the line. So in fact, you're affecting the sensory system. So by exposing it or disrupting that estrogenic signaling pathway during early life, you're affecting that neural development process that relates to olfaction. And this is super, super interesting to us, I think, because a lot of the some of the conditions like autism, for example, in people are related to they have problems with smell and smell development. It relates to sort of olfactory development. So there's something maybe probably interesting in there to sort of like to have a look at in relation to sort of the human world. That's for sure. And the last slide I'm going to put up is I just wanted to show, as you can see, I'm advocating the use and application of transgenics because I don't think enough people are using them in terms of the zebrafish systems because they're incredibly powerful now. And so this is a transgenic developed in the US. And the image you're seeing now is a video of a transgenic zebrafish. It's got a green fluorescent protein, and it basically, when extracellular calcium is produced, when that neuron fires, it glows green, basically. And it's linked to every neuron. So you can look at every neuron firing in the brain in real time. This is using light sheet microscopy. And you can do it in 4D because you can capture the whole brain of the zebrafish, but you can also do it over time as well. So you can look at the ontogeny of the response. So this is a zebrafish at rest, basically. So you can just see a little bit of activity going on here, basically. It's pretty chilled out. It's pretty happy. Now, actually, this is being exposed to a seizurogenic compound. We do some work with drug companies with these. And now you can see the firing pattern here. So you can build out that profile in terms of how that zebrafish is responding to whatever chemical. It might be an endocrine disrupting chemical, neuroactive chemical. And then what you can also do, you can build up these connectograms. So you can build up in terms of where does the signaling start, how does it link to other regions of the brain, and how that develops over time. And so that's a lot of the work that we're doing now, and actually quite a lot of work on some of the neuroactive endocrine compounds. And then, of course, what we're trying to do is relate these to sort of behavioural phenotypes, like many people have shown today in terms of these behavioural assays, basically, but really trying to, as Lou would always do, basically, try to sort of build in the mechanistic understanding to how a phenotype comes about. And I'm just trying to advocate now that, very different now to 30 years ago in terms of the tools and the systems that you have available now to be able to doing real-time monitoring of chemical impacts, and in terms of linking the mechanisms with the phenotype. We're really in a totally different era now. And it's just to remind us all, I suppose, really, and again, I'm preaching converted here, we tend to work in silos too much, I think, really, and we talk about endocrine disruption, but endocrine disruption isn't just about disruption of the hormone systems, basically, because if you're messing with the hormones, you're likely messing with the immune system, and you're certainly messing with the neural development as well. So, and a lot of people talk about now that new trends are systems biology. Lou was a systems biologist 30 years ago, 35 years ago. If you study whole organisms, you're a system biologist, aren't you? I would think so, anyway. So, yeah, and I won't dwell on these things, but just a few take-homes, I suppose, really. As Lou has always said, what's the take-home? I said, well, here they are, Lou. Here's the take-homes. And I suppose that what we need to understand more is some of the work that you guys have been doing, and that is really about more studying wild populations. There's not enough people out there studying wildlife populations. They're the sentinels for the environment health and to our own health as well. It's difficult to do. You've seen it in terms of with the alligator work. It's not easy getting a single alligator, but it's fundamentally important. If we can really understand the health of the environment out there, we can't just do it in a lab. We have to be out there working on natural populations. And certainly in terms of, I think, some of the technologies we have now in terms of imaging, molecular technologies, transgenic systems, they really help us understand the mechanisms, which we couldn't do 20, 30 years ago. And who scored? And I think everyone will agree here, and Lou was, you know, a big player in this, is that as many good researchers in the US have been, is that endocrine disruptions change the way we think about chemicals in the environment. It's really changed the paradigm, I think, in terms of thinking about sort of toxicity, much towards the concern about more subtle effects that these compounds can have, but nevertheless the important effects that they can have over time. And as Lou would always say, alligators aren't any different than humans, and I'd say fish aren't much different than humans. It is screwing up the systems in wildlife that we're screwing it up for us as well. And that's where I'm going to finish, just to acknowledge my team members. They work on all sorts of conservation projects and aquaculture projects too, but they've contributed to the slides today. And the last thing I wanted to say is in terms of when Lou would come to stay with me, I live in the very southwest part of the UK, and we don't have weather like you have here in Florida, in terms of, and I live just on the edge of a national park called Dartmoor National Park. And whenever Lou came, it was raining, it was blowing a wind basically. He was always wearing his shorts, but he was always, it was raining. And he always used to joke how awful it was where I lived, in a joking manner basically. And I couldn't argue with him, because when I came to Florida, or came over this way basically, it was wonderful weather, fantastic light for photography. So I just wanted to finish off to saying, well, actually sometimes the light's quite nice on Dartmoor. And here's just a few of my pictures from my backyard basically. So if any of you want to come and visit Dartmoor, it's a wonderful place. Don't listen to what Lou might have told you in the past basically. Some pretty fabulous things to see. And that one's for Lou, because Lou, of course, always loved owls. And of course, a couple of reptiles. You couldn't dedicate something to Lou without putting a couple of reptiles in. And I came came to Florida just a couple of days early, because when I came here, Lou introduced me to Payne's Prairie, and I went back there on a sort of nostalgic trip on Thursday, I think it was, and I took my camera, of course. And there are two images I wanted to show you, because I think Lou would like these. That's Lou, that's just Lou for me. And then I was very pleased to get that one too. And he loved the osprey. And thank you very much for listening.
So I hope this goes okay. I actually practiced not crying more than I did anything else, so hopefully that works. So I was going through the Google to try and find lobsters and American alligators in like the same picture, and so obviously it didn't really exist, so I used AI to kind of generate things, and it's not ecologically accurate, but I'm not an ecologist, so I think it works actually pretty well. So my journey didn't actually start with alligators or lobsters. It started with a Siberian sturgeon at Mote Marine Lab, and that's where I first became interested in something called nitrate at the time, and how it affected these animals. And then when I was interested in doing a PhD, I had a talk with one of the faculty that were there and told them what I was interested in, and he said, you should go talk with this guy, Lou Guillette. And at the time, he hadn't published in nitrate yet, and so I often think about sort of that fated conversation, because if we hadn't had that, I may have never found Lou, but I did, and so it has made an obviously profound impact on my life. Practice. Stop. Okay. So from here, I can't express, not only was he the most amazing mentor I can imagine, and friend and everything else, but the other people that I've met on the way just have made my life such a better place and such a better thing. And these people are all still a part of my life and will continue to be so. And it's funny, when I was creating this picture, Lou often would lovingly refer to us as either the innies or outies, just sort of a joke. And the innies were sort of the people who spent the lion's share of their time in the lab, and the outies were the people who spent more the lion's share of their time sort of in, these outer places. I wasn't doing this intentionally, but this is more the innies, right? So these are sort of the lab rats through and through. Conversely, we have the outies. I'm a complete poser here, by the way. This was, I don't know if you remember, Matt, but this was the very first alligator I caught, and so totally posing there, but I'm, still an innie. And so these are some pretty amazing experiences that I have been able to sort of have along the way, and yeah, what a massive, change in my life that has been this. These are just some of the pictures. Yesterday when I talked about the flood and all, these are some of the pictures that we actually took on that trip, which is pretty amazing. I've got some other ones, but for me, going and I had seen all of these amazing pictures that he'd taken, so I was interested in taking the pictures of the person who was taking the amazing pictures, because I was like, holy cannoli, like I'm here, you know what I mean? He's taking these things, and clearly I can't take pictures of the things that he can, and so it was just really fun, and this Boggs and Lou kind of walking up, and just some really, really, really cool things. But regardless of whether you're an innie or an outie, in his lab, at some point every year, all of us had to go into the field. When I say had to, it's not like, we were all super excited to go into the field. And so I had heard from Lou and others, and I was like, okay, we get to go out on an airboat, right? And we're looking for these nests, and then we get to take the eggs, and it was just, to bring back to the lab to do all these really cool experiments, and it was amazing. It was amazing. But what he didn't tell me was, okay, so this is Lolo getting dropped off so this was the very first time I went out. And this is us getting dropped off, right, at the nest. And so what he didn't say is we are here in the middle of nowhere, what felt like a very long time, and it was a very long time. And as we got dropped off, I don't know if you can see, there's not a good picture, but there's what can only be described as like a beefy dowel at best. And so we were told by the person who dropped us off, and I think it might have been Cameron, and he dropped us off, handed the dowel and everything else, and was like, okay, if she comes back, you've got to hit her. And we're like, okay. And then he stopped before he kind of drove off and left us there. He's like, hard. You got to hit her hard. And I'm like, okay. So anyway, he goes off. Lolo and I are here for the longest time. And I was really excited beforehand because I'm thinking, I get to spend hours with Lou, right? And he didn't mention this. He didn't mention this, right? And I think I was living actually with Lou and Buzzy at the time this happened. I was like, he didn't mention this. This never came up. Because he, right, is here, right? He's up here, right? This is him, right? That's where he gets to spend the day. But regardless of the sawgrass cuts and the giant bug bites and the leeches I later pulled off my ankles, I wouldn't change it for the world, because it was such an amazing experience on so many levels. And then we got to bring everything back to the lab, and then, happy birthday, little buddy. We get to see all of these things kind of hatch and do some really cool experiments with them. And just, all around, it was just, really amazing. But I do need to say that of all of these cool research experiments and all of these cool things, and scientifically, of course, he made me what I am, but the thing that I remember most about Lou is how he treated other people and how he made people feel. That is the number one thing that I've learned, and that I do my best to try and do in my own lab and in my own interactions, because that is a thing I will never forget. And that was a gift. That was absolutely a gift. And then these are some take-home messages, from some really cool stuff that we did, that I was part of, and some alligator work. But what I want to focus on today is just one of the most recent studies that we've done with lobsters, with American lobsters. And so I went to the AI again, and I was like, okay, give me a picture that shows a lobster and an alligator, but in the context of some cool stuff that Lou has done, right? Maybe asking, sort of asking for help or whatever. And this is what it generated. And I was like, oh my God, AI thinks Lou is a superhero. That totally tracks. That totally tracks. Yeah, and so that seemed super appropriate, right? But so American lobsters in Maine, we don't have American alligators, which is kind of sad, but American lobsters in Maine are tremendously important, both economically and ecologically. It's the most valuable single species fishery in the entire United States and Canada, $725 million in landings in just Maine for 2021. But it's really, again, hugely economically, but also culturally important. I mean, for generations and generations, we have families that, fish this really cool species. The other species I study a lot are Atlantic salmon. And so how this kind of comes together is salmon in aquaculture gets something called sea lice. It's just an ectoparasitic copepod that kind of lives on the skin, but it's a problem. And often they use pharmaceuticals to treat them, just like we do dogs and cats with fleas and ticks, right? So they treat these sea lice with pharmaceuticals. In Maine and in the United States, we don't use this compound called imidacloprid. We don't use this. But they do use it in Europe. And so before they bring it, we're considering bringing it here. I'm like, we really should do some work on some non-target species to see what it could potentially do to the most important fishery, right, in the United States, just to see what this might have. But how they deal with it, because imidacloprid is nasty, and they know it's nasty, so what they do is they take this, they take all of the fish from net pens in various places, and they put them on well boats, and then they treat them in the well boat, and then they take the fish out, and then they put the water through this, this clean, it's a patented treatment system. And so they treat the water, but when they release it at a point that is no longer detectable. But the limit of detection with HPLC could be as high as 30 micrograms per liter, right? So that's per billion. That's right. That still could be relatively high. So I'm interested, okay, well, let's not think about what happens if the wellboat, something happens and they're exposed at the dosage concentrations. Let's look at what's just released from the wellboat. And so what we did was we exposed subadult female lobsters to 3 different concentrations. So we exposed them to obviously control, but then we exposed them to that sort of effluent dose. So we exposed them to 0.3 parts per billion and 30 parts per billion. So it's sort of really low concentrations, but yet, you know what I mean, this is kind of nasty stuff, right? And we exposed them for 120 minutes because those are some models that we had it could be conceivable that that would be the longest that they would conceivably be exposed in that sort of natural environment if there wasn't a lot of tidal flow and a whole bunch of other things. And then we recorded behavior, which, thank you, Scarlett. And so we recorded behavior, but then we took a whole bunch of so their blood is called hemolymph biochemistry, and we did a whole bunch of biology stuff. And a lot of the kind of cool other sexy endpoints, right, if you're a scientist. And then after we exposed them for 120 minutes, we returned them to clean water for 5 days. To just say, okay, they had that flush, right, so maybe that wellboat did that treatment, they were exposed to the effluent, but then they go back to clean water. So we exposed them to clean water for 5 days later, and then rinse and repeat. And the behaviors we looked at were different things that were involved in escape response. So they flipped their tails to try and get away from things. I think I'll show a video of that in a second. We also put them on their back and documented the amount of time it took them to flip back over. And then we looked at activity level. So they have these little things called swimmerettes, which are right up in there, and I'll show you a video of that in a second. So we looked at how much they're fanning those, how lethargic they are, and whether they had something called claw trembling. I'll show you that in a second too. And then the threat display. So anyone who's worked with lobsters understands the Merrill spread, right? So it's one of those things that they use in agonistic interactions with each other. And so it's like if they come up on another lobster and the other one is sort of posing like, oh, do you want to fight? And then they were like, I don't know if you've seen these, right? And they raise their claws and see, look how big my claws are. And it's this really kind of cool thing that they do with each other. But why we were looking at those behaviors and why those behaviors are so important is, one, they have a lot of economic importance, because when the lobstermen are out collecting the lobsters, or when they actually go back to the pounds, where they're planning on, basically not pounds, but the facilities where they will sort them, only lobsters with really high vitality are sold to the live market. The ones that are kind of lethargic and those go off to immediate leg processing. But you get a lot more money from the going to the live market, right? So if you've got lobsters that are a little bit lethargic, right, that is a huge economic problem. The other thing is, for ecological importance, these are behaviors that help with survival, right? So these are things, and I don't know if I just do a forward and see. Oh, look, okay, so watch the little lobster, boom, right? So if you do it the jump scare, so basically what it's gonna happen is it's gonna use those tail flips to get away really quickly from predators, right? And so that's why when you pick them up, they're flapping their tails, because they're like, I don't know, this is what I do to get away, even though it doesn't really work, right, in that context, but these are all really, really important that way. Right, just kidding again. Okay, now we're gonna, okay, so here, and before I do this, I'm not gonna show you all of the figures on all of the chemistry parameters that we looked at and some other things. And in other contexts, we've actually done full transcriptomic studies, and those really showed us nothing, right? So there were no differences. So I think that's why I wanna show sort of the importance of behavior, because sometimes you're looking for a needle in a haystack, and there is an effect, right? And I'll, not to give away the punchline, but you can definitely see an effect. And so what we did was, so that's me in like the Tyvek suit and all this other stuff. So I basically held them up, and I'll show you the 2 videos. And we did this for all of the animals that we did. And then we had other students that were watching the videos and doing all of the assessments blind, so that they wouldn't know which treatment was which. I don't think I can play both of these at the same time here. I could on my own computer. So what I'm gonna do is I'm gonna play the first one. What I want you to do is to pay attention to tail flips, how the little swimmerettes are fanning, and marrow spread, and just basically overall activity level. So I'm holding up so you can see marrow spread, you can see the little swimmerettes going like crazy, little legs going like crazy. We doodle the feet, just because sometimes if they're not moving, that can get them to move, right? And these are typical videos. I'm not just cherry picking the best. These are typical videos. So that's the first one. This is the 30 parts per billion imidacloprid, right? And these are typical videos. And you can see the trembling, the claw trembling, right? There's palpable differences there, right, between the two. And this is with effluent concentrations that's supposedly been, treated. And then when we look at these things, so the number of tail flips, right, is absolutely significantly different between the control and the 0.3 versus the 30. But the other crazy thing is the chronic response. So 5 days later, right, so we put them in clean water. Things are looking better, but they're still significantly different, right? So over time, that really hasn't ameliorated or hasn't kind of cleared itself. And then when we look at writing time, which I didn't show a video of this because it's really hard to do, because healthy lobsters, it was really difficult to I could almost like barely, right, get it because they were just so fast. But you could definitely see a difference here. And here, the 0.3 was even, right, significantly different, because it does take a lot of neural circuitry coordination to flip yourself over. And so I think it's kind of collecting all of that. And again, we also saw that, even though it's a little bit better story, but we also saw that, right, in 5 days later. Holy cannoli. So really here, what are the take-home messages? Thank you, AI. And so this compound, even at really low and what we're calling ecologically relevant concentrations at this point, are different, right, between the control and between the others, with behaviors that are both relevant to survival and economic vitality. And the other take-home message is all of those other kind of sexy biology endpoints that we took didn't really show us anything. But there's a difference, right? And so we need to kind of figure out what those differences are. I'd be remiss if I didn't show all my students. And so I look at this too, and I'm like, this wouldn't have happened, right? These students, I just wouldn't be here with these students, et cetera, if it weren't for Lou, right? And just like he said, I mean, these students are sort of my legacy, but his. I mean, these are sort of his academic grandchildren, right? And so it's really kind of really cool. And this actually, so this is the work that I just showed was this from one of the chapters from her dissertation. And this student just actually got a faculty job in my department. I didn't have anything to do with that, by the way, but she got a job in my department, which is really, really exciting. And then I asked AI can you just give me a closing, even though it says questions and we're not doing questions, that's fine. But I said, okay, can you just give me like an end? Like how would you end this? And apparently this just ends. So AI, I don't think had anything to do with that. We're just gonna go with it. Yeah, okay, anyway, so that's how it ended it. And I was like, oh my God, that's super cute. But anyway, that's it. Okay, that's it.
Because our next presenters are Karen Helbing at the University of Victoria and Kathy Proper at Northern Arizona, and they're gonna be talking about frogs and getting people together to do science on Zoom. Yes.
All right, well, everybody, I have to say I've been listening in to all of the talks over the last couple of days. Heard everybody get a little bit emotional, which I think both Karen and I did in putting together this talk as well. And I do really want to thank the organizers of this whole symposium. Thea Edwards was the one that let us know about it quite a while back during CTAC. And Jerry and Nancy and everybody who's been instrumental in pulling this together. It's been a joy to sit and listen. All of that, my friend Lou and my colleague. So, our talk today, of course, is about bringing people together and letting science do the talking. We've heard a lot about Lou bringing people together, but I want to start with when I first really was introduced to this guy named Lou Guillette. And aside from his family, I'm probably one of the people that got to know him very early on. And so I titled this slide how I was duped by this guy named Guillette. I was doing my undergraduate research paper for herpetology at UC Berkeley. It was a course taught by a great herpetologist, Terry Green. And he really wanted us to dig into the literature, get some data. And in my naivete as a young, I think I was a junior or senior Lincoln graduate, I went back in the literature and I started early and moved my way forward. And by the time I was about 2 days ready to turn in my paper, I came across this paper by Lou Guillette, Evolution of Bioimpurity in the Lizard Genus Scalopris, which was pretty much verbatim the title of my paper. And I was like, oh my gosh, who's this guy, Lou Guillette? Well, ultimately, I moved to Oregon State University to work with Frank Moore, who came out of Dave Morris's lab, and Dave was in the symposium yesterday. But Lou was working very closely with Dick Jones, who was also next door to Dave Morris's lab. They all worked very closely together. And so, through Oregon State and through the Western Conference for Comparative Endocrinology that happened every year, I got to meet this guy named Lou Guillette while he was at Wichita State. And so that was my first formal introduction to Lou, and I let him know how he's speaking. All right, I gotta move a little bit of this out of the way. So I like to talk about Lou as a reptilian evolution, backwards and bigger. I know when I first met Lou, he was working on those slobbering lizards. He had actually published a paper on anomalous lizards looking at the impact of this neuropeptide that people didn't know that much about. That it was related to oxytocin, which was being used to induce labor in humans. And he had injected anomalous lizards with this and found that they were inducing uterine contractions, even in anoles. And vasotocin is, of course, the evolutionary precursor to oxytocin and vasopressin. And we now know how important both of those peptides are to not only reproductive physiology, but also behavior. So Lou then moved on to Wichita State, which is where I met him, and he was again studying really the reproductive physiology of scaloperating lizards. But he then moved on to University of Florida, where I got my first opportunity to work with him, as I had moved, because of Lou, to Dick Jones's lab to work as a postdoc. And again, I have to say, earlier in the presentations yesterday, somebody mentioned how Dick was really instrumental in helping Lou learn about mentorship. And it was both Lou and Dick, Frank and Dave, who really, when I was working in their labs, I watched how they worked with students, how they really provided mentorship that was strong and important. And I hope I've taken that forward into my career as well. So I finally got to collaborate with Lou. He was moving into a slightly bigger reptile, and again, down the evolutionary tree a bit, into these tuataras. And again, I did not and never did get the opportunity to go in the field with Lou. I wish I'd been able to go with him to New Zealand to collect the plasma from these guys, but he did send them to me, and we worked on developing a vasotocin assay specific for plasma from tuatara in order to determine that, again, vasotocin has this evolutionary action on nesting and behavior. So then Lou moved on, of course, to bigger and better things. We've seen this picture of Lou multiple times over the course of 2 days at the University of Florida. And that's when I'm going to now pass it over to Karen to talk a little bit about her introduction to Lou and ultimately how Lou brought the two of us together. Take it away, Karen.
Thanks, Kathy. I first encountered Lou at the 2000 Gordon Research Conference on endocrine disruption, and I met several of you there for the first time as well. I was a newly minted assistant professor, very wet behind the ears, and I had the honor of presenting my lab's work at that GRC, and I was talking about the effects of the pesticide acetochlor on thyroid hormone-dependent gene expression using bullfrog tadpoles as a model. And back in those olden days, the use of molecular techniques wasn't quite a thing, especially with wildlife. And my lab was developing like some of the early cDNA arrays. For those of you who remember microarrays, those are predecessors of RNA-seq, just saying for those of you who are too young to know these things. And also developing proteomics and metabolomics methods to fill in the gaps. So we were very interested in using bullfrogs as models and also other fish and wildlife as well. And at the time, like Lou's enthusiasm was infectious from right from the beginning, just like what everybody else has been talking about. And as an honorary Canadian, he introduced me to my new scientific family that literally shaped my career, like so many others. He introduced me to his wonderful family, Buzzy and Matt, Teresa, and his dad, and also his twin brother by another mother, Tyson Iguchi. And he got to know my family too, just like so many other families. And I really cherish the visits, and I'm really indebted to both Lou and Ty for their friendship and mentorship throughout the years. And Lou taught me about the power of storytelling, and again, a common theme throughout. He and Buzzy were such gifted teachers, and the point is that they were so willing to give, stuck with me. And also the importance of letting the science do the talking. Charles mentioned that just now. And, to be able to deal with, doing the best science that we could possibly do to counter the deniers and address the sometimes unpleasant responses that we get from industry about our sublethal effects work and stuff like that. So Lou Charles mentioned, was very, very keen on applying molecular tools to his lab's work, and so that's how my lab and his lab started collaborating. And we ended up with some really neat papers on gators, looking at thyroid hormones, which hadn't been looked at much at the time, and even finding serendipitous types of discoveries, like a tumor suppressor that we initially showed was involved with thyroid hormone signaling in frogs, might actually have a similar function in gators too. So that's kind of cool. And next slide, please, Kathy. And as has been mentioned over and over again, Lou was so passionate about teaching and training. Leaving a legacy behind was of people was super important to him. And I had the privilege of working with colleagues that he introduced me to. Many of you are in the audience. Hi, everybody. And numerous members of his lab, including this youngster here, Thea Edwards. Knew as a graduate student, worked with one of my grad students, Ashley, to investigate the influence of nitrates and nitrites on thyroid hormone and stress responses. And we're using a really cool serum-free organ culture system to do that. And the take-home message from that work was that the disruption, in addition to changing the magnitude of responses, can also take the form of a change in variation within a population that could have bad biological consequences. And years later, Thea and I still collaborate on related topics, looking at environmental DNA degradation and the role of microbes in that process. And then another introduction for Lou went something like this. Karen, I'm going to introduce you to Kathy Proper. You both have a lot in common, and I think you two will hit it off. Well, he was right, and that introduction turned into a long-time collaboration and friendship, including stints in each other's labs. One of the most memorable visits was Canadian summer camp, where Kathy brought her family up to visit my family, and Kathy and I had fun in our lab, the kids had fun at their science camp, and now Kathy's going to show you what we did at summer camp and beyond. Kathy, back to you.
Thank you, Karen. So yes, we came up with the whole family. We flew to Seattle. We took the boat all the way across to Victoria. The kids got thrown into the Victoria Science Summer Camp, and I told my kids I was going to go to my own science summer camp in Karen's lab. And that was really because I had been talking again to Lou at a meeting. And I said, we're doing these cool studies with thyroid hormone. We've got a microarray put together to take a look at the impact of wastewater. And Charles gave a wonderful talk today about the impact of wastewater on fish and those complex mixes. And we were beginning to look at that, and we had the microarray set up, but I really needed to learn how to do real-time PCR. And I hadn't done that, and I was talking to Lou about really maybe going over to his lab, and he said, oh, for frogs and this, you've really got to talk to this woman, Karen Helbing, up in Canada. I think you guys would get along great, right? So Karen and I got the same story from Lou, because again, Lou is mentoring and putting people together. And so I went up to Karen's lab, I learned how to do real-time PCR, we worked together on this project, taking a look at thyroid hormone-dependent development. And we were using the lab models, Xenopus lagus, as our screen. And again, people have talked about sentinel species, both in the field and lab, that are useful. And we found, I'm not showing you the microarray data, but through the microarray data, we found a few genes that were extremely sensitive, not only to the wastewater, but also to thyroid hormone. And here you can see two of those that are really renowned now for the importance of thyroid hormone-dependent development in these guys. We found the frogs developed faster when they were in wastewater, which again is one of those cool stories where we anticipated they would be developing slower. We got the opposite response, and we figured out that really the reason for that is if they had their own thyroid hormone going, or if we gave them thyroid hormone, it accelerated some of those thyroid hormone-dependent genes, the expression of those genes. And then, of course, Lou started out with the alligator those estrogen-dependent genes. And again, we've heard several talks about these over the last couple of days. Excuse me. So here what you see, this was animals taken straight from a wastewater pond. Again, we're working with bullfrogs here now in the field, and went into wastewater treatment facilities where bullfrogs were breeding downstream. We picked up frogs. We saw that there was enhanced feminization, or gonads with testes with ovaries, oocytes in them, and we saw elevated aromatase. But not only did we see an elevated aromatase, but in situations where we gave animals things like octaphenol, we saw decreases in a gene called steroidogenic factor 1, which is sexually dimorphic, and is really part of the initiation of the entire steroidogenic process. So from there, Karen and I started to talk about really creating comparative molecular tools across amphibian species. So again, this now takes into account a lot of the comparative approaches that Lou really used throughout his career. And with that, I'm going to pass the next couple of slides back to my dear colleague, Karen.
Yeah, so reaching across the amphibia, of course, there's about 260 million years of evolution. Next slide, please. That covers just the amphibia alone. And what we did was we wanted to see if we could generate some tools that could be the same tools that could work across all the amphibia. And in the quest of trying to normalize how we look at particularly thyroid hormone disruption and the enormous sensitivity that amphibia have towards this. And as a result, we were able to generate a number of different tools that are now used across the world for this purpose. And thyroid hormones are super important in all vertebrates, and amphibia are one example where metamorphosis is so dependent on thyroid hormones that it's like an amazing magical system as far as I'm concerned in terms of illustrating the importance, because metamorphosis is completely dependent on thyroid hormones. But yet we don't really understand much about how the thyroid hormones work, especially in different tissues and so on. And another aspect of thyroid hormones is that we know there are two different kinds. Next slide, please. And those two different kinds of hormones, are active hormones, thyroxine and triiodothyronine, are ones that are touted as being like T4 or thyroxine is often touted as like the pro-hormone to triiodothyronine, which is more bioactive. But we found through comparative studies that of the different tissues and so on, and actually doing a head-to-head comparison between the two hormones, taking into account the slightly different amount of biological activity, that depending on the tissue that you're looking at, the hormones have very different effects. And that is really important in terms of understanding ultimately how endocrine disruption also works. And so really T4, as we found in some tissues, isn't just a pro-hormone. It doesn't just get converted, it's not transported and then just converted by deionases to an active form of thyroid hormone. It's a hormone in its own right, without conversion, and different tissues use it in different ways. And this now is a sort of a working mantra for us to really uncover how these two hormones can have overlapping functions, and in some cases can have the pro-hormone to active bioactive hormone relationship, but it's different for different tissues. And we need to keep those things in mind as we're looking at endocrine disruption and thyroid hormone action in general. And now I'll pass it back to Kathy to round up our presentation.
Well, thanks. So most of you who know Karen probably know that she's really moved heavily into the area of eDNA and is probably one of the world's experts at validating assays to look at eDNA. That's environmental DNA. I don't think I need to explain what that is to this crowd, but it's the DNA that's left in the environment by organisms living there. And eDNA can help us understand both who's there, but also who has disappeared. And again, many species, and especially in freshwater environments. Sorry, I'm in LA next to the freeway. In freshwater environments are really losing their diversity. And so it's important both for us to be able to document what's there, but also understand what's not. And Karen and I both have been working independently with our local indigenous populations in Canada, the First Nations, and here in Arizona, I've been working with folks on the Navajo reservation, and one of my Navajo students who's really dedicated, they're all dedicated to really understanding how to best preserve the biodiversity that we have. But that comes with some problems, including that being able to just find the organisms, some of the taboos from the local indigenous tribes and groups on handling the animals, and then some of the ethical issues associated with DNA, including eDNA. And so our students are working together now to put together a paper on eDNA ethical practices for indigenous populations. And we're working on a number of projects. Karen's working on projects up in BC and across Canada, and I'm working with my student to reintroduce northern leopard frogs onto the Navajo Nation, where they've largely been extirpated for a number of reasons. And these amphibians are really critically important culturally, and the loss of them has been very important also to many of the different indigenous groups. And with that, we'll move it back to Lou, and how Lou's really brought so many people together, so many of us in the audience and across time. And from my very first introduction to Lou as the scoop of my term paper, all the way to my continued collaboration with Karen and others, and the importance really of taking a look at animals across evolutionary scales, the importance of it now with the change in biodiversity, and really the attacks on science that are happening right now within our country. And so I want to thank Lou for all the support he's given to us, all of us here in the room throughout the years. We miss him, and thank you.
Thank you, Lou, and you know the legacy does live on. I think it's very evident in the symposium that you know he's touched many lives and enriched them all for the better, and we hope to continue that legacy moving forward. Thank you.
So as I mentioned yesterday, I never actually worked directly with Lou, but obviously his work in Lake Apopka was an inspiration. And as I mentioned, you know, it was because of him that we got into what we're doing, actually. So contaminants, you know, are there. They've been banned for decades now, but they're still having effects on wildlife because they're very persistent. And in Florida, they were highly used for many years. And they, everyone in this room knows, they're endocrine disruptors. again, I hadn't thought about talking yesterday, but it was because of Lou that we started the biotechnologies and ecological and evolutionary and conservation laboratories. And my laboratory was the one of doing two-dimensional gels. And so I have a picture of a two-dimensional gel. It's not the one that we had from the fish, but basically we found a large protein. So at the time, we didn't have proteomics like we have it now, where you could put something in mass spec and identify exactly what it is. You could have a whole proteome by mass spec nowadays and identify all of the proteins. But back then, it was very tedious. You would cut out a spot, you would do the gel and transfer it to a membrane, cut out the spot, and then put it into an Edmund sequencer. And an Edmund sequencer is, it was like an HPLC-based thing, and it would take one amino acid off at a time and identify what it was. And so it was a very tedious effort. But we did that, and we were able to find out, the one spot from these fish that was the telogenin. And that's really what started the whole thing for me. Okay, so my work was centered back then in Lake Apopka, and again, for the reasons that everybody here knows, because of the contaminants, because of Lou's work in the lake, and because I was more interested in fish. And so Lake Apopka, the northern part of Lake Apopka, everybody knows about the spill of DDT into the lake, but that was in the southern part of Lake Apopka, down, oops, I did it. How do I get back? Ah. Okay, thank you. And so, around Board's Neck, at the very south part there, that's where the spill occurred. But all of the alligator studies of Lou's and the work that I was interested in was really because of the farms at the northern part of the lake. And that was never really considered a Superfund site, but that's where the contamination was. And it was there, that's what the farms looked like. And in the 40s and 50s, they were basically diked muck farms. A muck farm is muck. And so they diked it up, and so they could grow vegetables there. It was very fertile. And so it was beautiful, very fertile, and it looked like that. And at some point, the lake, those farms were purchased by the state of Florida. And then they decided to convert those farms back to the natural environment. And so they did. And then, took out the dikes, and birds came in, migratory birds, and 5,000 birds died. And they were like, wow, it was probably a virus. No, They did the studies, and it was the contaminants. But anyway, that's what the farms look like. They used to put 68 pounds of DDT per acre twice a year. And then when that was outlawed, they went to chlordanes, dieldrin, methoxychlor, toxaphene, and others. It was a ton of stuff. And ladies worked there. These were mostly black women that worked on the fields, and they told us that the planes would come overhead and spray them, spray their food, spray everything. So Margaret James was part of a project that we had together that was funded by NIEHS, a Superfund program project. And so we got to meet some of these people. Maybe this was 2015 or so, I think. Anyway, at that time, some of the ladies there, most of the people that had actually worked on the farms had passed already, but there were still some older women there. And they, told us about their health issues and their children's health issues. And so we were lucky to be able to get them to agree to give us a blood sample, and we tested their blood. And so they had a lot of DDE still in their blood. This was 2015. DDE hadn't been in use since the 70s, so it was a long time later. And some of them even had DDT in their blood. Now, DDT breaks down to DDE, so that was probably a more recent exposure. And I think possibly people kept DDT in their backyards, and it was very effective. And so maybe they were still using it, or maybe it's still out there. Who knows? So much of it was used. This is it. And so what they told us was that they felt like they had lupus, and an autoimmune disorder, and probably diabetes. A lot of them were overweight, but lupus was the one thing that came to mind. So we tested their blood, and yes, indeed, all of the people that we tested seemed to be positive, as you could see here against the little diagram there. And so, they told us that they were told by the medical institutions that no, there was no specific increase in lupus in their area. This was normal, this was natural, and they send them home. But their children also had a higher incidence of lupus. Okay, so, we tried to help them as much as we could, but my study's about fish. And so we did largemouth bass. And so why largemouth bass? Well, they are a good predatory fish. They're at the top of the chain. They have a lot of contaminants. And we've done a lot of work even past what I'm showing here. But we've been able to show, that these contaminants get distributed in their tissues, get into their brain, so they're there. And also the connection to people is that, well, there are a lot of these people who actually will catch these fish and eat them, and if they are Vietnamese of origin, maybe the whole fish gets boiled, and so whatever is in the brain or the organs, the liver, whatever, they're getting that. So it's a complex system, and like zebrafish, everybody works with now, they're, I think, a good model for human health because they are, a vertebrate. And what I really like about largemouth bass is that their reproductive system goes over a long period of time. So you can actually look at different parts of the reproductive system. They start in October, developing eggs, and then they don't spawn them until March, April, May, something like that. So you can look at different parts of that development. In any case, this is a Lake Apopka, and when they were trying to figure out why the birds had died, they went through and they did measurements of contaminants in the soil in the farms, and this is just toxaphene, but you can see that there are areas that are really highly contaminated, and basically where those little red dots are, the St. Johns River Water Management made a couple of ponds there, and they to test to see about if the contaminants maybe really were the cause for the die-off, and they decided from those experiments that they were, but then they still had those ponds, and they let us put fish in there to try to see what we were interested in. So we put fish in there and left them in there for about 4 months, and you could see that at the very bottom of each part, the white little bands are the pre-exposure levels of those contaminants. And after 4 months of leaving them out there, the contaminant levels in those fish were really quite impressive. And so based on that, we did a lot of studies with DDE, dieldrin, methoxychlor, and other contaminants, looking at them as endocrine disruptors. So usually you can distinguish a fish, a male fish, the blue dots, from a female fish based on their hormone levels. So male fish will have more testosterone, which is on the x-axis. Female fish will have more estradiol on the y-axis. And even in the, when they're estrogen levels in female fish are low, you could still distinguish them. But if you take fish from Lake Apopka and some of these other different polluted areas, you can no longer distinguish the males from the females, suggesting that there's, in addition, that the reproductive effects are multifaceted. It's not just that these hormones are acting as estrogens, but they're really interrupting the whole steroidogenic process. And so we were able to repeat the same kinds of studies in the lab by feeding them diets of these different contaminants. And so we did DDE, we did dieldrin, and as soon as you put that contaminant, most of our studies were 21-day feeding studies. You could see that after 21 days, you could no longer distinguish the males from the females based on their hormone levels. Okay, so systems toxicology that was alluded to earlier. So the usual toxicology method is to look at a fish, look at the toxicant, see how the phenotype of the fish changes, do histopathology. The apical endpoints are reproduction, growth, survival. That's what all the regulatory points are made from. Absorption, metabolism, and that kind of thing. And we early on decided to try to put those together with molecular methods. And so we marry those together with the omics. And so we developed, to begin with, dotted microarrays, then microarrays that were bigger, and now, of course, like everybody else, we're doing RNA-seq. But the very first pages or papers that we did were with these microarrays that we made ourselves. It was a lot of work. So we were interested in endocrine disruption, and sure enough, the fish from these ponds and the ones from our labs all had endocrine disruption. We could see that endocrine changes, vitellogenesis, female gonad development, oocyte development, hormone biosynthesis, all of the proteins or all of the transcription factors, all of the endpoints that we expected to see, we saw. And so that was great. Okay, so we were trying to understand reproduction, so this is what we were interested in. I didn't know anything about immunology. This is what we saw with immunology. Oh my gosh. It was way worse than maybe so many more endpoints than just the endocrine endpoints. And so inflammatory response, platelet function. And so this was from the fish that were out in the ponds, and then the ones from the lab, we did similar kind of thing. And we got all of these endpoints, immunological endpoints. And I thought, what? Wait, lupus? Lupus? And then the people had lupus. The fish have the same, I don't know that fish get lupus, but they have the same, vertebrate system, the same pathways. Wow, maybe those people are getting lupus because of their exposures. Who knows? And I think it's one of those things where you can be exposed today, and 20 years from now is when you see the result. So, a lot of autoimmune disorders seem to me like they're getting worse, but, I don't know. I haven't done a study of that, but I wouldn't be surprised. In any case, understanding organochlorine pesticides, I think, is really important. And for me was a mind changer, and I already mentioned a career changer. And I think it's really important to understand how these different contaminants work and the effects that they might have, because it's not just the fish, it's people as well. So, epigenetic effects, a lot of people have now shown that these contaminants have epigenetic effects. And again, the children of these people also had lupus. So, I don't know if that's an epigenetic effect or maybe it's just genetic, I don't know. but boy, I keep wondering. And I think the omics technologies have really opened the door to understand how these contaminants might affect different systems. We're now looking at lipidomics, and we're doing a lot of different kinds of things. It's so much fun. I'm still working because it's fun. So, you know, I love my students. Well, I don't have them now because they've all graduated, and I'm not taking any more because I'm going to retire. But I keep saying, everybody tells me I keep saying this, but I've been saying this for 10 years, you know, and I haven't retired yet. So maybe now is the time. Okay, thank you.
Thanks. Thank you. Thank you so much, and thanks for reminding us of that terrific story on Lake Apopka that we all know well. Jerry, did you want to make any comments before the break?
So I want to ask a question. So we have 2 talks left that will be about an hour. Do you want to just continue on, or do you want to break? How many people think we should take a 15-minute break? Okay, got it. It's a short break.
Thank you, Jerry, and everybody for organizing this. It's been really fun. As Heather mentioned, I'm more or less a grandchild within the lineage of Lou's legacy. So I actually didn't really get to know Lou myself personally very well, which I am sad about. But thankfully, I did get to meet him once when I was a graduate student, and that was like one of the most amazing experiences ever, because it was, you know, I think Thomas said this earlier, like meeting a celebrity, right? It was the most famous scientist that I knew of at the time, and I was so nervous. I was shaking like crazy. But within 5 minutes, he just had a way of making you feel comfortable, and an easy way of like just talking and conversing about science, and even, not even science. Like we were at a barbecue place having dinner, and he was just like, how's it going in general, right? Like having those conversations was just easy with him, which was really nice. And even though I only got to have that one, it was a really memorable moment that I will always hang on to. So as Heather said, I am an assistant professor now at Clemson University, and I kind of wanted to mention a couple of ways in which Lou has had subtle influences but big impacts on my research career and science career. So I first learned about Lou when I was a wee baby scientist in my undergraduate at St. Lawrence University, and I was taking an endocrinology class. And one of our assignments was to read the book Our Stolen Future. And so I was already working in the lab looking at mercury and how it distributes into Xenopus laevis as both tadpoles and adult frogs. So I was already kind of in the toxicology realm, but reading this book really solidified my interest and passion for going into the EDC field and the world of endocrine disruption. And Lou's studies, even though they're only mentioned a couple times in this book, really stood out to me. Like, I still have this old version of the book at home, and I opened it the other day, and the amount of like notes and highlights I have all over his sections is really funny to go back to and see. And because of that, I did end up actually, part of my assignment for that class was to present a paper, and I presented one of his papers because I was really excited about all the work that he was doing. So I first learned about him back in undergrad, and then as I went on to graduate school, I worked with one of his science children, which is Heather Padisal. She was my advisor throughout that entire time, and that was really how I got into the EDC research field. So I wasn't really doing endocrine disruptor work as an undergrad, but during my graduate work, I did a lot of work on endocrine disruptors and how they impact brain development and the placenta. And during that time, like I said, I was really fortunate to have one night to go to dinner with Lou when he visited NC State, and that was a really fun and memorable moment for me. So another subtle but big impact, you know, working with Heather and all of the traits that she acquired as a scientific mentor and teacher from her experiences with Lou really helped to shape my career and who I am as a scientist today. And then finally, kind of this last hit of influence throughout my career was when I returned to NC State for a second postdoc. in Scott Belcher's lab and started working with sentinel species as well as more of Lou's legacy children, so both Matt and Teresa, and learning how to conduct field studies with these amazing organisms and how they can be used to tell us a little bit more about what's going on in the environment and the health of our ecosystems. And so I've really taken a lot of those experiences and a lot of my research project, diverse research projects, little bits of them with me as I've established my own lab at Clemson, where I now work with a whole bunch of different organisms. So I've listed a few of them up there, but we work with mice to do human relevant exposure studies and look at mechanistic outcomes. We work with sharks. We're doing some projects with microbes. We have a bunch of cool collaborations with people on campus, including with Thomas Rainwater now, doing some alligator work and looking at mercury levels in them, as well as some human tissues. So we're starting to do some human projects with placental samples. And I will say the humans are the most difficult of the organisms we work with, for a variety of reasons. So really, when I think about my training, I've very much been in the developmental origins of health and disease world and prenatal programming. So when I think about this lineage, I think about it from a transgenerational approach or impact, where Lou would be the F0, all of the people that have helped shape my career in the EDC world are F1, and then I am the F2 grandchild, who is now trying to also impart a lasting legacy in some ways to the students and stuff that I mentor today. And so really it's the love of science, the teaching people how to be a good scientist, pursuit of things that you're passionate about, importance of collaborations and mentoring, and then science communication that have really been drilled into me through my experiences and my mentors, many of which are in the room right now, that I know that we see a lot of Lou in, right? There's a lot of impact from what he's done and the priorities and ideals he's instilled in his F1 population that have now trickled down into my training and development as a scientist. And so, I'm a new assistant professor, so I really want to talk a little bit about what we're currently doing in the lab. And I think you'll see hints of overlap between some of the research interests that both existed in my advisors' labs, as well as what Lou is doing. Which, again, is just another way of thinking about those lasting impacts that a really great mentor and scientist can have. So first, I was just going to talk a little bit about some of the human health studies that we're doing, mainly referencing some studies that are being done by current graduate students in the lab, geared towards looking at understudied windows of exposure that may be critical for female reproductive health, target tissues that are understudied, as well as some signaling mechanisms or axes that still need a lot of work to understand how EDCs and hormonal dysregulation can impact them. So the first is really focusing on the preconception window. So my graduate student, Miriam, who's just been an amazing addition to the lab, had never worked with mice before and can now carry on her own and lead her own mice projects or mouse projects. It's been kind of amazing to watch. She's really generated a lot of pilot data towards this study over the last year. So my lab's only been open for about 1.5 years, and she's done a lot of work already to establish this model. With the critical importance here being that this window between puberty and pregnancy is often when young women are establishing their beauty and feminine hygiene practices. And as we know from many talks already, those products are laden with phthalates and other endocrine disrupting compounds. And so what Miriam is doing is she's taking an environmentally relevant mixture of phthalates and exposing mice between puberty and pregnancy, and then looking to see what happens to offspring as well as the processes of implantation and placentation. So we haven't gotten into all the detail with the characterization of what's happening to implantation and placentation yet, but she does have some pretty compelling data showing that this exposure window can disrupt the estrus cycle, which is similar to the menstrual cycle in humans, in our mice, and has persistent effects on fetal body weight or offspring body weight throughout their life, both while they're in utero and well after when they've become adults. Additionally, our lab manager, Paige, has been spearheading some projects looking at the direct effect of phthalates on the vaginal microenvironment, which is comprised of a whole bunch of different cell types and microbes, as well as mucus. And all of these cells and microbes and mucus interact together to help to prevent things like infection and inflammation within the vaginal environment. But of course, a lot of the feminine hygiene products that are used these days, the common ones, the brand name ones that everybody's using, contain phthalates. And we still don't know that much about intravaginal exposure to phthalates. So what Paige is doing is she's culturing vaginal epithelial cells in the lab, exposing them to environmentally relevant mixtures of phthalates, and seeing how that's impacting some of these key functions that are being provided by the vaginal epithelial cells. So in this case, we looked at a few different markers for inflammation, and we do see dysregulation of these inflammatory genes with exposure to the phthalates. And kind of nicely, which is why I really wanted to highlight her data on the phthalate mixture, because of course we're also looking at the individual compounds, but we're kind of seeing these classic non-monotonic dose responses with our EDC exposures. And then we have a really awesome collaborator up at the University of Maryland who has actually been taking this one step further, and she's directly exposing vaginal microbes to see what it does to their cell viability and function. And in this case, across the phthalate mix and a couple of individual phthalates, we are seeing that a healthy vaginal bacteria, L. crispatus, is being killed off by these phthalates. So it's actually reducing the viability, the number of viable bacteria in these cultures. And then the last human study I wanted to plug was a project that really was developed by Ansley all on her own. So some of these other projects arose through, Hannah and I became friends when we were postdocs, and so now we're like, okay, how do we work together? And we came up with this direct exposure to vaginal cells and bacteria project, and then I'm a placenta person, so that's kind of where Miriam's project came out of. But Ansley joined the lab, and within like 2 months was like, I really want to look at the liver and the ovary and how they're communicating and how dysregulation of either one could impact the other, especially since there's a ton of literature showing comorbidities associated with dysfunction of the liver and dysfunction of reproduction. So she's been spearheading these projects. Again, this is focusing with phthalates right now. That's kind of the mixture that we've been going with for all of our human studies right now, at least in the mouse model and in cell culture. And she's been looking at various genes related to detoxification pathways, steroid synthesis pathways, and steroid transport, so like sex hormone binding globulin. And so here I'm just showing a little bit of her data with CYP1A1 and the changes in expression of CYP1A1 related to either phthalate exposure, human relevant levels of estrogen and progesterone, or the combination. And what's, again, kind of cool about her data is we're seeing that when we have the phthalate mixture, the effect is very different than when we look at hormones alone or hormones and phthalates together. So in addition to the human studies, we do have some wildlife studies going on. So I'm not going to talk much about the collaborative studies that we have with some of the people from Jim Anderson's lab and work with Thomas Rainwater in relation to the alligators, but rather focus on some of our shark studies that master student Mel Walker is leading right now. So Mel is really interested in looking at sharks in part because they're a keystone species for many marine communities as they regulate and help to balance those communities by regulating the number of their prey species that are present. They utilize coastal waterways a lot for very important reasons that could affect their population numbers. So things like feeding and breeding, pupping, and they serve as nursery grounds. And of course, this brings the sharks during vulnerable life stages close to the coast, where there's likely going to be a lot higher concentrations of pollutants because of human activities and urbanization of our coastlines. And of course, as predators, they're vulnerable to bioaccumulation of contaminants. So those contaminants that can bioaccumulate can reach toxic levels potentially in these animals and cause negative health effects. So what Mel is doing is she's collaborating with our amazing collaborators at UC Davis and Oregon State to look at seven-gill sharks, specifically those that live in either, and I guess I clicked a little too soon, in either San Francisco Bay, which by the overhead shot you can tell is much more urbanized than Willapa Bay, or in Willapa Bay, which is really not developed and comparatively pretty pristine when you look at it next to San Francisco Bay. And so far, her preliminary data, we're still collecting a lot of samples for this project, but does suggest that we're seeing differences across ontogeny, so across age classes, between organisms that are considered neonates or subadults or adults, as well as site-specific differences within age class. So that would be the one on the farthest right, showing that in San Francisco Bay, we have higher concentrations of mercury in this case in these animals compared to Willapa Bay when you look within age class. We will also look at PFAS in these guys, but mercury was a nice, easy way to start. We have a mercury analyzer in the lab. It's fairly easy to run. PFAS takes a little bit more time. So Mel is learning all the ins and outs of the analytical chemistry to process and analyze those samples now. And then what was super cool, and she put in an NSF GRFP for, is she was combing the literature and decided she also wanted to look at how feeding ecology impacts bioaccumulation of contaminants. So not just geographic location and proximity to urbanization, but feeding ecology. And uniquely, we reached out to Brian Fraser at DNR, and he was like, oh, I've got a great model system for you to do something like that in. We actually have these fine-toothed sharks whose diets are 95% Atlantic menhaden, which is a primary consumer. So we know that they eat basically only this fish most of the time. Whereas we have sandbar sharks that are fairly similar in size, have similar reproductive strategies, but are generalists. They eat a mixed diet of primary and secondary consumers. And so what she wants to look at is whether or not contamination or contaminant concentrations, including mercury and PFAS, are higher in these sandbar generalist feeders compared to the specialist fine-toothed shark. We don't have any data on that one yet. We've been on one sampling trip, so this is Mel with the sandbar shark that we caught that day, just more or less as a proof of concept that we could regularly catch these guys. We were successful. It was a good time. And then finally, I wanted to just highlight another master's student project that really kind of brings our entire One Health toxicology theme to the lab together, which is more focused on ecosystem health. And so this is where microbes come in, which I'm not a microbiologist, but he was excited about this angle, and so we rolled with it. So similar to the Save Our Seas Foundation project, where we're looking at sharks caught in either an urbanized estuary or a more remote pristine estuary, in this case, we're actually collecting water and sediment samples to look at both PFAS levels and microbial dynamics in communities in an urbanized versus pristine bay. And of course, microbes play incredibly important roles in ecosystem services provided by these estuaries, including things like nutrient cycling and degradation of pollutants. And so there's kind of two angles to this project. One being, what are the PFAS doing potentially to the microbes? Are they killing off and are they toxic to certain species and therefore compromising important ecosystem services? And could they potentially be used as a bioremediation tool? So far we've been working with our collaborator Andrew Tweel at DNR to do this project, and Zach has started to characterize the microbes and the PFAS in these sites across season and between some of these sites. So we are seeing some differences that are based on season and location in our microbial community structures, as well as our PFAS, not necessarily by individual congener, but the total load of PFAS is different between the two locations, higher in Charleston, lower in St. Helena Bay, or St. Helena Sound, excuse me, and different across seasons, where we're seeing higher levels of total PFAS load in June compared to October. So I'm excited about the continuation of this project because he's going to start doing cultures in-house, both anaerobic and aerobic, and starting to develop microbial enrichments so that we can do some studies to see if any of those microbes that thrive in the presence of PFAS are in fact degrading them, and how effective they might be at that. And then I just added this slide yesterday because we've heard a lot about how Lou is involved in science communication and outreach and telling stories through science. And so one of the things that we do a lot in our lab, which stems from all of my mentors, where in Heather's lab we constantly participated in things like Brain Awareness Week, and in Scott's lab we were constantly engaging with the community when we were out in the field. Our lab also does a lot with science communication, and it's a priority that we have in the lab that's been really fun. So our lab manager, Paige, spearheaded a summer project that we called Peppy Planarians. And we worked with students, local students that were aged 5 to 17. That was wild, all of them in one room together. To expose planarians, which are just little flatworms, to caffeine and to see how it changes their behavior, to teach them a little bit more about how chemicals in our environment can impact physiology and animal function or organismal function. And then we also made a really fun video describing and kind of trying to teach people how to look at product labels and assess them for potentially hazardous EDCs that they should be on the lookout for, which the QR code will link you to that video if you'd like to see it. It's up on YouTube and stuff. I'm trying to be all media forward as much as we can. So of course, I want to thank my entire lab because they've been amazing. We've only been at Clemson for 1.5 years, and we've already got a lot of projects rolling and a lot of interesting and fun data that's being generated by the lab. And all the names in italics are our undergrads. They've been a huge force in the lab, super engaged and participatory. Of course, our collaborators, one of which I didn't get to talk much about today, but Amy Crockett is a OBGYN at Greenville Memorial Hospital, and we're doing some work with her this spring looking at the expression of drug transporters in the human placenta and whether or not that's different across sex. So that's another cool project going on in the lab that I didn't really get time to talk about today. But in addition to my lab, I also just want to make a huge shout out and thank you to science, friends, family, near, far. We've had that theme a lot throughout the talks since we've been here. And it's really true, you develop this community and family through science. And a lot of those traits that Lou has instilled in his trainees have trickled down to his grandchildren now, so myself, where you'll see things like a silly photo of Heather and I pointing at each other when I defended, and awesome photos in the field with Scott and Teresa and Matt, and you'll start to see that in my own lab photos and stuff, where, it's good to take science seriously sometimes, but we also can have fun while we do it. So thank you to all of you and to my mentors that are in the room, and of course, kind of indirectly to Lou for all of the impact that he's had on my career.
Also want to thank Jerry for organizing this, and Nancy and all of our UF people here who have made this possible. And when I first heard about this symposium, I really didn't know what to expect. I didn't know the right tone. I was just kind of like, man, this is going to be tough. And but I think it's been really cool getting to see all of these talks. And I think if you take it kind of in total, what I think is this kind of this really nice encapsulation of science and friendship and fun and community that I mean, this is Lou's legacy that is here right now. And I think we're still kind of we're in it. It's not something that I think we have to be retrospective about. And so, as the title here implies, what I wanted to do today is talk a little bit about science, a little bit about legacy, and some other stuff. And I don't think it'll take too long. So I'm going to go ahead. The first thing I wanted to do is start with this and give a little bit of background about how I initially met Lou. Anybody got a clue of what this is here? What it kind of looks like a graboid, from Tremors, or they called it a dirt dragon, I think, right? This is a Drosophila testis, okay? So it's a testis from a fruit fly, and that's what I worked on for my PhD. And so the way this worked is you would do any sort of molecular work you needed to in the mornings, and then in the afternoon, you park yourself in front of a dissection scope, you get your forceps out, and you just dissect for hours and hours and hours hundreds of fruit fly testes because you needed them for your experiments, right? And so as we were wrapping this up, or my PhD up, I was like, man, what am I going to do starting to think about postdocs? Who could be the right how do I find the right postdoc lab? And I kind of came up with a few criteria. One of them is I wanted to work on something bigger. And it sounds really trivial, I get it. Sounds maybe intellectually stunted. And if you know me, then that doesn't, it's not so surprising. And then at the other time, or the other criteria was I wanted to work on something that had to do with the environment. So, we were working on these fruit flies, and we were knocking out genes and overexpressing genes and seeing how they affected these stem cells that are kind of staying there in green. And what we wanted to do is kind of look at these genetic pathways and describe those. But I was kind of becoming, I wasn't convinced that these genetic pathways were actually paralleling what we were seeing in nature and natural variation, so across populations or individuals. And this was also at the time the genome-wide association studies were coming back. This was like, I graduated in 2011, so it was like maybe 2008 we got the first, that's a guess, genome-wide association study on like heart disease. It was like 9, 10, 11% of the variation is what we were explaining. And I was studying genetics. I was like, man, maybe I need to shift focus to the other 90% or whatever. But all right, and then the last thing is I had worked with an assistant professor who was awesome. I was her first graduate student. She was great and always at the bench and really invested in my success. But for my postdoc, I wanted to work with somebody who was like a leader in the field, someone who was really established, and so I could kind of watch them and see, as Herm Edwards, famous football coach, said, he said, how are you going to be great if you don't study greatness? And so with that in mind, I started looking for labs, and I quickly found Lou's lab. And I started emailing Lou every week, and like for a long time. Like I was like a year out starting to look for postdocs, no response. I was like, man, maybe he's old school. So I printed out my cover letter and my CV in nice paper and mailed it. Nothing. Kept on emailing. Called Donna, who's the receptionist at HML, and was like, "Can you leave a note on his door, or like where, And then finally, I got this email, and it was great because it was one afternoon. He emails me and he goes like, "Hey, sorry, let's talk." And so I was emailed back, "Great, sure." And what was happening is Heather Hamlin was being recruited to her dream job at the University of Maine, and she was a postdoc in Lou's lab at that point. And so when she got her position, that freed up a position for me. And so I'm always thankful for Heather of getting her dream job and giving me an opportunity. All right, so then I got to Lou's lab, and this is kind of how I was thinking about things at the time. you know, the biology of the beast, that's the Louism I was thinking of. So that came from Lou, the biology of the beast. But this is really the general framework, I think, that I was thinking about things where you've got biomedical science here, and then you've got kind of ecology and wildlife biology, two separate fields with this really big thick line that demarcates this, and you're either on one side or the other. And that's kind of how we you know, I was trained and was thinking about my science. But what became really clear early on, going back and forth to the medical school, the campus, the REACH talks, or I can't remember exactly what they were called at MUSC, or going to the field with Thomas and Phil at Yawkey, was in Lou's lab. This line really did not exist. They didn't acknowledge it, and this is a slide that Lou would show medical students, where he was kind of thinking about, like, where did that come from, right, this line? And he thought it was kind of rooted in royal bloodlines. So this, our kind of cultural fascination with genetics that was rooted, you know, in early kings and queens, and then that kind of, this was a relic, this genetic determinism of how we were thinking about disease, how we were thinking about environmental health, and was really shaping that. I always thought that was really insightful. I think it probably still, we see relics of that in the way some of our politics are operating today. But one of Lou's superpowers, right, was not only to make those connections, but also to communicate these connections, right? He could see that and communicate those, and we've talked about that, or others have talked about that, and was really this pioneer of the sentinel species concept, right? And the idea here, this is the slide that others have shown that he would close out his seminars with, where he would say, if the environment is not healthy for her babies, it's probably not healthy for ours. And this idea was powerful, and it still is very powerful. It's incredibly powerful. And I feel like what we need to be doing as the F2s or the F3s is, how do we extend this? How do we strengthen this idea of the sentinel species concept? And how do we extend it? How do we push it further and do our parts? And so I think that's the big challenge for us now. And I think it's really key to how we engage or re-engage a weary and distracted public, right? It's like that was what was so key and powerful about Lou, is the sentinel species. It resonated with me and us as a scientist, but it also resonated with the public at the same time, and it didn't require a different set of language. It made sense. It's intuitive, and I think that's important. And so Lou was able to expertly communicate all this, and it's still critically important. But I think over time, what we've seen is that the EDC field, the endocrine disruption field, has kind of transitioned to more traditional models, mice, cell culture, et cetera. And I think that's right, all for the search of mechanism. And mechanism is really important, but I do think we want to be careful and to preserve our environmental relevance as we seek out mechanism. And people still care about the real world and intuitively understand this point, and we can't lose that point about if it's not healthy for her babies, it's not healthy for ours. That's so powerful. And I would argue also that as technologies improve, sequencing, we're kind of in this post-genomic era now, that non-traditional models like the alligator, like the sharks, all the stuff that we've heard about, they can not only serve as warnings now, but we can also learn mechanism here. And so what I wanted to do in kind of light of all of this is I wanted to give a brief update on our work about Apopka kind of over the last decade, that we've revisited Lou's work and tried to extend and build upon that. And so I'm not going to go over a ton of detail here about the lake and the system here, but these stars Nancy did a great job of talking a little bit about the North Shore here, where we saw all of the pesticide inputs and the chemical spill down here in the south west corner, and Winter Garden contributed probably a little bit too, I'm sure. And so over the years, though, Lou and like his army of grad students and undergrads and research scientists and postdocs were able to compare the reproductive health of alligators in Lake Apopka to Woodruff and see all of these things, right? Abnormal sex hormone concentrations, disrupted follicular development in the ovary, altered expression of particular genetic pathways, altered response to gonadotropins, and altered gonadal DNA methylomes, and reduced penis size. And whenever I go over this, I have to pick out, this is my second favorite paper that Lou ever published. I'll show you what my first one is in a few minutes. But this was reduction in penis size and plasma testosterone concentrations in juvenile alligators living in a contaminated environment. And what I loved about this paper was that you can see here, this is the penis tip length and the base length. And what they were doing is, this is how long the alligator is. And the penis size scales with alligator size, which is not the case in humans, I don't think. Maybe it is, I don't know. I'm a small guy, so. Anyway. All right, but okay, so that wasn't a planned joke. All right, so, but what was really cool about this, this study, though, to me, was that you had Woodruff and Apopka, right? So Woodruff is the reference lake, Apopka is the, I'll just use this pointer if I can. I'm scared, though, that I'm gonna mess something up here. So maybe I'll try to use this. All right, so here is Woodruff and Apopka, but then down here is the Gordneck. So this is this GNS here, and so he split Apopka up even, or they split Apopka up, and so out here the penises are bigger than here, which is kind of where that spill is. But so I'm curious, maybe that has, I was curious in Nancy's, maybe we can talk about that. I think that's kind of interesting. And then, so usually when I show this to students, I'm like, who's seen an alligator penis? And no hands go up, because it's this just incredibly odd. I'm gonna ask, let's see this. So who has not seen an alligator penis here in this audience? Oh, there's more than I thought, okay. All right, so this is still worth showing. And Brandon, this is for Brandon Moore if he's on Zoom right now. So this is an alligator penis, and it is just this wild looking structure here. And Brandon Moore has gone so much into how the shape and morphology of this thing is important, but yeah, it's just, I have to show that. All right, so now I want to go into a little bit of our own work. This is Matthew Hale here, Dr. Hale. And Matt came to the lab and helped me start a lab at UGA, and we kind of were asking this question of, okay, how do endocrine disruptors adversely influence reproductive health in nature? And in this light, this is kind of what we started to explore here. And what we did is Brandon Moore, Matt Milnes, all of our predecessors in the lab had already shown that what you can do is go out to the lab here, or go out to the field, collect eggs from Lake Woodruff or Lake Apopka, bring them back to the lab, you incubate them, you let them hatch, you raise them up under identical conditions, you've got toe tags on them, right, so the Apopka and Woodruff animals are crawling all over each other. You raise them to 5 months, and you would see these persistent effects on gene expression of particular pathways, candidate approaches. And so what Matt was really interested in doing is saying, okay, like, that's cool, we know things about aromatase, et cetera, these certain genes, but let's see, like, if we just take a transcriptome-wide, unbiased approach, what pathways might be affected was the idea. And so we incubated everything at all the eggs at FPT, female promoting temperature, wait till 5 months, dissect out the ovaries, and we look at the transcriptomes, and we largely see this clustering of the transcriptomes in this PCA space. And what ended up being the kind of take-home from this analysis was not a particular pathway or a gene ontology. It was that out of all the genes we could see, all 18,000 genes, 75% of them were differentially expressed. 3/4 of the genes in a Woodruff and a Apopka are only 30 kilometers apart in a straight line, right? 32 kilometers, I think. So that was really remarkable, and to us was like, okay, well, that makes a lot of sense, because every gene we look at with qPCR, it does look like it's largely different. And so we're like, okay, well, yeah, you got a 75% shot. So Matthew took this picture, which I really like. And I like it because it's showing kind of the vulnerability of this embryo, right? It's developing, it's a stage 27 embryo, but you can see the yolk. And it's totally reliant on this maternal yolk. And this is old data from Lou with the contaminants in the yolk, and I'll show you some more here in a minute. But the idea is that mom is depositing these contaminants is kind of our working hypothesis, and that these contaminants are estrogenic. They're interacting with the estrogen receptor during gonadal differentiation, and it alters and screws up the ovary. Okay, so if that's true, we started this thinking more about mechanism and wanted to kind of confirm this hypothesis or see if we could test it. And what we did here is we did the same experiment. So we go out to Woodruff and Apopka, we collect the eggs, we bring the Woodruff eggs back, and we made a couple of observations based off of previous reports and some of our previous work. So one thing we knew was that aromatase is not really expressed in the ovary until stage 25. So this is kind of after the right at the tail end of the sex determining window is when you start to see ovarian synthesis of estrogen. But if you look earlier, the estrogen receptors are expressed much earlier. So these are the two estrogen receptors, estrogen alpha, beta. And so what we were thinking is that, well, perhaps these contaminants are actually interacting with these receptors before endogenous synthesis begins. And we kind of had this idea that if we treated with estrogen at this particular stage, stage 19, and this is Woodruff egg, so if we have Woodruff, we treat with this precocious estrogen, we should be able to recapitulate these phenotypes that we're seeing at Apopka. And so that's what we did, and I'll show you some results. So we dissect these, we turn into alligator farmers for 5 months. You dissect out the ovaries, sequence the transcriptomes, and Matt comes into the office and he's like, this is amazing. The single dose of estrogen is able to recapitulate 76% of the genes that are upregulated at Lake Apopka and something like 78% of the genes that are downregulated at Lake Apopka. So that single dose of estrogen during development was able to basically mirror the transcriptional patterns we were seeing at Lake Apopka, which was pretty cool. We also did some morphology work where we looked at the follicles. Matthew quantified the follicular density in the ovaries here, and we see this big difference, if I can find a pointer. A big reduction here. So this is Woodruff, and this is Apopka, and then this is Woodruff plus that single dose of estrogen. And so what this was suggesting, what Matthew's work was suggesting really was that the transcriptional and histological alterations that we were seeing in Apopka were likely due to altered estrogen signaling. And we thought that this is occurring prior to the onset of endogenous synthesis. But it's kind of always bugged me in the EDC field because a lot of these contaminants that are estrogenic are incredibly weak. They don't bind the estrogen receptor and really activate transcription unless they're at pretty significant concentrations. And this is some older data here showing that, comparing estrogen receptor activation across different species. But it provided this kind of hypothesis and started to make sense that it's these EDCs at Apopka that are providing weak but developmentally precocious signals that disrupt the timing of ovarian development. That's kind of what our hypothesis was at this point working. And so at this point, what we did is we were like, okay, well, if it's the timing, then we should be able to treat with estrogen before the ovary starts to make, synthesize estrogen. And we can treat with the same dose after the ovary. So this would be like the timing is kind of normal, and this is precocious. And we should see the difference. And I'm not gonna go into a lot of detail on this slide, but I just wanna show you, this is in embryonic ovaries, that the timing has a huge effect. So here, this is the early dose, and we can see we get 123 upregulated genes, and the late dose gives you 65, but they're largely independent groups. They're different genes. So the same dose, but at different times, gives you almost a completely different subset of genes there. Okay, so this was our working hypothesis based on all the data that we had at that point. And the idea is that moms are out in the environment eating, accumulating, and these contaminants, and then when she reproduces, she's depositing these contaminants in the egg yolk. Embryo is exposed, and you get the altered gonadal transcriptomes and follicular dynamics. But there's these other pathways, and if we zoom out, there's this rich kind of ecological literature showing that moms also deposit steroid hormones into eggs as well, and that these can have really dramatic effects on offspring phenotypes in both adaptive and maladaptive ways. And this led us to think about the potential contributions of steroid hormones. And then with this in mind, we were like, okay, well, if you are actually kind of thinking about both of these, then it's also possible that the moms could be exposed to contaminants, and those contaminants could alter her endocrinology and lead to her depositing different levels or different concentrations of steroid hormones. And so we were thinking of this as kind of this indirect effect here, where maternal exposure leads to altered maternal physiology, and so you get disrupted deposition of these steroid hormones. And so to look at this was work from Chris Smaga, who couldn't be here. He's has an interview, he's about to graduate and go to a postdoc. And what he wanted to do is to kind of look at all three of these. He wanted to look at contaminants, hormones, and the gonadal transcriptomes and see what all of those connections were. And so this was his kind of experiment here. So Lake Woodruff, you collect eggs, Apopka, you collect eggs, you bring them back to the lab. But instead of thinking about this at the individual level, think about this as a clutch, okay? So a female lays a single clutch of eggs, and what we did is we took some of those eggs and we measured hormones, steroid hormones in the yolk. Some of the eggs we measured contaminants, and then some of the eggs we measured gonadal transcriptomes. And then we're comparing across clutches, okay? So we're making those connections across clutches. And if we look at yolk hormones here, this is just some, this is from 2021, and this is, so that we're right in here. This is obviously not an alligator egg. But you see some interesting results here. So this is just comparing across Woodruff and Apopka. We see that Apopka has elevated 17-beta estradiol and estrone is the opposite pattern, and ediacalanolone is also elevated at Apopka. And when we look at contaminants, these are the OCPs, we kind of see the usual suspects here, a little bit of elevated DDT and endosulfan and a big increase in DDE and methoxychlor. And then we also looked at PCBs, which were elevated as well. Okay, so we've got all of these contaminants. I think at Apopka we had 7 clutches, and at Woodruff we had 8 clutches. So we then sequenced the transcriptomes from the gonads, and what we were wanting to do is a ton of correlations. And so if you've got 18,000 genes, you lose all your statistical power with multiple corrections. So what we did is we took the transcriptome and we reduced the dimensionality with this module approach. So basically what this approach does is it's looking at genes whose expression changes together, and it says, okay, all these genes are kind of changing together, that's a module. These genes are changing together, that's a module. And in doing that, you get these 10 different modules here, and that helps us with the statistical power, right? And so this was what all of those comparisons look like, but I want to look at it slightly different here. So what we saw is that if you look, these are the gene modules here, are named colors, just arbitrary. So you've got this green module here that correlates with estrogen, and you've got the magenta one that correlates with estrogen, and the pink one as well. And it turns out that out of the 10 modules, over half, 6 of 10 modules correlate with 17 beta estradiol levels, which is a huge effect. It's kind of surprising that you see that proportion of the transcriptome correlating there. We can also look at those modules that change with both contaminants and hormones. So we see here that you've got the black module that is changing with estradiol and endosulfan. And then we looked at this indirect effect here. And so in this case, what we're looking for is gene modules that change with contaminants and hormones, and that's what's happening here in this gray module. So you've got this module that's changing with 17 beta estradiol, and it's also changing with methoxychlor. So you've got a steroid hormone and a contaminant, and it's changing with both. And then on top of that, what we wanted to see is the contaminant correlated to the steroid levels, and in this case it was. So methoxychlor is what we think is influencing the deposition of 17 beta estradiol. And so when we take it, zoom back out and look at this, we've got this general idea that that single dose of estrogen early in development is able to recapitulate the Apopka transcriptomes. And then if we look at the other side, kind of zooming out and thinking about these indirect effects, that Apopka moms are depositing more estrogen into the yolks than they do in Woodruff. And we don't know exactly if that maternal dose is sufficient to drive the Apopka phenotype, but it could be. And we have some experiments that we can do to test that using the estrogen receptor antagonist. Anyway. All right, so I want to close with just a couple of things here. This is my favorite paper that Lou wrote. It's a single author. It's a monograph in EHP, and it's where the title of the talk comes from, which is Endocrine Disrupting Contaminants Beyond the Dogma. And it's beautifully written. If you haven't read it, particularly if you're a student, I would highly recommend it. It's just good writing, good communication. It's inspiring. And one of the, this is from the abstract here, and I'm going to just read it out loud because I love it. Futures, and think about this today. This was written in 2006 or published in 2006. Future studies must broaden the basic science approach to endocrine disruption. thereby expanding the mechanisms and endocrine endpoints examined. This goal should be accomplished even if the primary influence and funding continue to emphasize a narrower approach based on regulatory needs. Without this approach, research into endocrine disruption will become dominated by a narrow dogma focusing on a few endpoints and mechanisms. And to me, it's just like that is so relevant today more than ever. And it was 2006, almost 20 years ago, which I think really is a testament to Lou just having these ability to see into the future almost. All right. So, John alluded to this, and I started asking this question, what would Lou do? I asked it, the morning after Lou passed. I can remember going into the HML director's, Holling Marine Lab director's office and thinking like, okay, telling him, what do we do about everything? And I was just thinking about it all the time. I thought, I asked this question, about medical school politics when I was looking for other jobs. I was asking it about science, and I've asked it about my career since and my lab group and how you mentor. And the problem with this question is that the answer usually involves employing one of the many superpowers that Lou had, right? So his ability to inspire, his ability to communicate, and his ability to read and connect with people, and his ability to see about 5 more years into the future than the average scientist. And so as many times as I've kind of asked this question and pondered it, it's rarely been useful to a mere mortal like me, because I don't have those superpowers. And over the years, though, the time has kind of passed, this last decade, and I think I've become more comfortable with myself. I think and my abilities and who I am as a person. And I think I've realized that's not the right question at all. What would Lou do? I think that's the wrong question. I don't think that it was what Lou did and made Lou so special was like a single decision. It wasn't like you could ask this question and just have the right answer. It wasn't about a single decision or a particular circumstance. What I think that made Lou so special is that he created something, okay? I think, Lou created a community, and that community that's second to his family is his legacy. And we're all here, this community. And what I think is that we can each create a community that will be our legacy as well. And that's what's the powerful thing. That's what would Lou do is, well, he would create a community. He wouldn't just make he would create this community that will be our legacy. And then the question is like, well, looking at Lou's community, what were some of the characteristics? How did he do that? What were some of the principles that you can then apply instead of a single answer? And I've come up with 6 of them here, and I'm sure there's others, but these are the ones that came to mind for me. One is that you push yourself and you push others intellectually, but you don't ever over-intellectualize the relationships. When I interacted with Lou, we would, talk about big ideas and push each other intellectually, but our relationship has to be primarily built on trust and respect. And it's not only a scientist. That's not the only value that it's not a transaction of science only. The second thing was you say no as rarely as you can, and you empower those around you. Lou just didn't say no that often. I would ask him, can we buy this reagent? And it just is very rarely I can think of that he ever said no, and I think that's important. The other thing that Lou did was he stayed grounded in the science that connected him to the natural world. And kind of, thinking back to that first slide about the Drosophila testis, that's what I was missing, is I was not connected to the natural world. And that is what inspires us as biologists. I think it's really important that we stay connected to our inspiration. Credit is infinitely divisible. We've heard that several times, but that's a principle, right? It's not just about one paper or one thing. It's something that you operate with. And then we speak often and respectfully of your academic mentors and collaborators. We're talking a lot about Lou's legacy, but I also noticed that Lou would talk about his mentors a lot. Howard Byrne, Dick Jones, and his collaborators, Ty, Charles, Shauna Swan, etc. It was really common for him to tell stories, and they were never negative. They were always like this reverence, and I think it's important that we're doing that today, and that we do that in the future. So with that, I'm going to wrap up. I want to thank a few people. Chris and Matt, I want to acknowledge for the work that I showed from them, and everybody else in our group over the years. We go down to collect these eggs, and we show up with a permit that the Florida Fish and Wildlife folks give us, but they also show up with like 4 airboats and people and gas, and that's the work doesn't get done without them. So Woody and Arnold and Vinny and Andrew and Franklin was there in 2021, which is the data that I showed you from all the hormone and contaminant data. And Russ and Brenton have always been out there as well. Thomas, for all of our support at in collaboration at Yawkey, which I didn't show a lot of data today. And then Illinois State University, Ryan Pates is the one who quantified all of the steroid hormones. And then the NSF and DOE for some of our funding for this work. And with that, I'll close it out. Thank you.
So here we are. I want to thank you all so much. Thank you for the memories, the stories, the lessons learned. It's just been terrific. What a legacy Lou left for science, and especially the endocrine disruptor field. And in this time, communication is so important, and we can really learn a lesson from Lou, who could work in alligators and work in an OB-GYN department and talk to anybody about what he was doing and get it to their level so they could understand. We all need to do a better job of that. But, what he said himself was his legacy. My legacy to science is not just the work I did, that's just brick and walls. Your true legacy is the people you leave behind. And we heard from many of them in the last few days. So his legacy continues through you and your students and on, and very soon it'll be transgenerational, all the way to 3rd generation. What a beautiful legacy. So thank you all so much. It was so wonderful. I appreciate it. I hope you had as good a time as I did getting back together with your friends and colleagues. But if you bear with me 2 more minutes, Nancy and I were talking, and we think we should do something to write up something from this meeting. So we wanted to have you think about that and give us some ideas. I don't think we want a meeting report where we go through every speaker and what they said, but somehow maybe just a 1,000-word editorial or op-ed or something to indicate that Lou's collaborators and friends got together, talked about his impact on the science, and here's some lessons we learned, something like that maybe. So anybody have some thoughts for us about that? Maybe that's something you can think about, and if you're interested to help us write something, Nancy and I'll kind of take the lead, but we then want to spread it around to some of you and get some help. Yes.
When I got married, you all know that my kids were fairly small. And Lou was always Lou to them until we got married. And then he became Papa Lou. And in the lab, when they'd go up to see Lou, they would refer to him as Papa Lou. And soon his students were calling him Papa Lou. And this went on for years. And as I think about it, it's Papa Lou who left his family. His students were his family. And their students were his grandkids. And I see this now going on to great-grandkids. And I am very pleased that Papa Lou had such a great influence on everybody.
Message. It's the influence he had on his collaborators and his students, and how that has influenced their careers, and now it's going down the generation. I think that's maybe a wonderful story to tell. But I hope you'd be interested to help us to do that. And if any of you are specifically interested, have a little group of 3 or 4 of us that could have a couple of calls to talk about that, let me know. Okay, so with that, I think that's it. Anything else? All right, thank you all so much.